Hello and welcome. I'm joining you today from the land of the Ngunnawal people, the traditional owners of the Canberra region. The Academy acknowledges and pays respect to the traditional owners and their elders past and present of all the lands on which we operate, live and work. They hold the memories, traditions, cultures and hopes of Aboriginal and Torres Strait Islander peoples of Australia. Thank you for joining us today. I am Anna Maria Arabia, the Chief Executive of the Australian Academy of Science. Today, we have the immense privilege to hear from fellows elected to the Australian Academy of Science in 2020 and 2021. Traditionally, we would have been in the Shine Dome together to hear these presentations, but today I'm here in the Ian Walk Theatre with Academy staff spread across the Shine Dome to deliver this event in a COVID-safe manner. It is our hope that at some time in 2020, we can be together again here in the Shine Dome, the home of science, to celebrate scientific excellence. And so our new fellows can sign the historic charter book that has been signed by every fellow ever elected to the Australian Academy of Science. Even though things look a little different, we are so grateful to be able to recognise Australia's scientific excellence in this virtual way, with the knowledge that we can share the experience with larger audiences than ever before. We thank you, our audience, for joining us today, and we also thank our event partners who continue to support the Academy and have helped us deliver Science at the Shine Dome 2021 online this year. A big thank you to all our partners on the screen, but in particular to CSIRO and DST, our platinum partners. Now, before we hear from our new fellows, please welcome Academy President, Professor John Shine.
I too am delighted to be here today for the presentations from fellows that were elected in 2020 and 2021. I'd like to add my thanks to the event partners whose generosity has made this online Science at the Shine Dome possible and allowed it to reach even more people across Australia and around the world. As mentioned, today's event would normally be held in person at the iconic Dome in Canberra. Though the location is different this year, what remains is the same, is that we are here to recognise and celebrate the extraordinary scientists elected to our fellowship in 2020 and 2021, and to celebrate scientific excellence in Australia. They were certainly humble beginnings when 23 founding fellows gathered in 1954. I doubt that they would have imagined a day when 873 scientists had been admitted to this institution. With 46 new fellows and 4 corresponding members elected in 2020 and 2021, today we boast 573 living fellows with a further 33 corresponding members. And of course, we pay tribute to the contributions made by those who are unfortunately no longer with us. While the founding fathers were all men of Anglo-Saxon descent, the modern Academy is a diverse and vibrant fellowship of women and men aged from 39 to 97, highly regarded scientists who have already left a lasting impression in their respective fields. The Academy is its fellows, living and deceased, who in so many ways have fundamentally shaped Australian science. They volunteer their knowledge and their expertise to bring science to the service of the nation. I would argue that few, if any, other organisation in this country brings together such a distinguished group of individuals who have contributed so much to society and who have been driven by public good. They truly are a unique national treasure. It is the fellows who enable the Academy to achieve its objectives of providing independent, authoritative and influential scientific advice, of promoting international scientific engagement, of building public awareness and the understanding of science, and of championing and celebrating excellence in Australian science. Without our fellows, none of this would be possible. In fact, of course, the Academy wouldn't even exist. Our fellows are involved in and contribute to the Academy in many different ways. By serving on the National Committees for Science, which are the national standard bearers for scientific disciplines. By providing expertise and gathering scientific evidence to influence decision makers, the national dialogue and Academy activities. By contributing to the selection of new fellows and awardees, by representing Australia internationally, by ensuring the public can receive accurate and trustworthy scientific material online, in schools, or wherever they may be. This list is by no means comprehensive, as the range of activities the Academy leads and is involved in is very rich and diverse. Today, we're here to hear from 49 of these extraordinary people who continue to carry out, practice and advocate for scientific excellence in Australia. The annual New Fellows presentations are a unique opportunity to capture in the course of one day the depth and breadth of scientific excellence in Australia and to take a glimpse of the next frontiers of knowledge creation. I very much look forward to learning more about this work. So thank you all for joining us today to hear from our recently elected fellows, and we certainly hope you enjoy their amazing presentations.
Thank you, John. As mentioned, we thank our partners who have helped make this event possible. To now hear from one of our platinum partners, please welcome CSIRO Chief Scientist, Professor Bronwyn Fox.
Thank you to the Australian Academy of Science for the invitation to speak today. I'm so honoured to be part of celebrating the outstanding scientific achievements of this very distinguished group. I'd like to take a moment to acknowledge the traditional owners of the land I'm speaking from today, the Wurundjeri people, as well as the land you may be joining from, and to pay my respects to their elders past and present. I'm really delighted to have joined CSIRO recently as Chief Scientist and to continue the long-standing partnership between the Academy of Science and CSIRO. Both organisations share the common vision of using science to improve the lives of all Australians and the common goal of bringing the sector together to do things that none of us could achieve alone. We also both understand the power of networks and play vital roles in connecting Australia's innovation ecosystem. CSIRO, the bridge between research and industry, and the Academy brings together Australia's elite scientific researchers. This role is critically important as Australia and the research community contemplates the scientific breakthroughs that are required to support our recovery from COVID-19 and address enormous challenges like climate change and energy transformation. I'm proud to be part of the efforts at CSIRO to build future industries around Australia's innovation opportunities and to help mitigate threats like natural disasters, droughts and pandemics. But most of all, I'm passionate about bringing together diverse ecosystems of collaborators to solve fewer but bigger problems together. Australia has brilliant scientific capabilities that will ensure we have a bright future ahead, but only if we work together. We need to bring together the most brilliant minds to solve our greatest challenges, which is why events like Science at the Shine Dome are so important. We also need to embrace our early and mid-career researchers and give them an opportunity to hear from those who are at the top of their field, like the new fellows being inducted today. We are all responsible for nurturing that next generation of Australia's scientific leaders. Congratulations to all of the new fellows being honoured today, and thank you for continuing to push the boundaries of Australian science. CSIRO is very proud of our own scientists who have become fellows of the Academy, including 2 new fellows today. Wenju Kai, Chief Research Scientist in CSIRO Oceans and Atmosphere, and Jenny Stauber, Chief Research Scientist in CSIRO Land and Water. Congratulations also to my predecessor, Kathy Foley. She has been such a wonderful mentor to me and has left some incredibly big shoes to fill. She's now doing a wonderful job as Australia's Chief Scientist, and I'm really enjoying working with her in her new capacity and mine. Over what has been 2 years of immense challenges for Australia, science has been a steadying, guiding hand. Government and everyday Australians have looked to science to help weather a pandemic, and they can look to science again as we help to rebuild our economy while at the same time protecting our environment. There are some big opportunities for us, from hydrogen to advanced manufacturing, digital technologies, and many others, and your brilliant research and your networks will help Australia unlock this potential for future generations. At the National Science Agency, we are here to work with you because we value your expertise and your sage advice, and we know that we can only tackle these problems when we work together. Congratulations on this fantastic honour today. I'm just so thrilled for all of the new fellows. And if I can ever be of any assistance, please don't hesitate to reach out to me. Thank you.
Thank you, Bronwyn Fox, and congratulations on your recent appointment at CSIRO. Today, each fellow will present for 10 minutes, followed by a 5-minute Q&A session. Throughout the event, we strongly encourage you to ask questions. Please submit these using the Q&A panel on the right side of your screen. They can be submitted at any time, and we will try to get through as many as possible. Now, to begin the presentations, please welcome Academy Council member, Professor Ivan Marusic, to introduce our first fellow.
Our next presentation is from Professor Kevin Galvin. Kevin's research is in the field of mineral processing, where he has made seminal contributions to the behaviour of fine particles in laminar shear flows. His research led to the invention of the reflux classifier, an innovative separator for recovering and concentrating high-value minerals such as iron ore, chromite, coal, magnesium, and lithium. Now let's hear from Kevin.
I'd like to begin by thanking all those involved in my nomination, especially Professor Sam Tang, who first suggested the idea, and also Professor Graham Jamison, a colleague and a mentor who led the nomination. Well, my research sits very much at the intersection between science and invention, and it always helps to start with a good problem. And here we have the separation of particles on the basis of density in water, which is an age-old problem. And anything beyond air or water in the mining industry, of course, would probably be too expensive. So let's look at natural settling and imagine an elutriator where we have an upward current flow of water. And so what we have is a collection of particles of different size and density. And so to the right of the red line, you can see there's a combination, a mixture of fast settling particles, which would go to the bottom. And on the left, slower settling particles who would rise upwards. Again, a mixture. So we really do have a problem. It's a real challenge. Let's look at the settling of the particles in this tube. And they settle quite slowly. But a slight tilt of the tube has a dramatic effect. And I look to bring this effect, known as inclined settling, into a number of technologies, including a reflux classifier. And here, really, the pertinent question was, what should the channel gap be? And we started off with around 60 millimetres. And I want to return to this question in a moment. Let's look at the effect of surface roughness on inclined settling. And here we have a model particle with surface asperities. And you can see that these govern the interaction, the mechanical friction, but importantly, the lubrication force between the particle and the wall, which together govern the translation and the rotation of the particle. And one of our discoveries was that the apparent hydrodynamic roughness could vary by upwards of 2 orders of magnitude, depending on the angle of inclination. Leighton and co-workers did seminal work in the area of hydrodynamic roughness, but I want to talk now about this paper concerning this interesting experiment. And what we have is a rotating device containing a fluid with a stationary upper plate here. And the particle you see is positively buoyant. In other words, it wants to rise upwards. It's pressing upwards against that plate. And so the whole idea is to increase the shear rate or velocity gradient in the fluid to bring about inertial lift. And so with inertial lift, eventually the particle would start to, once that force exceeded the buoyant weight force, that particle would start to fall away and things would become purely hydrodynamic. And you can see that in the data, the dimensionless velocity versus what is effectively the shear Reynolds number. Initially, the dimensionless velocity is governed by hydrodynamic roughness due to mechanical contact. Once that contact is lost at the critical point, away the particle goes. And I look to bring that into the reflux classifier. And I can say that I was profoundly disappointed when I did the calculation, which told me that I would need a channel spacing of around 1.8 millimeters. Well, so we put that idea to rest for a while, but eventually we had to return to it. And here you have the system. So a collection of particles, silica particles, and we have a 1.8 millimeter channel. We vary the fluid velocity and we look at the particles that emerge out the top. I'd like to share with you the data. And as we increase the superficial channel velocity, you can see the separation size increases, which really brings me to a moment in my career, because we had a hypothesis and in terms of what we suspected would happen. But as is the case in science, until it does happen, will it happen? And this is what we see. Critical lift, very reminiscent of Leighton's experiment in a completely different system. And you can see a similar effect for higher density particles and for lower density particles. And here's the underlying physics. And effectively, we have a velocity field, which is parabolic. You can see the particles residing on the upward facing plane. And what we're doing is tailoring the fluid velocity to the particle size. This has the effect of diminishing the importance of particle size, which is ideal in a density separator. Unlike a conventional elutriator where you have one nominal upward current velocity. Secondly, when the lift force, the inertial lift force due to the velocity gradient exceeds the buoyant weight force of the particle pressing down, physical lift occurs, the particle moves to here or to here and is then subjected to much higher velocities and travels upwards, up and away. And the third factor in all of this, you saw it in the video, just how profoundly fast incline settling is. So here's the data, there's the theory, no adjustable parameters, and we have the discontinuity at the point of critical lift. The same at higher densities and lower densities. Again, same theory, no adjustable parameters. Contrast that with natural settling. And you can see that we can separate particles on the basis of density in water as long as we get the geometry right, we get the boundary conditions and the hydrodynamic forces right. Here's an example of a continuous steady state separation of an iron ore feed at 7 tonnes of solids per square metre per hour, which is really quite significant. And what we have is the feed producing the product hematite and a reject stream. And this is affected over a size range from 0 up to 180 microns. So you can see the clean sands here representing the full recovery of the hematite from the feed. Well, this is how the technology evolved over time. And I'm an academic and I don't do things like this on my own. This is called industry-based collaboration. And at this point, I believe there's around 180 units that are being deployed worldwide across many, many different commodities in more than a dozen countries, probably having processed at least 20 and probably upwards of $50 billion in raw material. Well, we've returned recently to the Valley of Death. And what we're interested in is the reflux flotation cell. And this is really an upward side down version where we are processing predominantly air bubbles as in flotation. And finally, this year, we got to trial it at full scale. And this is now the subject of a significant European grant worth around $8 million and a whole range of projects that we're working on. In keeping with our interests in trying to produce faster and more efficient separations, this oil agglomeration technology is also moving to full scale. And you can see when we add a binder, and this is a concentrated water in oil emulsion, separation is achieved in seconds. So we're looking forward to the upscaling of the technology next year. And finally, look, it's my absolute honour and privilege to lead the ARC Centre of Excellence, what we call COE Minerals. And this is about transforming the way we recover and concentrate minerals to make metals, which are desperately needed for the renewable energy sector, for the batteries, for electric vehicles, smart electronics, and so forth. It involves 8 Australian universities, 35 chief investigators, and our industry partners, as you can see. Finally, acknowledgements, and look, I'd like to acknowledge all those who I have had the privilege of working with over the course of my career, and collaborating partners, and also I'd like to acknowledge major sources of funding. Thank you very much.
Thank you ever so much, Professor Kevin Galvin. It is now our great pleasure to welcome Professor Galvin live with us here. Congratulations, Kevin, and thank you for such a stimulating presentation.
Thank you very much.
We have some questions for you. We ask our audience for any additional questions they may have. Please pop them into the Q&A on the right-hand side of your screen. I might get started with a few. Kevin, tell us a little bit about the sectors that your science is applied in. Where do you most see your science applied?
Well, I referred to the minerals industry, and our particular focus is on mineral processing, which is really about trying to recover the high value materials that are ultimately needed to make the metals. So this is a sector that's going to absolutely have to transform itself. Change is absolutely exponential. If we deliver the copper required for the electric vehicles that we are all hearing about, the potential impacts in going hard to generate that, could have environmental consequences. So we are looking at ways to radically reduce that environmental footprint, particularly around energy and water.
Yeah, of course. So presumably then your work contributes and will contribute to the circular economy going forward.
Yes, well, that's a very good point. Look, there's a really interesting dynamic there. When you're growing a sector exponentially, the point is when you produce copper and it goes into a building, you don't get it back for 50 years, or it goes into a motor vehicle, you don't get it back for 10 years. So with that kind of exponential growth, the only source for the copper to drive that sector, electric vehicles, wind turbine, is going to require mining. And what we are looking to do is to make sure that there are no unintended consequences in doing that.
Fantastic. And could the technology have applications outside of the minerals and mining sector?
There's certainly broad applications because a lot of these technologies utilise air bubbles, and air bubbles can be important in processes such as electrolysis, for example, in manufacturing hydrogen. And so there are also other important applications in water recycling and processing water. So the applications are potentially very broad, but one of the other interesting areas is looking to make use of what was formerly waste materials in mining. And there's a lot of sands and other potential building products that could come from the cleaning and classification of those particles.
Fantastic. Kevin, tell us, you took us through a presentation where you presented some of the basic science and then its applications and spoke about industry collaborations. What for you is the most satisfying part of your work? You've covered the whole spectrum.
Well, that's an interesting question. Certainly in the moment when you are doing science, when there is that hypothesis and there's that, you know, what's about to happen? Is it just what you normally see, or is there going to be something new? And then when it happens, that is an incredibly rewarding experience for all involved. So for me personally, fundamentally, it is the science. But sometimes these systems that you get involved in to create, you know, they're very interesting systems in their own right, but unless you can actually succeed in having them accepted industrially, they just remain as a bit of a curiosity. They don't necessarily go anywhere. And so completing the full journey is also important and also very, very rewarding.
Many of our fellows have described that moment of sheer excitement where you say, this is it. You just know there's something about this scientific discovery. You're looking at something that nobody else in the world has ever seen, or you've observed something that nobody else has, and you know it will go somewhere. It is really one of the most exciting parts about being a scientist. And then, as you've just described, to be able to work with others, both scientific collaborators and industry partners, is an enormous achievement. Well done. Can I ask you about some of the people you have worked with? You've listed a number of people and organisations in your acknowledgements. How important have they been to achieve your work? Tell me a little about that collaborative effort.
The collaborative effort cuts across through academia, through into industry. And so there's a wonderful experience to work with young people. They don't have to be young people, by the way, but students who are coming through the system, and to guide them, you know, on the joys of science, on the joys of discovery. That's extremely rewarding. There has been many of them, but the collaboration with industry is also quite interesting, and it's not always about people from the industry doing what academics do. They contribute in their own unique way, and that's the hallmark of collaboration, bringing together the capabilities that you need in order to achieve those goals. So, and so there is a journey that you go on, and it's good to have that journey with others.
Thank you. I have a question for you now from Professor Ivan Marusic. Thanks, Kevin. He says, brilliant application of fundamental science to a crucially important problem. Can you comment on how important it is in maintaining laminar flow, a challenge as the size of the device increases?
Right. Scale up is incredibly important. And the interesting thing about this system is that the primary length scale is actually the channel spacing. So that doesn't have to change as you make the system larger and larger. But certainly the 1.8 millimetre channel, we actually use in the industry something closer to 6 millimetres, and we're moving to 3 millimetres at the moment. There's another effect, which is where you have a concentration of particles, which actually permits a slightly wider channel spacing. But the idea of operating a system in a very aggressive environment in this sort of industry, 24 hours a day, 7 days a week, operating with such small channels was was a big worry in the beginning. That's why it took a long time to actually accept that was the way to go. But those, if you're using a tube or a pipe with those sort of dimensions, it will block very quickly. But if it's a planar channel, there's an extra degree of freedom where the particles can just go around each other. And so it actually works extremely well.
Thanks ever so much, Kevin. Unfortunately, that's all we've got time for today. Congratulations again on your election to the Fellowship of the Australian Academy of Science. Thank you.
Thank you.
Now, to introduce our next fellow, please welcome Academy Council member, Professor Veena Sahajwala.
Thank you. Our next presentation is from Professor John Sader. John has revolutionized the characterization of materials using the atomic force microscope. His scientific contributions have enabled breakthrough discoveries in material science at the molecular and atomic scale. He has made important contributions across nanomechanics, plasmonics, rarefied gas dynamics, and fluid structure interactions ranging from nano to macro scales. Please welcome John.
In the next 10 minutes, I want to give you an overview of my contributions to a much broader international effort that enabled routine atomic scale measurements in laboratories worldwide. My title refers to a relatively new instrument called the atomic force microscope, invented in 1986, which is a mechanical microscope. This schematic shows its operation. Basically, it's a sophisticated record player with a stylus, a cantilever, roughly 100 microns long, with a very sharp tip, a control system to scan the cantilever over the surface, and a detection system to measure the cantilever's motion. Remarkably, this laser can detect cantilever displacements far smaller than an atomic diameter. Hence, the AFM can image atoms. Of course, it's not that simple. Here's a ball and stick model of a pentacene molecule. It's very soft. If placed on a surface and scanned with an AFM tip, you can imagine the tip would just scrape it away. There'd be no image. But here's an actual AFM image of a pentacene molecule taken by IBM Zurich in 2009. It's the same as the ball and stick model. But this is a real image. So how do they do it? Well, they scanned in non-contact mode, away from the surface. When the cantilever experienced a force due to the pentacene, its vibrational frequency changed. So the image was not of force, but of frequency. You can see it here. But force is the real physical signal. So how can we recover it? My good colleague, Susie Jarvis, asked this very question when I was on sabbatical at Trinity College in 2003 in Dublin. So, over the course of a few months, I tried to solve this problem. And this is what I found. This formula involves a little mathematical trickery. It's not exact, but it is accurate for any force law. It just needs the cantilever's oscillation amplitude, its spring constant K. Now, that can be hard to measure, and I'll come back to that later. And the measured frequency shift. It then gives you the force. I formulated this method in such a way that it satisfied what experimentalists needed after spending a lot of time talking with them. Here's that study by IBM Zurich, where they reported the force at different atomic sites on the pentacene molecule. You can see it marked here. All these force laws have been recovered from the measured frequency shift using the method that I just showed you. Here's a sample of other studies that also use my method. It's now become an international standard. Importantly, all these measurements are performed in vacuum. But how is an AFM cantilever's vibration affected by fluid immersion? Obviously important for applications in biology. It turns out that fluid viscosity does not affect large cantilevers, but becomes increasingly important as you miniaturize. Well, back in the 1990s, no one had worried about viscosity because they were interested in big cantilevers for mainstream engineering. So I set about deriving the simplest, rigorous mathematical theory I could imagine that included viscosity and could explain the vibration of AFM cantilevers in fluid. Now, without going into detail, my theory needed the hydrodynamic force experienced by an infinitely thin, rigid blade oscillating in a viscous fluid. So I searched the literature back in 1997. This involved getting in my car and driving around Melbourne to various libraries. There was no Google Scholar back then, but I found it. An obscure but beautiful paper by Ernie Tuck that had only one citation by Ernie. Now, Ernie evidently thought his paper wasn't very useful, as you can tell from this quote. Although this is a reasonable viewpoint mathematically, it does, of course, limit severely the practical application of the results. The amplitude A has to be extremely small in order to prevent nonlinear convective effects from becoming dominant. Well, in AFM, we routinely use super small amplitudes, far smaller than an atomic diameter. So Ernie's supposedly useless theory was, after 30 years and one citation, extremely useful. So I incorporated Ernie's theory into mine for a cantilever. And here's the comparison of my theory with experimental measurements. The blue dots are the measurement of the vibrational spectrum of an AFM cantilever. The red curve is my theory. It works beautifully. This was very exciting because we could predict the position and the width of the resonance peaks, that is, their resonance frequency and quality factor in any fluid. So this made me think, could I use this new theory to devise a method to measure the spring constant, which was really hard to do because AFM cantilevers are so small. Experimentalists had asked me this question 7 years prior to developing this theory. Well, here's the method I devised in 1999 by simply measuring the resonance frequency and quality factor. That is, by looking at those resonance peaks, AFM users could easily measure their spring constants, K, without even touching the cantilever, completely non-invasive. Here's the experimental tests we did back in 1999. We took 3 cantilevers of different length. These were special and expensive cantilevers that had accurately specified spring constants by the manufacturer. Now, this is unusual. We then measured their frequency and quality factor in air just by looking at the cantilever. And here are their spring constants measured using the method I just showed you. It's totally non-invasive and completely accurate. Well, this ended up becoming an international standard with companies around the world incorporating it into their products. Again, this development was all driven by many chats with experimentalists. So, to finish, I want to especially thank all my collaborators who are predominantly experimentalists for being willing and offering to show me their work and spend time talking with me. This has driven virtually all of my work over the past 30 years. And of course, thank you to the Australian Research Council and the funding bodies of my collaborators. Thank you.
It's my great pleasure to now welcome Professor John Sader here with us today live.
Good morning.
Good morning. John, congratulations on your election to the Australian Academy of Science, and thank you so much for a brilliant presentation.
Thanks very much. It's a pleasure to be here.
We remind our audience to please send through your questions on the Q&A panel to the right of your screen, and I'll have an opportunity to ask those on your behalf. To get us started though, John, tell me, what do you love most about your work to seeing atoms using mechanics? It's an absolutely fascinating mathematical presentation.
Well, what I find most fascinating, it's been interacting with people who've asked me the questions which have really driven my thinking. So, when I was first introduced to the instrument, I didn't know anything about it. And this happens quite often. So people will introduce me to a problem they see in their laboratory and say, can you help us with this? They think that you can solve things because you're a mathematician. And so they show you their experiments, they show you their data, and then you go away and think about it. And sometimes you can come back quickly, and sometimes you come back years later, but hopefully you come back with something. And so the real fun part is the thinking and then seeing it being implemented and seeing the results coming out in an experiment. That's really very satisfying. And the interaction with people is really what's really the fun part in all of this.
Yeah. It's fantastic. You spoke about the Sader method and the Sader-Jarvis method. So what kind of applications, or in what sort of products, or where do you see your methods applied?
Well, the atomic force microscope is extremely versatile. It's used in all fields of science. You'll find them in many laboratories. So, good colleagues of mine are using them in biology. So I've got collaborators at MIT who are working on a special type of cantilever and measuring like the metabolic processes of cells, right? Other people are using it for electronics. It's used in the semiconductor industry. So basically what we did is develop methods which people could use in any fields. So really they're general techniques to operate the instruments. Yeah.
Fantastic. Yours is a great story of collaboration and building on the work of others. I really love the Ernie Tuck paper discovery and your journey to a library to find that, something that wouldn't have to happen today, of course. And that, what seemed like an obscure and not very useful paper became so central to your own method. Is it a case that some discoveries like Ernie's are just ahead of their time?
We hear about this all the time. So, it's a classic thing. I remember talking to Ernie after I did all this work, and he was surprised, that it was so useful. Ernie was a very, very smart guy. And if you read the paper, especially, mathematicians reading, we'll really see the quality in the work. It's really very high quality work. And now, even though I had to look recently, it only had one citation then, but now I think it's his third highest cited paper, right? So basically he was ahead of his time. And because technology had advanced to a point which caught up with Ernie's theory, we could then use it like in an instrument and then be of value to everybody. So,
Yeah, yet another reason why we shouldn't focus on citations alone when we think about work. We have a question from our audience. Congratulations, John. The Sader method appears an inspiring application of dimensional analysis. Do you think this method seems to be better. Do you think the method needs to be better promoted in science?
I mean, look, I don't know about being better promoted. I mean, it's a technique that companies have chosen to promote as being accurate and useful to people. I think the value in this comes about in its utility rather than in its promotion. So, I mean, I prefer to be seen to be used, like, rather than promote. I mean, some companies don't even mention it, but they use it. So I find great satisfaction in that. So, I mean, having your work used is really what it's all about, I believe.
Thank you. Another question, this time from Professor Frances Saparovich. She writes, the act of measurement changes the observable. Could you discuss how an AFM tip may be changing the surface that is being observed?
That's a great question. So, I mean, in AFM, you bring the probe. I mean, if you're doing atomic scale measurement, you bring the probe right down next to the tip, and that can actually perturb the tip. So you may think you're doing it in a non-invasive way, and many people have studied this from a theoretical perspective as well. So the act of doing the measurement can, in fact, perturb the result. So you have to keep that in mind. It's a convolution between the measurement and the actual thing you're trying to measure. So it's a bit hard to get away from that. And I've had this question many times in using AFM to do atomic scale measurements in particular. Yeah.
That's fantastic, John. I'm afraid we're already out of time. Congratulations once again on your election to the fellowship. Thank you so much.
Thank you.
Now, please welcome Academy Council member, Professor Paul Mulvaney, to introduce our next fellow.
Thank you. Our next presentation is from Professor Kate Joliffe. Kate's research is in the fields of supramolecular and organic chemistry, where she has made world-leading contributions to organic synthesis, anion recognition, and molecular self-assembly. Her transformative achievements include the development of selective receptors for pyrophosphate and sulfate ions, and new methods enabling the synthesis of macrocyclic peptides. Please welcome Kate.
As an organic supramolecular chemist, my research focuses on the design and synthesis of new molecular structures or architectures that have a specific function. In the same way that we can take a set of Lego building blocks and put these together in different arrangements to form architectures with different shapes, sizes, and functions, an organic chemist takes elemental and molecular building blocks, generally in the form of carbon and hydrogen, but with a smattering of other elements, and puts these together in different arrangements to give molecules with different shapes and sizes. It's the specific arrangement of the atoms and bonds in these molecules that provides them with their function. And so by rearranging the atoms, we can generate molecules with different functions. For example, we can build structures that are drugs like penicillin. We can make organic dyes for solar cells, the polymers that we use in our everyday life, and even molecules that are capable of lighting up subcellular structures, allowing us to visualize details within cells. At the University of Sydney, my group work on the synthesis of a range of different molecular structures with different functions. But one of the areas in which we're particularly interested is in the design of molecules capable of selectively binding to anions. Anions are any ionic or molecular species with a negative charge. And our interest in binding these is really a result of the fact that there are multiple potential applications for molecules capable of selectively binding to anions in aqueous solution. For example, in the environment, anions like nitrate and phosphate are responsible for the eutrophication of our waterways. And so the ability to detect their levels or to extract them from water has obvious applications. In industry, anions are often present in feed and waste streams, and sometimes that causes problems and they need to be removed. For example, in the nuclear industry, sulfate ions cause issues with the vitrification of nuclear waste, preventing it from being turned into a stable solid form. So the removal of sulfate ions from that nuclear waste would allow us to generate a much more stable waste form for storage. In biology, anions are everywhere. They're the starting materials, the intermediates, and the products of virtually every biological reaction, ranging in size and shape from small spherical ions like chloride through to large polyanionic species like DNA, which has a backbone containing phosphate derivatives. The ability to monitor the concentrations of these anions in a biological setting or to transport them from one place to another, for example, across a cell membrane, has applications in both the diagnosis and treatment of disease. However, binding to anions selectively in aqueous solution poses a fundamental scientific challenge. It's just really difficult. And so we aim to try and overcome this fundamental challenge through the synthesis and careful molecular design of structures that are capable of binding selectively to anionic targets. To do this, we construct molecular architectures that complement the size and shape of our target anion in solution, so that they will bind to this more strongly than they will to any of the other species in solution. We also incorporate a signaling unit into our molecular structure so that we can detect if a binding event has occurred. This signaling event might be a change in colour, or it might be a change in fluorescence. And you can see from these photos at the bottom of the slide that if we add a range of different anions, one to each of the tubes here, only one of them is giving us a change in colour or a change in fluorescence. And that means we're able to selectively detect the anion that we're interested in. One example that I think really illustrates the applications of this research is in the development of sensors for cell surface phosphatidylserine. Our cell membranes are built of a range of lipids, over 1,000 different lipids, in fact. Many of these have very similar structures, but there are small differences. You can see here on the right-hand side of the slide, the structures of some of the most common phospholipids present in a mammalian cell membrane, from phosphatidylethanolamine to phosphatidylserine, circled in blue here. It turns out that in our cell membranes, these phospholipids are not symmetrically distributed across the outer and inner leaflets of the membrane, but rather our cells spend a lot of time and energy maintaining an asymmetrical balance of these phospholipids. The reason is that these lipids act as cell signaling units. As an example, in mammalian healthy cells, all of the negatively charged or anionic phosphatidylserine projects towards the inner leaflet of the membrane, poking into the center of the cell. None of it is exposed on the cell surface. However, when cells undergo death processes, one of the first things that happens is that phosphatidylserine flips to the outer cell membrane. So the ability to detect this anionic charge on the cell surface gives us a means for detecting cell death processes. In order to detect this anionic phospholipid on a cell membrane, we need to come up with a nice design of molecular structure that will be selective. To do this, we've developed a molecule shown in cartoon form here that has a membrane anchor to help it insert into the cell membrane or keep it close to the cell membrane for binding purposes. We have binding sites for the anionic phosphatidylserine head group, and we have a signaling unit which only turns on once binding has occurred. For the chemists in the audience or those who are interested in the molecular structure, I've shown it on the right-hand side of the slide here. You can see our anion binding sites are the zinc ions, and the coumarin on the left of the structure in grey at the top and green at the bottom is our signaling unit, which only fluoresces when it's released from the zinc binding. That happens when another anion binds into the zinc, in this case, phosphatidylserine. We can look at this using fluorescence microscopy in cells and see that cell membrane in dying cells. So if we take some healthy cancer cells, these are HeLa cells, we can stain them with an orange stain that simply shows the interior of the cell. And you can see that in the top left panel here. In the central panels, we have the green channel on the microscope, which is the fluorescence of our probe. And on the right-hand side, we have the overlay. So in the top row, we can see there's very little green fluorescence showing up. This healthy cell has very little cell surface phosphatidylserine. In contrast, underneath that, we have cells that were pre-treated with an anti-cancer drug, camptothecin, to induce cell death processes or apoptosis. And we can see now in the middle bottom panel, the green halo that represents the cell membrane, very clearly the cell membrane, if you look at the overlay of the inside of the cell and the green channel. So by using our anion binders, we're able to visualize cells that are undergoing cell death processes. And this will be useful in monitoring whether cancer drugs are having their desired effect or not. This is a nice example of anion recognition requiring careful molecular design in order to generate a functional structure that does what we want. There are many other examples of this type of functional structure towards other anion receptors being generated in our labs, and also molecules with other functions. I want to finish up by thanking everybody who's contributed to my research over the past 19 years that I've been at the University of Sydney, early career researchers, HDR and honours students, and a group of fantastic collaborators without whom none of this would ever have happened. Thank you.
We are now delighted to have with us Professor Kate Joliffe. Welcome, Kate.
Hello.
And congratulations, Kate, on your election to the Fellowship of the Australian Academy of Science.
Thank you.
What a terrific presentation and such a wide application of your research. As always, we encourage the audience to ask questions by popping in questions in the Q&A panel on the right of your screen. I'd like to start by asking you, what drew you to the study of supramolecular structures?
I think I've always been interested in how molecules interact with each other and the beautiful designs of supramolecular chemistry where we can create structures that no one's ever seen before and really try and design them specifically to do something, which is the kind of exciting part, to see whether something you create can actually do what you think it should.
It's fantastic. There's actually so much art in it. It's this and design. And for me, I'm not a trained chemist. It does bring it to life. The visualizations bring it to life in a way that's particularly impactful and understandable. Kate, you mentioned the usefulness of your research for assessing the function of some cancer treatments. I presume you collaborate with cancer researchers. And are these sensors in use by other research groups?
Yes, we've sent these sensors to a number of groups around the world. Some of them are using them to analyse cancer drugs. Some of them are using them for completely different purposes. But we have a number of collaborators in different biological areas who are looking at them at the moment.
We have a question from Professor Francis Aparovich. Congratulations, Kate. Regarding the anion binding, how selective is it? Would it distinguish PG from PS? For example, bacteria have a lot of PG in their membranes.
Thanks, Francis. Great question. We have looked at the selectivity against different phospholipids, and for this particular probe, we get a much stronger response to phosphatidylserine than to phosphatidylglycerol. And we're actually looking at the moment at probes that would light up the other phospholipids in cells as well.
Thank you. I'm going to go back to the designing of structures. You spoke about designing structures. Can you pretend I'm a Year 11 student? Can you talk a little more about what designing a molecule involves?
Yes, so the first thing I think that you need to do is think about what it is that you're designing the molecule for. So for an anion receptor, for example, we know that the anions have negative charge. We know that therefore they're going to be attracted to positive charge. And we also know the shapes of those anions. So a sulfate ion, for example, is tetrahedral in shape. So we would try and arrange points of positive charge in a tetrahedral manner and connect those up with a structure of carbon atoms. And we need to make the shape and the size right in order to match that particular anion to get the selective binding that we're interested in.
Right, fantastic. And can I ask you to think about the future for a moment? What do you hope to see in the future of your research in the coming years? What are the up and coming projects and areas you're working on?
Yeah, so one of the things, as I mentioned, that we're looking for probes for different phospholipids. At the moment, as I said in my talk, there are 1,000 different lipids in cell membranes. Some of them have different lipid parts, the fatty chains, but some of them have different head groups. And at the moment, biologists don't really know why there are so many and what some of those phospholipids do. So being able to detect all of the different phospholipid head groups with different probes that shine in different colours would enable us to start unravelling those biological questions. And that's what I'm really looking forward to.
Oh, fantastic. Not unlike the fellows who have presented this morning, your list of collaborators and acknowledgements is vast. There are many people all around the world. Tell me a bit about those, particularly international collaborations, and how important they are to your research.
Yeah, so collaborations are hugely important to my research for a number of reasons. So we've collaborated with people with very different skill sets, biologists, biomedical researchers, to apply the molecules we've made. I need to actually go out and talk to biological researchers and find out what it is that they're interested in looking for in a cell, for example, to think about where I'm going to take my research next. I want to make molecules that are actually useful to people. So being able to talk to people and find out what they're interested in is one of the good ways of doing that. And it's also probably the most fun part of research, really, is actually interacting with other people. And I find that you always come up with better ideas when there's more of you in the room.
Yeah, there's no doubt. Kate, that's all we've got time for. I very much look forward to seeing you in real life, in person, here at the Shine Dome in the not too distant future. Congratulations again on your election to the Australian Academy of Science.
Thank you very much.
Now, please welcome Academy Council member, Professor Colin Raston, to introduce our next speaker.
Thank you. Next, we'll hear from Professor Alan Rowan. Alan is well known for his seminal work in diverse fields, ranging from single enzyme studies, single molecule magnets, organic solar cells, biomimetic catalysis, through to biohybrid catalysis, biomimetic gels, and polymer therapeutics. Now we'll hear from Alan.
Good morning, everybody. My name is Alan Rowan. I'm Professor and Director of the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland, and I'd like to thank the Academy for the honour of being elected a Fellow. As in everything, it starts as a journey, and this journey began in the 80s at the University of Liverpool, where I did a degree in chemistry and then a PhD in computational chemistry and NMR, whilst at the same time flying and being a pilot in the Air Force. This was a passion of mine, and it wasn't until I finished my PhD that I really realized my future lay as a professor, not as a pilot, because I'm not very good at taking orders. The journey then moved to the Southern Hemisphere, to Otago, the University of Otago, where I worked with a talented young professor directly out of Cambridge in the area of supramolecular chemistry. The idea of self-assembling molecules to give properties which are more than a sum of their parts. And then back to Europe for 22 years as assistant, associate, full professor, and then head of school in the Radboud University, Nijmegen in the Netherlands, where I studied supramolecular chemistry, self-assembly, and ultimately nanomaterials. At the same time, having a part-time position at the University of Stellenbosch, South Africa, to study polymer chemistry towards nanomedicine. In 2016, the University of Queensland called me and I became the director of the Australian Institute for Bioengineering and Nanotechnology. But this journey of science is really about people. And this is the myriad of postdocs, PhDs, and master's students who have been an inspiration to work with, who have taken the ideas I gave and turned them into wonderful science. And in particular to one person, Professor Roland Nolte, a mentor to me for many, many years, who was effectively a philosopher, not a scientist, an artist, not an organic chemist, who took such deep joy from trying to understand complex problems and trying to mimic them. And this really was what drives me, and it always has been. I've been a curiosity-driven scientist. How do things really work? And I can't help being inspired by Richard Feynman, who said, "What I cannot create, I do not understand." And paraphrasing Leonardo da Vinci, who basically said, unless you can fit it to a mathematical equation, you don't really understand it. And that's always been, can I mimic it? Can I fit a mathematical equation? And then I start to understand it. And to do exciting things at the interface of chemistry, physics, and biology. And as I said, I've been a curiosity-driven scientist, just looking at questions which intrigued me. The first one was single enzyme kinetics. We do so many chemical reactions in large flasks with billions and billions and billions of catalysts and molecules. But what is the property of just one? How would you do that? So in the early beginning 2000s, we started building new types of spectrometers, new lasers, new confocal microscopes, new chromophores, surfaces, and mathematics and kinetic measurements to study how does one enzyme work. And the enzyme we chose was actually a washing powder enzyme, found in many of you, a lipase, washing powders. But it turns out this enzyme just doesn't work, daily doing its duty. No. It does a few things and then falls asleep. 97% of the time, it does nothing. And we understood a whole new area of the dynamics of enzyme activity. And in the national newspapers throughout Europe, it was known as the lazy washing powder. But not only can we look at one enzyme, what's the smallest reactor we can make? Well, we can also make a reactor using viruses, a topic very prominent today, where we can actually take out the RNA, leaving this empty capsid, put an enzyme inside and make the tiniest nanoreactor of 20 nanometers. So we might measure in real time as the substrate comes in, gets converted, and then goes out, and study the kinetics and diffusion rates of reactions in tiny volumes. Not only can we study, as it were, catalysis, but also interested in the catalysis inside the body. This one particularly DNA exonuclease, which is a trimer of proteins, the green, red, and blue there, which run along DNA like a curtain rail, a ring on a curtain rail, running along in one direction, chopping up one strand of DNA. So it can be replaced and repaired. But how would you make that? How would you study that? How would you understand the mathematics? So we go back to the lab and we make catalysts which have a hole in, which can thread onto a polymer, and then they can run down the polymer, converting every single double bond until they get caught at the stopper. And to measure the kinetics and the theory, and develop a whole new theory about what's called processive catalysis. And not only once we could start to understand that, we could also start to mimic it by actually taking the natural building blocks from nature, modifying them with our own catalyst, such that now they would go and bind onto DNA and run down DNA doing different chemistry. So they self-assemble, bind onto DNA, and then run down until they come across a very, very sequence-specific part of the DNA and do one reaction and cut it like a laser in one place, bang. And then we can measure that in real time and also in high resolution using STM AFMs and see where each different dot here on the side is at 5 nanometers resolution, each different interaction. But it's not just catalysis I was interested in. I was also interested in simply self-assembling molecules to make wires where electrons could move down or energy could move down, to make a single wire 40,000 times thinner than a human hair to transfer energy, to build these into solar cells which are 20 times thinner than a human hair. So you could stack 20,000 of these solar cells on top of each other. And to work out how they all worked, we would study the properties of one. We would take one of these polymers and label it so it was dyed blue in amongst all its other polymers, a bit like a blue worm amongst all the other worms, and then measure in real time with the resolution of 2 nanometers how these individual polymers move. We had to develop new techniques, new mathematics, so we could actually understand how this works to prove for the very first time de Gennes' theory of polymer materials, and see that as this polymer moves, it gets trapped and then jumps out and then moves and then jumps again, forming this almost nano ballet in front of us. Intriguingly though, these polymers, when we were using them, we suddenly found that there's something we had not expected. We found that when we put them in the fridge, they would dissolve in water. And when we pulled them out, they would form a gel, almost like a reverse jelly you'd have at home. And more interestingly, they were super gels, i.e. if I took 1 kilo of this and put it in your swimming pool, on a sunny day, the whole swimming pool would become a gel. It turns out that the gel that it forms is just like the collagen in your body. And this became a really interesting development. Many, many companies around the world came to us to make these super gels, the powder that gelates swimming pools. And here's a picture of a cell. And so this led me to a whole new area called mechanotransduction. How do cells interact with the gels in your body? And this symbiotic relationship where the gels surrounding the cells controls the cell's behavior. And now together in Brisbane, I'm building a new team where we build new microscopes to allow us to look at aspects which had never been done before, but machines which allow us to look at zero gravity to study the effect of cells in space. We build new materials and develop new catalysts to allow us to self-assemble these gel-like materials and then study the mathematics. And once we know all of that, we can then apply it to problems. And the problems are, how can we seal wounds? Using a snake venom, we can control the healing so that we can actually seal a wound, not in 5 minutes, but in 30 seconds. We can make this wound by controlling the properties of the fiber network the cell's in, seal up without leaving you a scar. We can also control the gels of the cells inside your bone, which make bone, to understand what goes wrong with them, which leads to osteoporosis and aging. All this because we understand the basics. As director of the AIBN, it's a privilege to actually not be just about me, about others. 450 people where we tackle problems such as stem cells and aging, precision nanomedicine, advanced materials for energy, industrial biotech making vaccines and therapeutics, and applying it also to agriculture. But more importantly, working closely with the Indigenous community to try and translate their knowledge for their community. Working hard at gender equity and diversity, mentoring the next generation, and solving the big problems of today together. Less ownership, more leadership. Thank you to the Academy for the honour, to all the postdoc PhDs and master's students who allowed me to have this honour, my many other academic colleagues who have been an inspiration for many, many years. And I'll leave you with one quote again from Richard Freeman. Fall in love with some activity and do it. Nobody ever figures out what life is about, and it doesn't matter. Explore the world. Nearly everything is really interesting if you go into it deeply enough. Don't think about what to be, but what you want to do. Thank you.
What a wonderful presentation. It's my absolute pleasure to welcome live with us Professor Alan Rowan. Hello, Alan.
Hello, thank you.
And congratulations on your election to the Fellowship of the Australian Academy of Science. I greatly enjoyed your presentation. You're not just a distinguished scientist, but a terrific communicator, and I thank you for that. There were some very important life lessons in your presentation, but we'll come back to those. I invite our audience to ask questions by popping them in the Q&A panel on the right-hand side of your screen. I'd like to start. You spoke of the many applications of nanoscience and your work in mechanotransduction, including scarless wound healing, osteoporosis, agriculture, nanotechnology, and precision nanomedicine, just to name a few. Can you share with us how your basic science translates to these different areas, and if your main collaborators are other researchers, or do you work closely with industry to achieve this?
Well, it varies on the topic, and in my own personal journey, I mean, I've worked with industry. I've set up spin-out companies to try and take it from just the science through to a bigger area where more people can apply it, and more recently work a lot with clinicians. So I always come back to the basic principle. You know, you can sometimes make things, then you go to clinicians and say, can you use this? I say, well, we sort of understand this. What's your problem? How can we apply it? So I tend to do more the reverse, and I think Kate beforehand mentioned about listening. And it turns out it's a lot better if we start to listen, then we can actually deliver things for impact a lot better. And I think we're not always good at that as academics because we tend to love our own area. but when we start to listen a bit more, we find that we actually translate it better. So industry, with clinicians to try and apply it to a variety of different areas, and all of them look very promising.
Yeah, so very much that co-design when you want a mission-driven research project.
Yeah.
I was fascinated by this concept of nanowires and stackable nano solar cells. What benefits does a material gain, or what properties is it afforded by being made of optimized nanoparticles?
Well, I mean, the nano, by being able to define things at a better level, you're able to just get better quality. You're able to correlate the relationship between the architecture and function in a much better way. I always use the analogy, nano is just a size, and all that it means with all the nanotechnologies we have now, which allow us to see things at a smaller size, it's simply like putting on a pair of glasses, which I certainly need now at my age. And so I can see things better, so I can understand the function and the relationship better, so I can design better. So all of that work at the beginning was really to try and understand the fundamentals, which have then been picked up by many people in the future to design better devices. So,
fantastic, thank you. Professor Roland Nolte was your mentor, and he clearly left a very deep impression on you. And may I suggest you have inherited many of his traits. You described him as a philosopher, not a scientist, and an artist, not only an organic chemist, a true Renaissance man, it seems. You then spoke of a nano ballet, which I loved that idea. It really evoked for me a very clear sense of what you were saying, and reminded us to think about simple things and what we want to do, not be. You've clearly done this throughout your career. How important has it been to you to surround yourself with mentors, and do you mentor others?
Well, sometimes you don't realize the value of a mentor till sometimes later, but then you see the impression. And absolutely, when you realize that relation, and that's also hit me when I became director here, it's no longer about me. Everything I do is for other people. And that's a really refreshing side, because then you realize the importance of mentoring and sponsoring, the difficulties and the struggles that the young EMCRs go through, and how can we help them. But I always try to come back to probably that Renaissance type concept to say to people, love it for its beauty, create things which have not happened before. And when you go into it in depth, you're going to find joy.
Fantastic, thank you. And as much as we can do to assist our early and mid-career researchers, the better, I say.
Yes.
So do you find that making time to understand Indigenous knowledge and to invest in equity and diversity and mentoring others not only deepens your knowledge, but also improves the scientific results?
Oh, absolutely. I mean, it's something which has certainly enriched me coming to Australia. And I think we have to empower everybody, gender equity and diversity, to have the ability to achieve what they're capable of. And I think we don't always do that. There is always bias in the system, and we're moving away from that. But simply, I just want people to have the chance to do what they want to do.
That's
and I see a huge improvement in the culture and the sense of being and the sense of challenge. They take on more challenges because they feel supported. Absolutely.
We're so thrilled to have you in our fellowship. I think the life lesson or guide of less ownership and more leadership is spot on. Thank you ever so much for your presentation. I look forward to seeing you here in Canberra at the Shine Dome. Thank you ever so much, Alan.
Thank you very much. I look forward to it. Thank you.
Thank you. Now, please welcome back Professor Paul Mulvaney to introduce our next fellow.
Thank you. Next, we will hear from Professor Alison Rodger. Alison invented 4 techniques that use polarized light to reveal the structures of complex biological molecules. She has also improved techniques to extract extra information about these biomolecules. These approaches are particularly valuable in the development of biopharmaceuticals. She has successfully commercialized her inventions, which are now widely used by major companies, and have significantly changed the biopharmaceutical research and development process. Now to Alison's presentation.
Good morning, everybody. It's a pleasure to be talking to you, but sad that we're not together in person, as I'm sure many people have already said. What I want to do in the next few minutes is just share a little bit of my world, which I've entitled Imagining Molecules. So I imagine particularly chiral molecules, and I think about how they perform and interact in the world. And I just want to take us back about 20 years to when the Engineering and Physical Sciences Research Council in the UK funded doctoral training centres at the Physical and Life Sciences interface. I led the Warwick bid, which we entitled Molecular Organisation and Assembly in Cells. And it was a really exciting programme where we took first-class mathematicians and physicists and chemists and biologists, trained them as a cohort to really think in the languages of all those different disciplines. After about 3 months of a really intense programme, it was so exciting. I could see it working. Strangers couldn't tell the first discipline of the students. And so that was great. But a little bit later, after 12 months, I realised that although I personally could do mathematics and physics and chemistry and biology, somehow my neural structures seemed to have got fossilised, and I could only see the world in terms of its molecules. I was stuck being a chemist. So within my chemical world, where am I? I love crystal structures and microscopy images, but my goal is to really understand how molecules function in their native environments, with biomolecules and their assemblies being my absolute obsession. I use quantum electrodynamics theory to understand how light interacts with the molecules, and this has led to inventing new polarized light spectroscopy techniques. And then when I get data, I try to work backwards to see how molecules are interacting based on the data. As a techniques-based person, I've got a lot of wonderful collaborators and who've come with interesting molecular systems, but probably my most common comment is, oh, I didn't know that. And I think that's probably one of my important lessons is it's okay not to know as long as you're prepared to learn. And it leads me to science is really as much about people as it is about data and experiments. And here's a list of the few people who've been really important to me. And I think collectively they're inspiring, but they also completely failed to understand what I felt my limitations were. And at this stage, all I can say is thank you to all of them. And so where did I begin my macromolecule career? I started off with flow linear dichroism of DNA, and I built a cell with the help of one wonderful technician in Oxford, but it required 1.5 to 2 mils of sample. And my biology friends just wouldn't share that much sample with me. So I went away for about 3 years and eventually reduced the volume by about 20-fold. And I also set it up so that I could do fluorescence detection, which then takes you into the sensitivity of fluorescent assays. So what can I measure? If I just measure absorbance, this example of DNA and ethidium bromide, I get everything that's in the solution. When I start flowing the sample and measure linear dichroism, which is a differential absorbance technique, I only measure the DNA and the things that are bound to the DNA. And then if I further restrict things by only counting emitted fluorescent photons, I only detect the fluorophores. So I go from measuring everything to measuring only the ethidium bromide that's stuck on the DNA. And that really does open a lot of doors. But the trouble is, when I moved to Australia, I bought a new generation of instrument, and I'm absolutely convinced there's some problem with the electronics that some engineer thought was a great idea, and the experiments just aren't working as well. So somehow we're always learning and always having another go. So where are we? One day I was just focused on DNA, and I gave a talk about DNA, and Tim Dafford came up to me afterwards and said, "What about proteins?" And I said, I don't know, but clearly he did. So we said we'd give it a go. And he collected a whole lot of fibrous proteins, and we put them into my linear dichroism cell and instrument and got beautiful data. We then moved on to studying bacterial cell division, particularly via the fibrous protein FTSE that constricts the center of the cell. And we can measure the orientation of the guanine chromophore that is a key to a lot of the activity of FTSE and the backbone. And then we could measure the kinetics of how it was forming and see what it was forming with microscopy, and really got a good idea of the dynamics of what was happening in cells. So what are we doing? We're trying to see what the molecules do from a sort of noisy, scrabbly line on a piece of paper. And I confess, I turn it into pictures that are not much more sophisticated than thinking of supercoiled DNA is a bit like a ball of string. Linear DNA might be a bit like spaghetti. And then when we cut the linear DNA, and so it doesn't orient as well and we lose our negative signal, then I think of it as chopped up spaghetti. But that actually works and really does help us understand molecules. So Tim particularly has done a lot of work on M13 bacteriophage. The basic M13 is a wonderful chemist's cell, and you can actually do a lot of chemistry on the outside with the bacteriophage still staying intact. So it's DNA in the middle, proteins around the outside, and our spectroscopy can probe the DNA and can probe the proteins, and we can find out what's happening in that M13 cell as it forms fibers, interacts with other things. Where's one of our challenges now? Membranes. They're not really the ideal chemist's biology system. They're far too complex. So we combine lots of different techniques and we can extract information and we can find out mechanisms, for example, of peptides and proteins binding to membranes and inserting using spectroscopy and getting to simplistic molecular pictures that help us understand. So where next? I love electronic spectroscopy and I've worked with it all my career. But the trouble is it doesn't do everything, particularly when you're working with biopharmaceutical products at really high concentrations. So we've moved into the infrared regime. And there we're just starting to work on the film linear dichroism of fibrous proteins. And over on the left here, I've got the most beautiful picture of our first fibrous protein that we measured. We get a beautiful positive peak for the amide 1 band that goes along the carbonyl and a negative one for the transition that's perpendicular. Then my collaborator, Paul Wormel, went rummaging in the freezer and found hemoglobin and thought, well, if those peptides work, why not hemoglobin? And I wasn't there to tell him don't do a silly thing. And what we found is that we really don't understand how hemoglobin molecules bind together. And so our challenge at the moment is why are both the transitions positive? Wherever you go, do one extra experiment and you get stuck and you've got a question that takes a lot of effort to understand. But it's exciting and it's a journey. I thought I'd just finish with a final comment on my applications of chemistry to women in STEM. Ever since I first realised that girls had a tendency to avoid maths and science, and I'm not going to go into why that is so, I've worked to create opportunities to convince us all and the rest of the world that we can do it if we want to. And a lot of this is because I think everybody needs some scientific confidence in order to understand not only the wonders of my molecular world, but also to make sensible decisions about whatever the current challenge of the day is. And I think back to the early days of Athena Swan in the UK. They were really exciting because it was all about using data, analysing, and then acting to change things. And it led me to a whole new world of learning to actually respect qualitative as well as quantitative social sciences research methods, which made me think about my chemistry. It's precisely what I do with molecules. I take spectroscopic snapshots at times and places, then I analyse, and then I try to understand their behaviour. So it's all a small world and disciplines integrate with each other on many, many levels. Thank you.
Please now welcome Professor Alison Rodger. Congratulations, Alison, and thank you ever so much for such a wonderful presentation and for bringing us into your world.
Thank you.
It's such an honour to meet you and to have you as part of our fellowship. I have some questions, one straight up from the audience, from Professor Francis Zaparovich, who writes, congratulations, Alison. I'm so excited about in-cell NMR. So would like to ask you more about CD with cells. You mentioned bacteria. How do you cut back the light scattering? And is it possible to pursue with larger cells?
The answer is yes, but you have to do things to get rid of the scattering effect. And you can do that a combination of experimentally and theoretically. But I have to say it's part of the reason why I've gone to the infrared, because the scattering is much less. I love spectroscopy, great techniques, but in general, one technique won't give you the answer. And I'm sure Frances knows this because she's thinking NMR and then how do I do other techniques? We need to bring lots of different spectroscopic measurements that tell you about different parts of the molecule in different ways. Bring the ideas together to give us a picture that lets us do the next stage. And there'll be errors in our picture, but at least we can then move on and ask more questions. So I'd love to try some more, Frances. My whole cell experiments have been a mixed success to date.
Fantastic. Thank you for that. You said you're moving towards the infrared regime and fibrous proteins. Where do you think that would lead? I know there are many areas you probably haven't imagined yet, but at this stage, where do you think it will lead?
I think it's probably in the overlap between materials science and biology, because increasingly, and we've heard it this morning, people are more and more controlling what biomolecular systems do. Synthetic biology is a really exciting way of turning biological systems into chemistry factories. And then we need to have ways of assembling molecules and organizing them to do our other applications. If the synthetic biologist can get cells to produce things, then what do we do with it after that to make it into the kinds of materials we want? It's little steps at a time. And often I just think I don't know where we're going, I'm confused. But at least if you can start to formulate that question, you can then begin to answer it.
Yeah, one step at a time. Thank you. You spoke about understanding the language of multiple sciences. What advantages has that given you?
I think it's given me the confidence to go and find experts often. Sometimes I can do things myself, but it's always a really good idea to check with someone who really is the expert in the field. And I think if I hadn't at least felt I had a start on the languages, I wouldn't have gone and found an expert optics physicist when I was having trouble understanding some of the infrared spectroscopy I was doing. But at least it gave me the confidence to think, yes, I can ask questions. Once you can ask a question, you can usually find somebody to answer it. And there's a confidence of communication.
It's almost a matter of scientific literacy to be able to have that discussion.
Hands.
Yeah, that's right. You mentioned your work promoting science and maths to young women and girls. Thank you for that. Where do you think your own scientific confidence came from at a young age?
I think, first of all, I didn't realise there was an issue. I just got on and I was very conscientious and I did the homework I was given and it didn't occur to me not to do it. So therefore, I just did all of it. And when I moved to Sydney, I went to North Sydney Girls High. And the wonderful thing about all girls schools is that if there is a physics class and there is a mathematics class, it doesn't occur to you that it's not designed for girls. And I've got many friends who went to mixed schools and I think they faced a much greater battle because when you look and you see that the physics class is nearly all boys, you might say, well, maybe that's not for me. But at an all-girls school, I think that really, really helped. And then later, my husband also didn't seem to understand that I couldn't do things. And that was really, really helpful. So I think it's friends and people and those very subtle messages.
Excellent choice of a life partner, might I say. Surround yourself with people who believe in you, I think. Look, there is certainly evidence that girls don't study physics and maths, but no evidence whatsoever that they are any less able to. So if there is a message to any women or girls listening today is back yourself. There's no reason why you can't do this. If you're interested in it, go for it.
Absolutely.
You have a very positive approach to challenges and setbacks and see them as an opportunity for growth and learning as something new. And I think perhaps in Australia, we don't have enough of a positive culture around failure in inverted commas. How do you think this attitude has helped you in your work and in moving across different disciplines and applications?
I think it really helps, because if you've got a track record of remembering that last time you did make it work. Now, academics tend to forget the past success and move on to the next challenge immediately. And so I very consciously, for myself and for others, try to get people to stop and celebrate what they have actually succeeded, so that when they hit the next barrier, because without a doubt you will, your paper gets rejected, your grant doesn't get funded, somebody disagrees with your idea, it's tough. And something needs to keep you going, and it's partly past track record, and it's partly, in my case, that personal passion to understand, and great friends and colleagues.
Fantastic. Thank you, Alison. I'm afraid that's all we have time for. Congratulations once again on your election to the Australian Academy of Science.
Bye-bye.
Now we'll take a short break and be back at 10:45 a.m. for session 2. Welcome back to Science at the Shine Dome 2021, where we're hearing from fellows elected to the Australian Academy of Science over the years 2020 and 2021. Don't forget, you can ask our fellows questions throughout the event by using the panel in the portal where you are watching. Now, on to our next presentation. Please welcome Academy Vice President, Professor Malcolm Sandbridge, to introduce our next fellow.
Thank you. Our next presentation is from Dr. Wenju Kai. Wenju is a world authority on the climate variability, ocean dynamics, and ocean-atmosphere interactions. His work has revealed the links between climate change forcing factors and changes in the global ocean circulation, extreme climate, and extreme weather, including Australian drought and bushfires. His work has led to fundamental improvements in climate simulations and climate projections. Now we hear from Wenju.
Welcome to this presentation. Today I'd like to talk about El Nino and the Indian Ocean dipole in a warming climate. I hope to give you an overview of what my group has been doing. Australian climate is complex. It's under the influence of several important drivers. We have the southern annular mode, or the same. When sea level pressure in mid-latitude is higher than normal, it is lower than normal in the high latitude. And that high sea level pressure takes weather systems that deliver rainfall to southern Australia to the ocean. So we miss the rainfall. This pattern has been trending up due to ozone depletion and increasing carbon dioxide. To the west, we have the Indian Ocean dipole. When the Eastern Indian Ocean is cooler than normal, Western Indian Ocean is warmer than normal. The low temperature moves convection and moisture away to the Western Indian Ocean. Moisture that supports rainfall over southeastern Australia comes from this region. So we have drought conditions during a positive Indian Ocean dipole event. To the east, we have El Nino. When the Eastern Pacific warms enormously, convection and rain bands shift eastwards. Western Pacific countries and Eastern Australia experience dry conditions. So these are the three modes of climate variability that affect our climate extremes. Much of our work is on how the IODs, El Nino, La Nina, or ENSO, respond to climate change. We use sea surface temperature, or SST, to define these events for the IOD is west minus east. This is the time series for the index over the past few years. We can see two consecutive events, 2018, 2019. The 2019 is one of the strongest since 1900. For El Nino, we use SST in the Eastern Equatorial Pacific, or Nino 3. Nino 4 is for La Nina combined. Nino 3-4 is for ENSO cycles, evolution over the past 70 years. And we see those extreme El Nino events. Importantly, we got two consecutive El Nino events, 2018-19, and these conspire with the IOD, leading to the Black Summer bushfire that was devastating. We can also use rainfall to define these events for impact assessment. El Nino's impact is global in scales. In the Western Pacific, we had drought, bushfire, whereas in the Eastern Pacific, Ecuador, America, they have flood. So the impacts extend to agriculture production, public health, fishery, and biodiversity. IOD's impact is also broad. While we were having the devastating bushfire, African countries were suffering from floods, landslides, and malaria outbreaks. Many people were affected. Because of the vast impact, how the IOD and ENSO respond to climate change is one of the most important scientific questions in climate change science. Now, climate is changing and is projected to change further. IPCC projects a 5-degree increase in global mean temperature from the pre-industrial level. Has the climate change affected ENSO and the IOD? This is a very tough question to address. We look at 120 years of evolution of ENSO, El Nino here, and La Nina. And these are extreme El Nino, extreme La Nina, the black dots. We see that the post-60 period has seen some increases in extreme El Nino and extreme La Nina. Because this is the only one realization of the real world, we have no way to exclude the possibility that these increases are not due to climate variability. We can address how ENSO and IOD will respond to climate change using climate models under projected increase in carbon dioxide. And IPCC provided several emission scenarios. And the business as usual, there is no intermodal consensus for many generations of models. For example, El Nino, the variability, some projected an increase, some a reduction, no consensus. These kind of approach, however, assumes that models, ENSO and observed ENSO, could be depicted by the same index, assuming they have the same anomaly center. That is not necessarily true. That's not necessarily true. While model could produce reasonably good statistics of ENSO, the anomaly pattern could be very different from, as can be seen from these two models. And if we look at SST variability at the anomaly center unique to each model, there is intermodal consensus. Comparing the variability in 20th and 21st century for ENSO, you can see that majority of models produce an increased SST variance at this anomaly center, and that is due to enhanced coupling in the ocean and hemisphere, between the ocean and the hemisphere in the tropical Pacific. And the increased variability translates to more frequency of extreme El Nino and extreme La Nina. We can look at rainfall. Those are the extreme El Nino events. So in the Eastern Equatorial Pacific, this is a rainfall. Rainfall and SST have a non-linear relationship. So when SST surpasses a threshold value, rainfall increases dramatically. IPCC are projected the Eastern Equatorial Pacific is warming faster. And because of that, it's easier to shift convection. So even if sea surface temperature variability does not change, because convection tends to follow where the maximum temperature is, so these pattern of change translate to a large increase in extreme El Nino and extreme La Nina using rainfall definition. We can say the same thing about IOD. We can separate moderate from extreme, and moderate IOD tend to decrease in variability in majority of model, but extreme IOD increases in variability, translating to more occurrences of the extreme IOD that we see in 2019. For rainfall, similarly, we are now comparing rainfall IOD. We are now comparing rainfall IOD, extreme IOD, in the 20th and 21st century in 23 models. Each red dot represents an extreme positive IOD event. You can see that in the 20th century, the extreme IOD frequency is almost triple as often as the 20th century. So, as I mentioned, the climate is projected to change further. IPCC is projecting that without taking any actions, we may see an increase in global mean temperature by as much as 5 degrees C, and that would take us back to the climate of 50 million, 60 million years ago. And with the changing mean climate, it would exacerbate further the impact of extreme El Nino, La Nina, IOD, which themselves are projected to increase in frequency and extremity. It is certainly not a world we like to see. Thank you very much for those who are interested. I listed some of the paper that we published recently. I thank you again.
Thank you ever so much, Dr. Wenju Chai. It is a great pleasure to have you with us live this morning. Welcome and congratulations on your election to the Fellowship of the Australian Academy of Science.
Thank you. Thank you very much.
Thank you for that fascinating presentation, and a very timely one, given that many of the world leaders have gathered at Glasgow and are just winding up their deliberations about this very issue. I ask our audience members if they have any questions to pop them in the Q&A. May I ask you about your own science and scientific interests? When did you first become interested in studying climate variability?
When I was a boy or a young man, we had a farming family, and climate variability is actually very important for us. Being able to predict it's going to be a good or bad year is very useful, and the same applies to here right now in Australia. And so, particularly in Australia, we are so much affected by different kind of modes of climate variability, and that's where the interest starts from. Yeah.
It's fantastic, and really that application is still alive and well today, isn't it, for our agricultural sector and well beyond. Thank you. Your work is largely focused in the Southern Hemisphere. Are there corresponding climate indicators that are studied in the Northern Hemisphere, and do you collaborate with those people, with those scientists collecting that data?
Oh yes, so this sort of climate research is really global, and much of our work involves collaboration with many people around the globe. In the Northern Hemisphere, they are relatively more research and more observations. And so in fact, a lot of climate research actually started earlier and much more comprehensive in the Northern Hemisphere. And that's why a focus on Southern Hemisphere is really important, and it also can generate a lot of more exciting new things that people have not discovered. Yeah.
Thank you, Wenju. I'll just remind our audience, if you're following the schedule, Wenju Chai is back with us, and you're able to ask questions by putting them in the Q&A to the right of your screen. Wenju, where do you get the data from to inform your modelling of climate variation? I imagine there are many sources. Take us through some of that modelling and data collection.
Right. So our modeling really comes from the IPCC processes. So IPCC has this report, assessment report, and each time when we had a new report, they have done a lot of work to generate a lot of data. So right now we have some 60 climate models in the world, and these models are forced under historical forcing carbon dioxide, ozone depletion, and so on. And then also with projected a possible emission scenario into the future. And that allow us to look at, to compare climate variability, such as El Nino or Indian Ocean dipole, which are so important to Australia, may change into the future. And of course, the climate models need to be assessed using available observations. And so in the ocean, we had different kind of products of observation from satellite, from autonomous flows like Argo flows, and also from all sort of reanalysis and proxy data. And those are really publicly available to all scientists, and they are heavily used.
Thank you, Wenju. Look, one final question, and it may seem like an obvious one. Give us a bit of an insight into your future directions, or where you hope the future of your research field lands. I know it's a very critical issue at the moment, and we've all been particularly interested at looking at the outcomes of the Glasgow COP26 summit. Tell me from your perspective, what does a future research landscape look like?
Right. So future research has a lot of areas in this very important sort of field. And so we are hoping to understand uncertainties in our projections. At the moment, I talk about, we have so many climate models, 60 of them, and each of them are projecting a little bit different in the future climate. Although most of them are saying that the climate extremes are going to increase in frequency and intensity, but there is a huge range in that projections. And whether that is due to a set of butterfly effect stochastic processes, I heard people talking about the importance of the stochastic processes. All that is, it definitely is dependent on the emission scenarios. The less we emit, the less risk we will face in the future. But there are a lot of internal uncertainties that we hope to pin down to really narrow it, to reduce that uncertainty.
Thank you, Wenju. I am so delighted we were able to get you back online and speak about your work. And I also very much look forward to working with you as we take the scientific evidence base and use it to inform government policy in such a critical area. Thank you, and congratulations again on being elected a Fellow of the Australian Academy of Science.
Thank you so very much. And it's a great honor. And I'd like to thank all my colleagues, my collaborators. Without them, this would not be possible. So thank you.
Thank you so much. So now, to introduce our next fellow, please welcome back Professor Malcolm Sandbridge, who will introduce the next presentation.
Thank you. Our next new fellow presentation is from Professor Dorrit Yihong. Dorrit's innovative and world-leading research exploring shallow and deep carbon behaviour is unravelling the underlying mechanisms of carbon mobility, sinks, sources, and speciation at nano to global scales. She has pioneered research in Australia in the emerging field of biomineralisation and leads significant new directions globally by integrating advanced imaging and microanalytical methodologies with innovative life science approaches. Now to Dorrit's presentation.
Geochemistry of the carbon cycle. I work in two different research areas. And both ends of the carbon cycle. I call this the shallow end, where I look at where I do work on biological form carbonates, namely shells of aquatic organisms like bivalves, gastropods, foraminifera, and so forth. And I also do research work at the deep end of the carbon cycle, namely on natural form diamonds from the Earth's mantle and their inclusion. And I'm going to give you a little bit of a glimpse of what my work is in both areas today. Biocarbonates are important archives of past environmental conditions. We use them to reconstruct past environmental conditions, but we also use them to forecast how the climate might change in the future. They are like bones and teeth in our bodies. They are formed by organisms, which means that every ion must pass through a living cell. Now that has consequences. For once, there is strong metabolic control over shape and composition of every mineral formed. There's also, it's not solution chemistry, but it's active transport through membranes. And the end product is a nanocomposite of organic and inorganic materials, such as pearls and shells. They have, therefore, different compositions, and they're also different in mechanical properties from their inorganic geological counterparts. As you can see from below, these beautiful different morphologies for geological calcite as opposed to calcitic coccoliths in plankton. Now, just to give you a glimpse of the work that I do in nacre, so nacre or mother of pearl consists of 4% organics, 3% of water. Apart from that, it is aragonite, calcium carbonate. There's an image here on the left. That's a SEM image of a broken piece of nacre. Many people refer to the structure as a brick and mortar structure, where the so-called bricks are pseudo-hexagonal platelets of aragonite, as shown in the schematic below, that are sheathed by an organic membrane, or at least an organic sheath. And they also have, between the layers of aragonite crystals, they have these intercrystalline organic matrix sheets. If you look closer with on the image on the right here, which is taken with an atomic force microscope, where you can just see the outlines of the little nacre platelets, you can see that each and every one of these nacre platelets actually consists of granules. These granules are about 50 to 100 nanometer in size and consist of calcium carbonate. Now, it's all crystalline aragonite, we thought, but if you look closer, and this is one of our studies we did with transmission electron microscopy. We've also done Raman spectroscopy on these things. And we can actually show that when you take an electron diffraction traverse across one single granule, we can see that these granules are in fact amorphous. They're not crystalline, but they have diffractograms shown here on the very right that are consistent with amorphous calcium carbonate. So these shells of those bivalves they are not completely crystalline. They have areas where there is amorphous material in them, and it stays amorphous for the entire lifetime of this animal. Which is then led to our finding that bivalve shells in fact crystallize from the amorphous phase, from amorphous calcium carbonate, and still contain patches of ACC later when you harvest them later. Now, this was an interesting finding, because this is not what we know how crystallization in the classical sense work, where we build a crystal by ion adding one ion by another. In this case, we are looking at particle attachment growth, and these particles in our case are in fact amorphous and only crystallize later once they're attached and organized. We have since found evidence for this type of growth mechanism in many other marine calcifiers, and here's an example for foraminifera. And of course, that changed how we look at the models for our paleoclimate reconstructions, because these models are all either based on empirical data or they're based on a model that sees the classical crystallization hypothesis in action, where calcite and aragonite crystallize directly from seawater without this pathway through the amorphous phases. So clearly these models are not appropriate to describe reality. And my new discovery project going into the future is actually about assessing how far off we are from reality if we use such models to describe our data. Now, the other area that I wanted to let you in today is my work in diamonds. And here I look at a specific species of diamond, namely the polycrystalline diamond aggregates. Polycrystalline diamond aggregates are very fine grained, and I've got pictures here on this side. You can see that nowhere near as beautiful as their gem diamond cousins, but they're still part of the diamond population unearthed from many of those localities worldwide. And while they're not as beautiful, they're research-wise even more interesting. At least that's my opinion. We've had some work published in the year 2000 where we could show that these diamonds are much younger in parts than, or in some areas, than the normal gem-sized diamonds. Some are formed just a couple of 10,000 years before the volcano erupted that brought them to the surface. They also form episodically over the lifetime of our planet. There are rapid formation, then we can tell because they're very small grained. They come from a highly supersaturated solution originally, and they're in fact rich in inclusions, in mineral inclusions, and rich in fluids. One of the telltale factor here is that they are actually quite porous. They have up to 30% porosity, which is quite a rare sight in a rock that comes from so deep in the mantle. Now, sometimes you're lucky enough to find these fluid inclusions still intact. And here on the left, there's a little bit of a video from where we found one such inclusion intact in one of our focused ion beam cut foils under the transmission electron microscope. You can see from the changing electron diffraction contrast that there is actually liquid in this cavity in this diamond here. Further evidence for the composition of these fluids we can find on the right here. This is a cathodoluminescence image that shows the diamond growth zones. You can see different small fractured grains of diamond that have aggregated together. So they're actually grown together, but in different steps. Each one of these brightness differences is a different growth zone that shows up in cathodoluminescence. The points on the side show you what the carbon and nitrogen isotopic composition is of these grains at these specific points. And we can say from this that unlike their gem-sized diamond cousins, these little polycrystalline aggregates grow in pulses, where the fluid pulses differ in composition. So work in the future is going to look at how we can unravel the timing of these pulses and where the sources of the fluids come from. And lastly, the other thing that we could learn from these specific different species of diamonds is that some of them carry direct evidence for how diamond forms by redox reaction in the mantle. And again, here we were relying on a transmission electron microscopy with electron transparent foils. And on the left side here, you can see one of those diamond foils, the diamond in gray, with two inclusions in them. In this case, these two inclusions are iron sulfides, pyrotides. And you can see on the bigger inclusion on the left that there is a small reaction rim around them, which we found is made of magnetite. On the right image, you can see electron diffraction imaging of this, and what we could see is this the sulfide is deformed, so which means it had to be solid when it was included in the growing diamond, which then of course means it's not synchronous, it's not syngenetic, but it had existed before the diamond grew, so it's a protogenetic inclusion. However, we could also see from this small thin magnetite rim around it that doing the electron diffraction on them, we could see there's epitectic relationship between the sulfide, the magnetite rim, and the diamond, which means that this magnetite rim is in fact a reaction product of a redox reaction where the sulfide, the iron sulfide, was oxidized into magnetite and H2S, whereas the diamond forming fluid, which was most likely a carbonatitic fluid, was reduced to form diamond, and they all crystallized on this protogenetic sulfide inclusion. The other thing that this inclusion also showed us, that when we analyzed the magnetite more closely, we could see that it was full of twinning boundaries, which were a remnant of a pre-existing higher pressure phase that was destabilized, and reconstructing the pressure boundaries for where this higher pressure phase would have been stable showed us that this diamond assemblage was formed at 300 kilometers depth, which is well at the base of our lithosphere or even beyond. And that was in fact the first time that anyone could pinpoint the pressure, the exact pressure estimate for where these diamonds come from. So as you can see, I think while they're not as beautiful as gem-sized diamonds, I think they have to offer a lot more than those gemstones in terms of research. Thank you very much.
That's terrific. Thank you. We are now delighted to have with us Professor Dorrit Jacob. Congratulations, Dorrit, on your election to the Fellowship of the Australian Academy of Science.
Thank you very much. It's my pleasure to be here.
And thank you so much for your fascinating presentation. We have some questions for you. There's one from an anonymous person in our audience who says, so can diamonds form from shells?
Yeah. Well, eventually they probably could, and I wouldn't mind that. But the connection between these two fields of research for me is really the technical part of it. So the methods that I use is what connects these two fields.
Fantastic. Dorrit, Professor Malcolm Sandbridge says congratulations, and you mentioned that a 30% porosity was anomalous for aggregates from so deep in the mantle. Do we know that the porosity existed at such high pressures and was not formed later as the material rose up?
Yes, I think we can be quite sure that the porosity is a primary feature. We have gaseous inclusions in the diamonds, and the morphology of the crystals around these empty places are very much are sort of rounded, which gives you more an impression of that there was some fluidy, non-defined thing in there, and not something that was dissolved later on.
Thank you. Dorrit, could you share some of the examples of how your findings using these proxy archives have led to insights into understanding environmental conditions in time and space?
Yeah, great question. So I think we approached these topics with plain curiosity, and it was actually brought to us from the jewelry industry originally. It all crystallized from a question that was brought from retail to us and then led to all of this. And we realized while we're pursuing this path of really answering the question why, that we found that the way that these shells form is not the classical way. And we were not the only ones finding that. And this whole finding had actually moved to change a paradigm in chemistry and crystallization theory. So we're really at the start of looking at how our findings will change how we reconstruct past climates, because it's really a hodgepodge of things that we're looking at here. It's how they've crystallized, but it's also how the organism controls through their metabolism, how what ions are incorporated.
All right, thank you. Are there potential applications for your work on biologically formed carbonates in engineering bio-derived materials?
Yes, in fact, nacre or mother of pearl has been a topic for quite a while as a biodegradable and biocompatible replacement coating on hips, artificial hip joints and these things. And I've just read the other day that there is a joint venture between the Australian pearling industry and a researcher in Curtin, I think, developing such materials.
All right. And another question for you. What can your research on bivalves tell us about the response of carbon depositation processes under elevated carbon dioxide?
Yeah, I think shells as such form a little bit too slow to help us deposit more CO2. But it is really the plankton that will probably play a role in this process. But it's also at the same time with the further acidification of our oceans, it is at a great risk that particularly in the food chain, plankton plays a fundamental role. And the harder it gets to actually form these shells against these pressures, these environmental pressures that come through the change of our climates, that will impact greatly our food chains.
Okay. An audience question from Ian Jackson. Dorrit, thanks for your presentation. By way of follow-up to Malcolm's question, is the implication of the porosity that the fluid pressure was at least transiently comparable to the prevailing pressure?
Thank you, Ian. Great question. And short answer would be yes.
Excellent. Dorrit, look, that's all we've got time for. Congratulations again on your election to the Fellowship of the Australian Academy of Science, and we look forward to welcoming you in Canberra.
Yeah, thank you very much.
Thank you. To introduce our next fellow, please welcome back Professor Malcolm Sandbridge.
Thank you. Next, we will hear from Professor Lidia Morawska. Lidia's diverse research involves groundbreaking applied studies in the areas of radiation physics, environmental physics, and atmospheric physics. She is recognized as a foremost authority on airborne particulate matter in the context of atmospheric pollution and its sources, as well as its impact upon human health and the environment. Now to Lidia's presentation.
Good morning, and thank you for the opportunity to give this presentation, which I titled The Science of Air, Helping Us Thrive. I'll start by saying that I'm deeply honored to be elected to the Australian Academy of Science. It was when I was in primary school when I decided to become a nuclear scientist. I was hoping that this would allow me to gain deep insight into how nature is built and how it operates. My idea then was that I would be working in a nuclear reactor and be part of a large team. I was thinking that maybe my contribution to this science would be small, but still it would help to advance this science. I achieved this objective. I gained a degree at the Jagiellonian University in Krakow in nuclear physics of low energies. During my master, I developed an interest further into something slightly different, which was environmental radioactivity. And the topic of my PhD was radon and its progeny in the air, radon emanating from radium present in the soils. One of the very first of my papers was this influence of sealants on radon emanation rate from building materials. What then attracted me to study air pollution? I was a postdoc at the University of Toronto, investigating various aspects of radon and its progeny in the air. And there I had an opportunity to use what's now called differential mobility particle sizer to investigate the sizes of airborne particles to which radon progeny attach. Somehow I thought that this science was somewhat exhausted then, while of course important in terms of mitigating radioactivity in the air. On some occasions, I was in a way playing with this instrument and trying to see what would happen if I measure from outside the window. And then I discovered thousands, hundreds of thousands of particles present there. And the question was, what are these particles? There was no answer to this. There was basically no science available on this. I thought that this was fascinating and very important, and I decided to answer these questions. Very soon after, I was appointed as an academic at the Queensland University of Technology, and to work in this field, I established the International Laboratory for Air Quality and Health. Soon after, I joined QUT, and initially, the name of the laboratory was Environmental Aerosol Laboratory to stress its specific focus on aerosol particles in the air. Since 2004, we are a WHO collaborating center. The change of name stresses more that particles, aerosols are in the air, not by themselves, and the specific focus on health. After years and decades of working on this topic, I have answers to my initial questions, which I posed in Toronto, and many other questions. In a very simplistic way, I could say that the vast majority of these particles are ultrafine, which is smaller than 0.1 micrometer. We also understand where do they come from. Most of them are emitted directly by the sources, roads on which there are cars, and other forms of transportation, aviation, shipping, and so on, like presented on this diagram here of the size distribution of diesel particles. The particles also are formed in the air from gaseous precursors, and then they are called secondary organic aerosols. It is a very complex and fascinating physical chemistry of the processes leading to formation and fate of these particles in the air. But these particles are pollution. So what to do to control them? How do we control air pollution? The most important document which puts the foundation for doing this is the WHO Global Air Quality Guidelines. The current edition of the guidelines was announced by the WHO in September, and I'm proud to say that I was part of the guideline development group. If we look at what the new values are in 2021, they are all lower than those which were in 2005. The last document, I was also involved in this. And the reason why they are lower is because our science advanced, and now we understand that there are impacts of the pollution at a much lower level. But then you may ask, what about the ultrafine particles, which I just mentioned? The problem was that there was not enough epidemiology which would give foundation for setting guideline values. However, there was a lot of science available to do something about this. And with this in mind, a few years before, I established a group. We called ourselves Thinking Outside the Box team, which we brought together the science of ultrafine particles as an evidence for policy makers. I'm pleased to say that this evidence is included in the WHO guideline document as numerical values stating best practice, what to do. We now understand very well the pollution, the aspect of the pollution, and the fact that combustion is the main source of air pollution, and as well the impacts of this. Therefore, there's urgent solution needed, which is transition to clean energy. And this is the direction of my work now. But there was also another angle of my work, and this was the indoor air and human exposure. This work started in 1995 with the first event I organized on this topic, International Conference Indoor Air and Integrated Approach. Since then, we've done a lot of work in this field, like, for example, this study where we investigated exposure to airborne particles, these larger particles covered by the WHO guidelines, as well as ultrafine particles in homes, schools, and offices. One specific aspect of indoor air quality or pollution was the physics of respiratory infection transmission. I got attracted to this topic in 2003 during SARS-1 epidemic, when I realized that there's basically no physics existing. Soon after, I published my first paper on this topic, droplet fate in indoor environments, or can we prevent the spread of infections, obtain research funding. We've built infrastructure for this, like, for example, this tunnel to study particles which come from human respiratory activities, and published numerous papers on this topic. This first one, size distribution and sites of origin of droplets expelled during respiratory activities, in a way, laid foundation for the science in this area. When COVID-19 pandemic started, equipped in the science, I was in a position to present the need to control airborne transmission to the WHO and the national public health authorities, and was supported in this by a group of over 200 scientists, which I gathered together, and the open letter, as we call it, was published in the Clinical Infectious Diseases last year. I'm pleased to say that the next day after it was published, the WHO acknowledged the need for considering airborne spread. And since then, the approach has changed, and this mode of transmission is gradually being controlled. But this is not the end of this particular line of science and its application. As we've published in a science paper this year, there needs to be a paradigm shift to combat indoor respiratory infections in terms of building ventilations, and not only respiratory infections, but the science of airborne pollution as such. There's a lot of technology, a lot of science already available, but bringing this together of this complex system as is building will require a new way of looking at this and working on this, which will be the direction for me for the years to come, with the idea of making clean, healthy indoor air the norm. I'd like to summarize this presentation saying that there were two aspects which can characterize my career. Curiosity, which fueled my childhood interest in nuclear physics, then to expand to atmospheric building and human exposure science, but it was the need for purpose of my work beyond satisfying curiosity, which motivates me every day. I'd like to thank you, the Academy, and I'd like to thank you, all the colleagues, hundreds of them from across the world with whom I've worked over the years, as well as my students who I was and I am privileged to supervise. Thank you.
It is now my great pleasure to welcome with us live today, Professor Lidia Morawska. Welcome, Lidia, and congratulations on your election to the Australian Academy of Science.
Thank you very much.
We've all followed your work very closely. It is so relevant and topical at the moment. So thank you for your contribution over a career, but certainly the very important contribution and timely contribution at the moment. I've got some questions for you. I'll start with this one. A large part of your work has been associated with air pollution, and you share how growing scientific understanding and evidence of ultrafine particles has been used to inform policy and drive change in relation to air quality monitoring and control. Why do you think this has been successful, and what are the obstacles and barriers you have faced or needed to overcome? And finally, how might this experience transfer to your current calls to make clean, healthy indoor air the norm?
Well, this is a big question.
A big question.
Well, why it is important, as I explained, first, I started investigating this out of curiosity. This was so fascinating and so complicated, because when we are going down to something in the air which is of molecular sizes, and you need to investigate this, as well as much bigger particles for which you need completely different instruments. So basically everything is a huge scientific instrumental challenge. There was no infrastructure about this for this, and still around the world, in many places, there's very few instruments which enable this kind of measurements. So the first challenge was to build a lab to be able to do this. So this was certainly my direction. But then with time, like with anything to do with air pollution, I realized that it's not just the science, it is what we do with the science. And initially, when I was talking to my colleagues in the government, in the Queensland government, federal government, it was kind of, all right, it was an interest, because it was harmless in that sense that there were no standards, no regulations. So, okay, whatever we would find would not require any action from the government. But gradually it started shifting, and the government started realizing that perhaps there's need to do something. And then the dialogue or conversation started being different. It's not until now that this is in the WHO document that the government see something must be done in some way. It's still not a formal recommendation. But now if you see how long it took basically 30 years from that curiosity, 30 years, exactly 30 years I came to Australia, 30 years later, this is in the WHO guideline document. These things don't happen fast. They happen slowly. But we shouldn't be really deterred by the fact that it's slowly and it's not going to happen by tomorrow. We should patiently, persistently keep pushing, and this is going to happen.
Thank you. We have a question from David Balding. Professor David Balding, thanks for an excellent presentation. You mentioned that the World Health Organization air pollution guidelines have come down over the years, but has this had the desired impact on policy and outcomes in Australia?
It has, because during all the time I've been involved with the Australian decision makers, whether on state or federal level, there's been always reference made to the WHO guidelines. Mm-hmm. There's no government in the world, in the developed world, which is not looking at the WHO guidelines. But it's always the discussion at what level the values of the standard, national standard, would be set compared to the WHO guidelines. And it is acknowledged that in many countries, the levels will not be at the WHO level. It will be not possible. Therefore, the WHO has also interim targets for pollutants. So in Australia, these conversations have been happening. For example, for one of the most significant pollutants in terms of health, PM2.5, particles smaller than 2.5. It was quite a long time ago, initially, when it was set up as a reporting standard. So we are looking at this, we are not doing this, but looking at this. Eventually, a standard was set, and I must say that when it was set, so for annual value, it was actually lower than below the WHO guideline level. Now it is slightly above. But it definitely has had an impact. So this was good. I wish that the same will happen in relation to indoor air pollution, for which Australia doesn't have a standard yet.
You've partly answered it, but I will share with you Professor Francis Separavich's question that relates to the Australian context. Francis writes, congratulations, Lidia. I'm sure that the work of you and your colleagues is guiding how the new quarantine facilities are being designed. Is there a move to change the ventilation guidelines more widely?
Well, I would have hoped to have an answer yes, but I don't have this answer yet. From the federal government at this point, I haven't heard not a single time the word ventilation. In different states, there's definitely move. Victoria started first started looking at the ventilation guidelines for schools. New South Wales is moving in this direction as well. In other states, the conversation started, but this is with the focus on schools. But schools is just one type of facilities. The adults are not in schools. We are everywhere else. So not really. It hasn't really started. But I suspect that there's a lot of discussion already happening at all levels of the government. And the complexity here is that we are not talking about just one government department. It's not just the Department of Health, which should say that this is a health problem. It's not just the Department of Education. It's just basically, half of the government departments which have some role to play with this, which makes it extremely difficult. In addition, there's a need for changing national building codes, which again is not a process which is happening fast. But all this, from what I hear, it's slowly happening. And sadly, my colleagues and I are doing our utmost best to push this process so it is completed as soon as possible.
Lidia, thank you. I know we've worked together to look at ways in which we can work with you to help influence the policy landscape in Australia. And it certainly is a complex one, not just to achieve best practice, but right down to the very beginning. As you've said, building codes and other such regulations don't at the moment have air quality requirements, and it's something we need to work hard to achieve. I'm afraid that's all we have time for. I would love to have asked many more questions, and I'm sure others would have too. For now, thank you. Welcome to the Australian Academy of Sciences Fellowship, and thank you for your excellent work and presentation.
Thank you very much.
Please now welcome Academy Foreign Secretary, Professor Elaine Sadler, to introduce our next presentation.
Thank you. Our next new fellow presentation is from Professor David McClelland. David is a gravitational wave scientist who's played a critical role in improving gravitational wave detection sensitivity and in designing, installing and commissioning instrumentation that has underpinned the discovery of gravitational waves. David was elected in 2019 but could not join us to present in that year, so we are really delighted to hear from him today.
I would like to thank the Fellowship of the Australian Academy of Science for electing me into your ranks. It is a great honour, and I'm looking forward to finally signing the book. I've been most fortunate to have been involved in one of the great quests in physics, the direct detection of gravitational waves. It was and is big science involving over 1,500 people from more than 15 countries. My group's success begins with the great support we've received from the Australian National University and the Australian Research Council over almost 3 decades. They had the wisdom to understand that investment in fundamental science not only inspires the nation, but leads necessarily to applied outcomes. I've had the great privilege to work with a fabulous group of people in the Centre for Gravitational Astrophysics over my time. My election to the Academy reflects in no small part on such a wonderful team, many of whom have devoted their research lives to this grand endeavour. Now to our gravitational wave story. Einstein predicted the existence of gravitational waves, or waves in curvature of space-time, that they stretch and squash objects as they pass through the universe. So he made this prediction in 1915, but it took 100 years to verify. So this is what I call the waves of the century. It was on September the 14th, 2015, that the LIGO detectors in Hanford and Livingston in the United States had just finished commissioning. In fact, it was early morning in the US, 1 hour after commissioning ended, when this sound was heard across the universe. This is the characteristic chirp that's generated when 2 black holes of 30 solar mass collide with each other. And that signal, that gravitational wave, passed through the universe, and it passed through the Earth through the LIGO detectors, and this is a signal we recorded. It was not only heard across the universe, it was heard around the world. This is now an iconic symbol of gravitational wave detection. This binary black hole source, we've seen a number of those since then, until on August the 17th, 2017, about 4 years and 22 days ago, we observed this event. These are 2 neutron stars spiraling around each other, and as they're spiraling, they're generating gravitational waves in the band that LIGO can detect, until eventually they collide in a massive burst of gravitational waves. And 2 seconds after that burst of gravitational waves, a gamma ray burst was detected. 10 hours and 52 minutes after that, optical radiation was observed, and then infrared radiation. 15 hours and UV was observed. 9 days until the X-rays were seen, and then 16 days later, radio waves from this event were observed. 70 telescopes were trained on this event. It was the most observed astrophysical phenomenon ever. It started the multi-messenger astronomy using gravitational waves and electromagnetic waves. So what we've observed since 2015 is shown in this plot here. Masses, we call it, in the stellar graveyard. In purple are black holes that are discovered using electromagnetic means. Yellow neutron stars that were discovered using electromagnetic. The blue and orange are gravitational wave detected signals. The orange are the neutron stars seen with gravitational wave detectors, and the blue black holes. This presents some very interesting findings. For example, between 2 and 5 solar masses, you'll see there are objects there. These seem to be too light to be black holes, too heavy to be neutron stars. We wonder what they are. Then there's a mass gap around 80 solar masses that should have been there, where we don't think that stellar evolution would produce black holes of those sizes, but there are many of them. There's this 160 solar mass black hole, the biggest one we've seen to date. So these are phenomena, these objects are beginning to drive the field of gravitational wave astronomy. But let's go back to the beginning, back to 2015, because the signal just didn't create the beginning of gravitational wave astronomy. It presented a pin-up, a precision measurement so far not copied. We reached an amplitude, we measured an amplitude of 2 × 10⁻18 of a meter. That's an incredibly small amount. It's 1,000 times smaller than the size of a proton. We've effectively observed the separation between the centers of mass of 40 kilogram objects oscillate from 30 hertz up to 240 hertz with a peak amplitude of 2 × 10⁻18 meters. The standard quantum limit of motion for an equivalent object is only 2 × 10⁻19 meters, very close to putting these objects into quantum ground states. So we may be soon able to observe human-sized objects behave quantum mechanically using these gravitational wave detectors. But let's now take a look at the instruments we use to do this. This is the layout of the LIGO interferometers, and it's a fairly busy slide. I'll take you through some of the major points of it. Light from a pre-stabilized laser, 200 watts, is shone into the interferometer. It's cleaned before it enters, passes through conditioning optics. At this point here, called the main beam splitter, half the light gets sent vertically and the other half goes horizontally. The light is then stored in cavities in these arms before it returns to the main beam splitter and is compared to the light coming out of the other arms. And that light then enters through steering mirrors and is shone into our photo detection system. Now we want to set this up and have it such that in the absence of a gravitational wave, there is no light coming out of the detector. So that when a gravitational wave comes through, which squeezes and stretches the arms, a signal is produced. Now to do that, we have to make sure all the optics, everything in this device is totally isolated from everything on Earth. So it's in massive vibration isolation systems, massive vacuum systems, and so on. But we also have to make all of the elements, all of these optical elements work together. And that's called a control system. That's one of the main things that my group worked on to begin with. We developed this control system by using an auxiliary laser, using a green laser, along with the infrared laser, which is the main science laser, this red beam. We shine both a red laser and a green laser into the interferometer, and we can use the information extracted to figure out where all of the optics are and make them work together. Now, this diagram here is in the end station of one of the interferometers. You can see how big this is. This is a person standing inside the interferometer. You can see the green beam, the auxiliary laser beam we use for control systems, and the infrared beam would be bouncing along this arm here. Another contribution we've made is in these devices called steering mirrors over here. Now, these steering mirrors are small optic suspensions. There are 20 of them throughout the LIGO interferometers, and they're used to steer the path of the lasers. For example, the light that comes out of the interferometer is directed by the steering mirrors through this device called a mode cleaner into the photo detection system. Now, these steering mirrors were designed at the ANU and built in our workshop. So it's kind of cool to know that we've got these mechanical devices sitting in these massive detectors overseas. The last thing I want to point out on this plot is the most recent contribution, and that's squeeze light injection. Now, it turns out that no matter how well we try to control the laser, its amplitude and its phase are fuzzy. This is due to quantum mechanics. At the very limits of measurement, there's a slight fuzziness in how precise a laser beam is. Now, normally this fuzziness, this quantum noise, doesn't affect us. But when you're trying to measure an amount which is 1,000 times smaller than the size of a proton, these effects can mask the signal. Now, we can't get rid of quantum noise, but what we can do is to manipulate it so that we can use something called a squeezed light state, inject that into the interferometer and manipulate it so that the quantum noise doesn't affect the measurement. Squeeze light generators have been pioneered at ANU. We developed the first squeezing for gravitational wave detection in around 2004, and it took to around 2016 to work with the community to install these squeezers on site. And this shows some of the ANU team. But what's next? Well, NEMO. NEMO is the Neutron Star and Extreme Matter Observatory. It's a pathfinder to a future Australian third-generation detector. But that's a story for another time.
Thank you, and it's my great pleasure to welcome now Professor David McClelland live with us. Congratulations, David, and thank you for your presentation.
Thank you very much. As you said, it's great to finally be joining the Academy. Well, I've been there for 3 years, as you said, but I'm looking forward to getting the pin.
Yeah, who would have thought not being able to give a presentation in 2019 might have delayed it 3 years. We're really pleased you've been able to deliver it, and we look forward to welcoming you in the Shine Dome in person to sign the beautiful charter book that does have the signatures of all of our fellows. We have a question from the audience for you, and it reads, should Australia participate in a future gravitational wave observatory?
Well, I think you've probably already guessed the answer. I would give absolutely. Absolutely for a number of reasons. One is that having a global array of these detectors is the best way to get the science out of them. The global array allows us to locate in the universe on the sky where the signal's coming from, and that information is then fed to the other spectrum, the other carriers, and we do the best science. But it's also detector that inspires. It's inspirational to have these sorts of facilities built on our shores. The SKA is an example of one such facility. And radio astronomy started with Australia at the vanguard, and I'm hoping that gravitational wave astronomy has us at the vanguard as well.
I want to take you back to the moment where you first realised LIGO had successfully detected gravitational waves. What was that like? And do you think a future observatory will produce many of those extraordinary moments?
Well, that moment was absolutely incredible, almost unbelievable. It was, in fact, we all took pause. We'd had a number of events in the past which may have been signals, but after thoroughly examining and checking them out, they were excluded. So when this signal came in, we thought, well, it's probably some artifact, or in fact, what we call a blind injection. Often we inject signals just to test the system out. But so it took a few days before it became clear that this was none of those. This was actually a real signal, and that's when the elation hit. 30 years worth of our effort, 100 years in science time, fabulous. Now, will we feel that elation again? Well, I think we will. I think the Neutron Star event was almost as incredible. It launched that field of multi-messenger astronomy, which is really where the future's going. But I think there will also be signals out there we haven't predicted yet. This happens when you open new windows, when you build new telescopes. The pulsar itself was the little green men, they thought it was, back in the 1960s. I think we'll discover new phenomena with gravitational waves, which might revolutionize our understanding of the universe.
Yeah. You described that chirp that was very characteristic. It sounds like an odd question, but is it always a chirp that would identify something new? Are there other sounds that one might expect? And how do you know that it's not any old chirp or a machine going ping?
Well, there are many sounds. It's a forest of sounds, just like you're in a forest with thousands and thousands of different species of birds, and they're all making these different sounds. How do you distinguish one from the other? And we do that by, if we know what the signal looks like, we use something called match filtering to pick out that signal. But if we don't know what it looks like, and that's what's going to be the most exciting thing, then we have to work really hard to make sure that everything else that it could possibly be is eliminated. And then we can use that information to construct what the source might have been in the first place.
Yeah. Francis Aparovich says, this is a non-scientific question, but do you still get emotional when you hear that sound? It's so amazing.
Yes, I do. The whoop, and we love to use it with school children wherever we can. It went around the world, as I said, and it was one of those events which brought the world together and in a positive way. And I think that's one of those magic things that can happen. The last time I remember that really happening was the lunar landing, the man standing on the moon.
Indeed. David, I'd like to ask about any other applications for your light squeezers. They sound fascinating. Where do you see that science being applied?
So it's actually interesting that the first true application of squeezers which needed a quantum state is in LIGO. So whilst they've been around a long time and there are applications in the measurement field like that, now they're also being used to if you have to measure a delicate molecule with a laser beam, you don't want to put too much power in that system because you might destroy it. So you want to lower the quantum noise, so you can do that by using squeezing. But squeezing, and this might be a bit debated by others, squeezing comes out of a field called quantum optics or quantum information, and which powers many of our centres of excellence now. What's really interesting is that quantum information, trying to understand the effect of quantum mechanics on measurement, began with the gravitational wave field back in the late 70s and early 80s, with people trying to understand noise in those systems. So that's one application that led to industries today. But within my own group, we've had a number of spin-off technologies. Dan Shattuck launched a company called Liquid Instruments with test and measurement gear. Kirk McKenzie uses technology for Earth observation from space. Bram Sagmolan, earthquake sensing, and Zhong Chao, exploration, security, perimeter sensing. So the technologies that we develop for that fundamental application, which drives us, that's what's driven us to do the work, has the potential for many applications. And it's a great example of how investing in fundamental science can lead to positive technologies.
Absolutely right. David, that's all we have time for. Thank you once again. We really look forward to welcoming you at the Shine Dome, and congratulations on being elected a Fellow of the Australian Academy of Science.
Thank you very much. Truly honoured.
Please now welcome back Professor Elaine Sadler to introduce our next presentation.
Thank you. Next, we will hear from Professor Susan Coppersmith. Susan has made outstanding contributions to several subjects in condensed matter, focusing on the fundamental nature of systems that are far from thermal equilibrium. She's developed theories unifying phenomena that are observed in areas as diverse as population biology, biomineralization, granular materials, and superfluidity. She's also made important contributions to the development of quantum dot qubits in silicon germanium heterostructures. Now we'll hear from Susan.
Hi, I'm Sue Coppersmith. And first of all, I'd like to thank the Academy. I'm very honored and excited to be elected as a new member. And I am also really grateful for the opportunity to take a few minutes to tell you about my work. I am a condensed matter physicist, a theorist, and I study complex systems. And so condensed matter systems are complex, but then also some of the concepts developed in condensed matter physics have turned out to be relevant in a broader array of scientific fields. And seeing those new applications has been part of the interest and excitement of working in the area in which I work. And so I have worked on a bunch of different subjects in the course of my career. But today I'm just going to tell you about one example, which is called stochastic resonance. So let me first tell you a little bit about stochastic resonance. It was originally developed by some Italian physicists in the early 1980s, motivated by the observation that ice ages tend to reappear with a periodicity of roughly 100,000 years. And there is an effect that affects the amount of light reaching the Earth from the sun, which is that the eccentricity of the Earth's orbit has a variation on that timescale due to the effects of the other planets. But that leads to quite a small effect and not big enough to explain the change in climate due to ice ages. And so in the early 1980s, a group of Italian physicists made a hypothesis that it was due to amplification of this noise due to the presence of the little signal due to the presence of noise. And it just so happens that this year in 2021, one of the Nobel Prize winners in physics was Giorgio Parisi, and he's one of the originators of this idea. And so I think it's really fitting to tell you about it, partly because of his work on the subject. So let me tell you how it works. And so one way, the simplest example, and which provides the intuition so you can get an idea of how it works, is that if you take the signal that you have, which is small driving, and you add noise to it, you can get a large response, because basically you can think of the thing as a two-state system with two different places and with a barrier. And the noise gets you over the barrier, and yet the signal makes it more likely to be on one side than the other. So this is a driving where the size of the barrier is bigger than the drive, but in order to get over the barrier, that little bit of drive isn't enough, and the noise helps you get over. But of course, if the noise is too big, then it just wanders around, and then you're not sensitive to the signal at all. And that's what leads to a resonance, the fact that it has to happen at the right rate. So, so here's sort of the picture of it. Again, the idea is that you're driving it. If the temperature is small, you just hang out in whatever well you started, and you hardly ever make transitions. If the temperature is really high, you go back and forth really quickly. And if the temperature is intermediate, you actually can synchronize to the drive. And it's not generally agreed that this is the explanation for ice ages, but stochastic resonance has unambiguously been found in quite a large variety of systems, ranging from crayfish neurons to nanomechanical resonators. And again, there are many, many applications where it is convincingly shown that stochastic resonance is operating. So what did I do in this field? Well, what I did in collaboration with Lidia, who is a student, we together realized that stochastic resonance also can occur in quantum systems. And this is not so obvious because what I was telling you was that you have to go over this barrier, and that barrier leads to a very temperature-dependent, noise-dependent rate at which you cross the barrier. But quantum systems can go through the barrier, and there's not a strong temperature dependence. And so therefore, you might think that you wouldn't get this effect where noise really matters. But what we realized was that if you have an asymmetric system where these two wells are not symmetrically placed, but one has higher energy than the other, well, that difference in energy leads to very similar phenomena where one of the rates becomes very noise dependent. It kicks you up to the higher well, and that is very sensitive to noise. And so it turns out when you work it through that leads to very similar phenomenology. So here is the picture of quantum stochastic resonance. Again, the thing that matters here is not how high the barrier is, because you can tunnel through it, but what this asymmetry is and how big the temperature is relevant to this asymmetry. The temperature just being a measure of the amplitude of this noise. Okay, and if the temperature is low, you don't make it up to the other well, to the upper well. If the temperature is really big, then you go back and forth, and it doesn't care what well it's in. But there's an intermediate temperature where when you drive, you can really change the occupation of that upper well significantly, and you get a really large response to the drive. And now just recently, in the last year, this paper was published just a couple of months ago, a group in Stuttgart has measured stochastic resonance in individual iron atoms, the spins of individual atoms. And this is really interesting because now the context is spintronics and looking at the magnetization of individual iron atoms. And the way the experiment is done is using an STM tip, scanning tunneling microscope, and then you basically have a conducting tip that's magnetized, and here's the spin on the atom, and you apply a voltage, and the current that passes through depends on what direction the spin on the atom is pointing. Okay, so we can resolve individual spins on individual iron atoms and measure them through the STM tip. And it also turns out that the voltage you apply changes the level of the noise in a way that we can characterize really quite quantitatively. And here now, again, you see very similar things to what you saw in the classical system. Here, your drive is very slow, so you make lots of transitions and you're not synchronized to the drive. Here, your drive is very fast, and there are many periods of the drive between the transition, but at intermediate frequency, there's a frequency at which what happens is the up transition. This is like from the spin up to the spin down. You go, here you are, you hit spin down and you rise this one misses, but every period there's an up transition. And that is the transition that, it does depend on the amplitude of the noise. And so the noise is synchronizing the response to the drive. Then this is, again, just the schematic of the experiment. You just apply a voltage through this tip, which is magnetized. And then that current that you measure tells you what spin state the atom is in, which I find quite amazing. All right. And so here are the data. You can quantify, do a quantitative analysis of these data. And you can see that if you ask, what's the time between the successive up transitions, if you don't drive it, you get a very broad curve. And if you do drive it, you get a really big peak, a very sharp peak at the drive frequency. And if you just say, what's the height of that peak and what's the probability that your response is in that, the length of time between the ups is in that little time window, it can get up to close to 50% efficient. And one of the things I'm very proud of is that the theory, we did a complete theory of this, and with no adjustable parameters, you can get essentially perfect agreement. So not only have we qualitatively shown that quantum stochastic resonance occurs in the system, but we're also able to do a real quantitative analysis and extract physics from the experiments. So that's what we're doing now. We're using quantum stochastic resonance to understand and control spin dynamics, and also to probe new physics, because we have a theory that with certain physical assumptions is quantitatively accurate. So it fails to be quantitatively accurate. We know that new physics is arising in the system. And then also the analysis we're extending to analyze more complex systems with more than two states, because we've been spin up and down, but obviously we can do more. So to summarize, stochastic resonance is an amazingly applicable concept, and it applies in many, many situations. And one of the things I've done in my career is to apply it to quantum systems. And recently we've done a specific experimental realization of this. Relevance is to the spin dynamics of individual atoms. Thank you very much.
Thank you ever so much, Professor Susan Coppersmith, and congratulations on your election to the Australian Academy of Science. Thank you for such a terrific presentation.
Well, thank you so much. It's such an honor.
Susan, we have some questions for you today. I remind the audience, if you have questions for Susan, please pop those into the Q&A panel to the right of your screen. I'm really interested to know how you came to investigate stochastic resonance in quantum systems. What path led you there?
That's a great question, because in all of my work, I always wonder what led me there. But I'd say one of the wonderful things about the kind of work I do is you hear about something and you say, that's a really interesting idea. Is it good for something else? And I think that is something that theory can really bring to science more generally. And someone just told me about stochastic resonance. And I had been working on quantum systems and saying, that's a really cool idea. Could it apply? And then there was this, oh, can't apply. But wait, what if we do something a little different? And I think that's been really useful overall, where actually in the previous talk, again, this thing about the quantum squeezing came up in the context of gravitational waves, but then applied in the quantum optics realm. And so there's a lot of that in physics, which something that I find really exciting.
Yeah, and how fascinating to then find it present in so many different ways. You mentioned the different, not just applications, but places where you've detected it. So your work is helping to understand and control spin dynamics. Why is that important? What are the applications there?
Well, the applications that motivated the actual experiments that this group was doing is this idea that you want to make a memory, a computer memory that's smaller, can contain more information, and yet you can get all the information out. And they were studying this fundamental question of how dense could you make it? And if you make a single atom, can you actually store it and get the information out again? So they were addressing this fundamental, what is the fundamental limitation? But obviously understanding the fundamental limitations is important as we scale everything down, and our phones can only be a certain size, and yet we want them to be more powerful. So that motivates that whole area of research.
You mentioned your work is helping probe new areas of physics. What kinds of areas do you think you're likely to expose, or what's the general direction it might be going in?
Well, some of my work is relevant to this, trying to make more powerful electronics. And I do also work in the field of quantum information, where that's explicitly, we're using a new type, using quantum physics to get more computational capability. But some of the work is looking at sort of the classical electronics, using the phenomena we know now, but making them more efficient and coming up with new materials that can make sort of something similar to what we have now work a lot better. And so there's there's just an incredible array of things that people are studying. And again, every once in a while, one of them becomes commercially important, and then we don't even think about all the research that went into it. Sometimes.
I imagine that we'll see an explosion of discovery here. And it is always important to remember, and we've seen it with COVID-19, we've seen it in many areas, that where we are today has relied on decades, sometimes centuries of research. And that investment in curiosity-driven research is so important, so that we then can have those applications further down the track. Susan, you mentioned that one of the recent Nobel Prizes was awarded to one of the originators of the concept of stochastic resonance. What does that recognition mean for the field?
Well, it's interesting because this is Giorgio Parisi, and he's done a lot of really important work. And this is not probably his most famous work, but what he's done is he's really, and he's in this community, the people who are leading this community, again, realizing this incredible range of situations where fundamental physics concepts can really give new insights. And I think that the validation of, yes, it's really important that these insights can be important in the original fields, as well as in terms of providing an intellectual framework where things are related, I think is real validation for the field. So the people working in the field were very excited by this Nobel Prize.
Thank you ever so much, Susan. That's all we have time for today. Congratulations again on your election, and we look forward to welcoming you in Canberra.
Okay, well, thank you very much.
To introduce our next fellow, please welcome back Professor Elaine Sadler.
Thank you. Our next presentation is from Professor Tim Bedding. Tim has played a leading role in establishing the field of astroseismology, the study of stellar oscillations, where he pioneered the detection of stellar oscillations with ground-based telescopes, including the first clear detections of oscillations in sun-like stars, and determining which red giants have started to burn helium deep within their cores. Please welcome Tim.
Hello, it's a great pleasure to be with you today. So I'd like to talk to you about some of our work in studying starquakes and using these to probe the interiors of stars. So this goes by the fancy name of astroseismology, which basically is Greek for starquakes. And the idea is we use oscillations of stars to understand their interiors. So here are two versions of the interior of a star. On the left, we have the cartoons version, but on the right, slightly more accurate, is what it actually looks like inside our own sun. And right in the center of our sun, we have fusion, and that's turning hydrogen into helium, and that's the energy source. That energy then makes its way outwards, in the first part through radiation, which is basically photons traveling, And in the outer part of our sun, the energy is transported physically by convection. The gas actually physically rises and takes the energy with it. And that's quite a vigorous process. And so this convection shakes the sun and it causes it to oscillate in what we call standing waves. And that's what we study. And we study these in other stars. So here's just a movie of the surface of our own sun sped up from a ground-based telescope. And you can see all those little dots, bright and dark dots coming and going. Now in the middle is a sunspot. That's a region of strong magnetic field. But around that you see those light and dark regions. And they're the tops of the convection cells that I was talking about. So we're familiar with convection at home. Convection is the process where hot gas rises and takes energy with it and then falls. And to do this, you need an energy source at the bottom. So if you have, for example, a large empty room and in the bottom you place a source of hot air, then you'll get the convection currents flowing. I have to say I use a version of this slide in my public talks, and I'm a bit worried because I'm recording this talk now in October, and I'm hoping by the time you see it that the two faces here will still be the same ones, because they do tend to change quite often in Australia at the moment. Anyway, here's a demonstration. I'm going to show you something called a Riker tube, which is basically a hollow brass tube like this, but in one end there's a piece of metal gauze. And then I'm going to heat that piece of metal gauze with my burner here and make it nice and hot. And when that metal is hot and I remove the heat source, I'll be able to set up convection. And you can hear that beautiful standing wave inside here as the hot gases rise and fall. So that's a fairly short tube. It had a high frequency. Now I have a longer one here, which I'm going to heat. Again, get it nice and hot. Now this is a much longer tube, so the standing wave that we excite, we expect to have a much lower frequency. And indeed it does. Isn't that beautiful? So the idea is that these oscillations are excited inside a star. A star is a three-dimensional version of this, and we use the oscillation frequencies to understand what's going on inside the star. So how do we measure these oscillations? Well, there's basically two ways. One, the first way is velocity. So we have a star that's expanding and contracting like this. And so the surface of the star that we see is moving towards and away from us. And we measure the light from the star. We measure the Doppler shift with the spectrograph, and we can see the star is moving in and out like so. And this is the method that we used in the beginning. And we did this using large ground-based telescopes, such as the one at the bottom left, which is in Australia, in Coonabarabran, and the one at the top left, which is in Chile. And we use these to measure the Doppler shifts of a few very bright stars. And here's an example. So these are observations made over several nights. And you can see that the velocity of the star is oscillating up and down by a few meters per second, a very small velocity change. So it needs very, very sensitive equipment. And the time scale for this is over minutes. So the star is basically breathing in and out, expanding and contracting in a complicated way like this. Now we analyze these data using what's called a Fourier transform to see what frequencies are present. And here's just one example for 3 stars. In fact, the middle one of these is actually the sun itself. Each one of these blue peaks here is an oscillation frequency of the sun. So the periods of these correspond to periods of around about 5 minutes, 3, 4, 5, 6 minutes. And each one of these peaks is a different oscillation mode. So we didn't do the sun, of course, but we did the top one here, Alpha Centauri A, and the bottom one, Alpha Centauri B. And these are 2 stars very similar to our sun. One is a little bit bigger and one is a bit smaller, and that's why their oscillation frequencies are slightly different. So this is what we did basically for the first part of my career, painstakingly measuring the oscillations in other stars. But it's quite difficult to do, 1 star at a time. And there is a second method. And that method is to measure changes in the brightness of a star, because as a star oscillates, it also gets a little bit brighter and fainter. The problem is if you're stuck on the ground, the Earth's atmosphere makes those measurements almost impossible, because the twinkling of the stars, which is so pretty, is in fact just caused by the Earth's atmosphere. And so you need to get above that. Unfortunately, NASA have launched a couple of wonderful space missions which have produced data that we've been able to use for this. And in doing so, we've been able to measure many, many stars at once. So the best mission for this was called Kepler. It was launched by NASA in 2009 and operated for nearly 10 years, a wonderful space telescope. And this space telescope's main aim was, in fact, to find planets. So how do you find a planet around another star? Well, if the planet happens to pass in front of the star, you see a very small dip in the light. That's called a transit. And that's what Kepler was designed to detect. So Kepler was really, really good at this. It measured the brightnesses of thousands and thousands of stars over and over and over again. And here's just a few examples of planets that it found. So each one of these dips you see is a star getting fainter because a planet is passing in front of it. Now, we can use exactly the same data to measure oscillations in stars. So these stars may or may not have planets, but they're certainly oscillating. And here's just a couple of examples. These are stars that are getting brighter and fainter periodically in a very complicated way, as measured with the beautifully accurate data from the Kepler mission. So this has allowed us to do astroseismology to measure oscillations in thousands and thousands of stars, and it really has revolutionized the field. So what can we learn about all this? Well, obviously, there's a lot of physics we can learn from studying oscillations in stars, and in these few minutes, I'll only mention very briefly 3 of them. But let me start with a bit of physics, just to remind you from your high school physics. If we have an object like this tube that I showed you earlier, and it has a standing wave in it, that standing wave can have several different patterns. It can have the fundamental mode, the first overtone mode, the second overtone mode. And I can in fact simulate those with this plastic tube here. If I swing this around my head, you should be able to hear the different modes being excited. So this should be familiar to anyone who plays a brass instrument, for example, blowing the overtone modes. So these are all different modes, but a star, of course, is 3-dimensional, but it's basically the same thing. It's a big ball of gas held together by gravity in 3 dimensions. And it will also oscillate in these different modes of oscillation. And in fact, stars can do this in many modes simultaneously. So that's why the graph that you see there of the brightness is so complicated, because the star is oscillating simultaneously in many modes. And as again, we analyze these using Fourier analysis. So 3 things that we can learn very, very quickly. 1, we can measure the sizes of stars very, very accurately. Just like my 2 metal tubes here, you could tell without looking that one of them was bigger than the other just by the notes that they were playing. Big stars have a lower frequencies. Why should you care about that? Well, one reason is you may have found a planet around such a thing. And the planet, as you remember, is a dip of light. But the amount of light that's missing is only a fraction of the area of the star. So you need to know the size of the star in order to know the size of the planet. And we can measure the sizes of stars very, very accurately using this astroseismology. So that's one of the things we're able to do. The second thing we're able to do is measure the internal rotation of stars. Stars are big balls of gas, and they do rotate, but not as a solid body. They rotate differentially because, of course, they're made of gas. And we can use the oscillations of stars to measure how much their internal rotation is, how rapidly they're spinning on the inside compared to on the outside. And then the final thing that I'll mention very briefly is ages. So how old is a star? It's actually quite hard to tell. We can use theoretical calculations. We know that our sun is about 4.5 billion years old, but we basically know that by measuring the age of the Earth from the rocks. So the idea is that as a star evolves over time, it turns hydrogen into helium on the inside of the star. And when hydrogen changes into helium, the speed of sound changes, and therefore the oscillations also change, because these oscillations are standing waves of sound. Now, if this was a public talk at this point, I would breathe a helium balloon and make my voice go funny, but you get the idea. So by measuring the oscillation frequencies, we can tell how old a star is, and that's really a very useful measurement to be able to make. So the field of astroseismology is still in its infancy. We've been working hard over the last few decades on this, and we've got plenty more things to do. So I've already mentioned Kepler, this wonderful spacecraft. That's not working anymore, but it has been succeeded by another spacecraft called TESS, which is up there at the moment observing thousands and thousands of stars. And finally, there's a nice ground-based network which we're involved in called SONG, which is using the first method, the velocity method, to measure oscillations in stars. And as you can see, there's one of those in Australia. In fact, it's at the University of Southern Queensland being built at the moment. So thank you very much, and I wish you all well, and I hope we can meet in person very soon.
It is now my great pleasure to welcome Professor Tim Bedding with us live today. Congratulations, Tim, and thank you for a fantastic presentation.
Thank you very much. It's a great honour, and I'd, of course, like to thank the Academy and also my wonderful colleagues and students over the years.
Thank you. Look, one of the advantages of online presentations is that we've been able to see some of those fantastic things. I'm not sure that we can have flames and whatnot in the Shine Dome. It might test our workplace health and safety rules, but it's so good to be able to bring that to life. It really resonated with me, and I'm sure with many others. If our audience would like to ask questions, please pop them into the chat. Indeed, one's just come in, so I'll ask that one first. What do you hope to learn from TESS that you couldn't with Kepler?
So TESS is going to cover the entire sky, whereas Kepler was focused mainly on one relatively small patch of it. An interesting patch, but the sky is very different in the Milky Way galaxy. So with TESS, we'll be able to see stars in different parts of our galaxy, measure their ages, and understand much better the structure of our own galaxy, how it formed, and how some bits are older than others, and how it all came together. That's a process they like to call galactic archaeology.
Fantastic. So other than discovering new planets, which is really very, very cool, I've got to say, what other applications outside of learning about the stars does this have?
Yeah, I wondered if you'd ask me that question. And I'm tempted to say, for example, that we need to understand our sun much better to understand its future, for example, related to climate change and indeed the solar cycles. And that's, of course, true. But the main thing, of course, is this is pure science. This is really understanding the stars as well as we can for science's sake. But it's another example. We've had several talks already today about resonance and oscillations. And this is just another example of a beautiful oscillating system. And so I think we see it as a sort of special case of what we can learn about oscillations in general.
Yeah. Can you clarify, when you're determining the age of stars, what do you use for a reference for the oscillations? Is it the sun?
Yes. So the sun is, of course, our best studied star. It's right next door. And with the sun, we can see more types of oscillations because we can see the disk of the sun. So we can actually see patches of the sun oscillating in and out. Whereas for our other stars, they're so far away that we really only see them as a single point of light.
Yeah, right. So obviously international collaborations are terribly important for this work, especially when you're using multilaterally operated observatories or accessing space telescopes. Has COVID-19 impacted these collaborations, or is much of the work accessible remotely?
Yes and no. So I think the main impact is simply not being able to travel and see our colleagues and go to conferences. We are amazingly still able to observe. In fact, just a few weeks ago, we observed remotely from a telescope in Chile, and obviously the space telescopes are quite happily going around. So I think the main problem is really, for example, students who are not allowed to come or haven't been allowed to come on campus and interact.
It has been very difficult for our younger scientists, there's no doubt. I have a question from Professor Malcolm Sandbridge. He writes, great talk, Tim, thank you. When you say that you measure the age of the star from the free oscillations, are you actually estimating the internal structure of the star and inferring the age from that?
Yes. So basically, we compare the oscillation frequencies with those from a theoretical model involved in a computer. And you evolve the model through several billion years and measure its oscillations, which of course change with time because its structure's changing. So it's a very much an iterative process, backwards and forwards between oscillations and observations.
Thank you, Tim. I'm afraid that's all we have time for today. Congratulations again on your election to the Fellowship of the Australian Academy of Science, and we look forward to welcoming you at the Shine Dome.
Thank you very much.
Now we'll take a short break and be back at 12:45 p.m. for session 3, which will be hosted by my colleague, Academy Director of Communications and Outreach, Paul Richards. And I'll be back with you tomorrow afternoon for more presentations from our inspiring fellows. Thank you, and we'll see you a bit later.
Welcome back. I'm Paul Richards, Director of Communications and Outreach at the Academy. Thank you to Anna Maria Arabia for taking us through this morning's presentations. I'm delighted to be able to fill this role for the afternoon. Thank you all for submitting your questions for our fellows. Don't forget you can ask questions throughout the event by using the Q&A panel on your screen. Now on to our next presentation. Please welcome back Professor Veena Sahajwalla to introduce our next fellow.
Thank you. Next, we will hear from Professor Peter Corke. Peter Corke has made significant contributions to the field of robotic vision, in which he has developed a solid foundation for the theory and practice of visual servoing, a topic on which he is the world authority. Now to Peter's presentation.
Hello, I'm Peter Corke. I originally trained as an electrical engineer, but for the last 30-something years, I've worked in the field of robotics at CSIRO and at universities here and overseas. As a roboticist, I am continually struck at the contrast between the ability of robot systems that we are able to build today and the capabilities of human beings. And in this short talk today, I'd like to focus on one particular area of competence gap, and that's in the area of hand-eye coordination. The industrial robots that you see here are a very mature technology, over 60 years old now. These robots are fast, strong, accurate, and relentless. What's not apparent is that these robots have got very limited understanding of what's around them. They're not using hand-eye coordination as we know it. What they're doing is relying on their innate accuracy and the fact that manufacturing engineers have designed their entire world so that a machine that cannot see but is very accurate is able to perform useful tasks. This is in great contrast to the way human beings go about things. We use our eyes to understand the state of the world, and we use our eyes to do a task like threading a needle, even though we don't have very precise positioning capability in our fingers, and nor do we have very precise location capability in our eyes. In fact, we can thread cotton into the hole in a needle without knowing where the end of the cotton is or where the hole in the needle is. We just move them together so that the cotton goes through the hole. Human beings solve problems in very, very different ways to the way robots solve them. And there's a whole class of problems that are really very dynamic. And this is an interesting characteristic of our vision system, is that we're able to predict what's going to happen in the near-term future. And this is really very critical for our survival. We need to know not just where things are now, but where they'll be in the future. And here are some examples of some crazy driving and acrobatic skills. This sense of vision I find completely fascinating, and the sense of vision is 550 million years old. It first appeared during the Cambrian explosion, and today there's all manner of different types of eyes in organisms that are in existence right now. So we have two eyes in the front of our head, but there are many other configurations that you can see here. Insects have thousands of very simple eyes, spiders have 4 eyes, and many underwater creatures have eyes designed on quite different principles to our own eyes. Vision really enables life. Almost all organisms today have eyes, and they use it for the most important tasks. So here you see a bee, a simple creature with a 1-gram brain, which is able to follow instructions, fly from its hive to flower, land on a moving flower, harvest the pollen and nectar, and return to its hive. All of that with a tiny brain. Humans have much bigger brains, about 1.5 kilos of brain. A third of that is dedicated to processing visual information. So to me, vision is a very effective solution to the problems and challenges of life. And the fact that we invest a third of our brain, 6 watts of power to power the visual part of our brain, all the delicate apparatus like eyes and eyelids and eyelashes and tear ducts, all of this stuff is clearly a good trade. Yes, it's complex apparatus to have, but it gives us a wonderful sense of the world from very close to our eyes out to the horizon, what's happening now and what's happening into the future. It's a wonderful solution to the problems of life. So for a long, long time, I've been interested in how can we emulate even some of these capabilities. And some work that I first started in 1988 with a robot arm just trying to track a moving object. And that really set me off on the road to try and understand how we can make high-performance visual tracking and then many other tasks that can be derived from that. CSIRO, some years later, we started looking at how we could build a flying machine, a quite primitive helicopter by today's standards, And it was entirely controlled using the sense of vision. So it had a pair of cameras on board. It could sense how it was moving with respect to the world and how high it was above the ground. Critical skills or competencies for a flying machine. There was no GPS involved in controlling this particular helicopter. And we took that same technology and we applied it to an underwater vehicle. This is in conjunction with my colleague Matthew Dunbabin. So this vehicle has got a number of cameras on board. It looks down, it can sense how it's moving with respect to the seabed, how high it is above the seabed, and front-looking cameras give it some idea about impending obstacles. More modern versions of that work have ended up in a prototype vehicle for injecting crown of thorns starfish. And the vehicle on the right hand side is again a much more polished version that can perform quite sophisticated marine surveys. But the fundamental architecture is still the same. Two cameras looking down to give it a sense of where it is, two cameras looking forward to look for upcoming obstacles. A little while later, we entered into a competition run by Amazon, and the idea of this competition was to have a robot that could recognize a wide variety of everyday objects, pick them up and move them to an appropriate place depending on where they are. And again, this is a task that a very small child could do. It's still a difficult task for a robot to do. And so we made some progress on this particular task, and in the year 2017 we won the competition. This is the work of one of my PhD students, Fangyi Zhang, and what you see here is a robot that's using information from a camera through a deep neural network to directly command the speed of the robot's joints. And this robot's been trained to reach always towards the blue cube. So wherever the blue cube is, irrespective of the distractions all around it, it is able to reach relentlessly for that blue cube. Some of this work was commercialized by some people from my lab. They formed a company called CSIRO, and they're looking at the problem of how do you grab and pack irregular shaped objects like fruit in a packing shed. And this is a problem at the moment trying to find suitable labor to do these tasks in remote packing sheds. So it's an example of what a robot with hand-eye coordination could do. Another of my students, Rob Lee, has been looking at the problem of how do I manipulate flexible material. So here you see a piece of rope. It always starts off in the horizontal direction, and in each of the three videos there, the robot is being tasked to pinch, grab, move the piece of rope in order to get it into the desired end shape. Visit to our lab, Patrick did some work on handing objects over to the robot. So again, it's like the problem of grasping, but now we don't quite know where the object is in space. It's wherever the human happens to leave it, and there's clearly a safety angle here that we want to keep the human as safe as we possibly can. So another of my students has been looking at the whole problem of where it is that we place the camera, and although we are built with eyes in our heads and fingers on the ends of our hands, there's no reason that a robot has to be built the same way. So we're thinking, well, what if we put the camera, the eyes, actually next to the fingers? And that's actually got some really interesting advantages. So what you see here is a hand-eye view of a robot picking up a cube, and the camera is now mounted between the fingertips. And this is another example of the robot coming in and grabbing that cube and lifting it up and taking it away. And actually the trajectory of the cube, it's not static in the world, it's actually moving in a somewhat random way, the way that's not known to the robot that's doing the grasping. So as it comes in, it's got to see where that cube is, pick it up and grab it and take it away. So this is the work of my PhD student, Ben Burgess-Limerick. Other work by my student Jesse Haviland is to integrate some of this robot grasping with mobility. So here the robot comes in, reaches down, can grab the particular object, and then plan a time-efficient path and drop it off in a tote on the other side of the room. So that's a really quick run through of some of the challenges, the deficiencies, if you like, of robots trying to perform hand-eye coordination, a wonderful intrinsic skill of human beings, one that I think we continue to underestimate, and my research is all about how can we give that very useful capability to robots. I think that we human beings are proof that a machine with just 1 or 2 cameras could be almost as capable as we are. But at the moment, there's a huge gap between what a robot with 1 or 2 cameras can do compared to what a human being with 2 eyes is able to do. So a huge number of challenges still ahead of us. The challenge is not in the cameras, and it's really not necessarily in the computers. It's really understanding how do we process the rich sensory data that comes from the various sensing modalities, from cameras, from tactile and haptic information, and integrate that into a robust control framework. So there's still an awful lot of work to do. Thank you.
And joining us live now is Professor Peter Corke. Welcome, Peter, and congratulations.
Thank you very much, Paul.
So we've got some questions here for you. And a reminder, if you're watching along and have a question for Peter, please enter it into the Q&A panel on your screen. Peter, just to start off with, so many amazing breakthroughs throughout your career. Are there any in particular that stand out so far?
It's hard to pick a favourite. It's interesting when you look back, and I'm just appalled at the technology that I had to put up with at the time, and think if only I had what I have today, I could have done that thing 20 years ago so much better. So no favourites.
And, you know, you showed some of your work from back in the early 2000s of the helicopter flying using cameras to sense its surroundings. How far has the technology come since then, for instance?
It's come an amazing distance. So there are companies now that sell a drone that will basically follow you. So, you know, the model, you go skateboarding or you go downhill skiing and your drone follows you and comes behind you and moves in front of you and takes videos of you doing everyday things all by itself. So, you know, that's sort of as far as the way that that technology has advanced. There are other companies who are using this visual navigation technology to fly drones in underground domains. So, you know, big applications in mining. If you can fly a drone using visual information, because you don't have GPS in an underground, deep underground. So there's lots of potential and starting to see commercial outcomes.
And I suppose autonomous cars would be one area that most of us might be sort of familiar with this kind of technology. Would it be relevant in that case?
Correct. So Tesla in particular have made quite a big punt on the use of cameras to guide their cars, as opposed to the other companies. Generally have all followed a different approach. But Elon thinks cameras are enough, and I agree with him on that. I think they've had to add a few other things like radars, which are going to be useful when it's foggy or it's heavily raining. But yeah, quite a strong emphasis on cameras to guide those cars.
What areas of teaching robots hand-eye coordination are proving the most challenging at the moment?
I think it is the fact that when we pick things up, it's a rich mix of different sensory modalities. So as we reach toward it, we use our eyes. There becomes a point where our hand is probably in the way, but we just keep moving sort of out of habit towards it. And then at some point we make contact, physical contact with it. Right? And then we use our sense of touch to do the last part. And I think this is something that we haven't really nailed. It's the transition from when I can see it, but I'm not touching it, to when I'm touching it, but I can't see it. And I think in the future, that's an area where I'm going to be spending some time, but also to do this stuff really quickly. The robots that I showed, still too slow to my liking. To be cost-effective robots have got to be wickedly fast.
Yeah, interesting. And how do you overcome the challenge of processing sensory data and integrate it into a control framework? Is it a matter of coding new artificial intelligence systems, or what goes into that?
So these days, almost all of the approaches we're using are artificial intelligence-based approaches, deep learning techniques. Some of these things take a very long time to train. The example I showed of the robot reaching for the blue cube, that probably took weeks to train, but actually is able to make the decisions within 10 to 20 milliseconds. So there's a big disparity between training time, which you only have to do once, and the cost at runtime.
Right. And one of the areas that you showed us is that technology being commercialised is in fruit packing. Where else are we likely to see it in the near future?
I think the other big area where we're going to see it is going to be in e-commerce and logistics, where E-commerce has come from nowhere in the last decade, right? And with lockdown, the amount of e-commerce that's happening is growing and growing. There are more parcels to be shipped from one part of the world to another. Handling all of those parcels, routing those parcels through postal centres, this is going to be a very big application for robots with this kind of skill.
And we've just had a question come in from Malcolm Sambridge, who wanted to know, is there a trade-off between the numbers of eyes or sensors and the level of sophistication of visual processing required? For example, can we make the processing problem simpler by having more eyes?
This is a really good question, and this is what biology does. So there's so many different sorts of eyes in nature, and the species has evolved eyes that help it do its task well. So, you know, a squid has got a particular sort of eyes, a shrimp has got different eyes, dragonfly's got different eyes, and they are optimized to provide the information that organism needs in its ecological niche. And it's probably a trick that roboticists should take more notice of. We tend to build things in our own image, and that's not necessarily the best solution for a particular problem.
Terrific. Well, I could talk to you for hours, Peter, but that's all we do have time for today. Thank you so much, and congratulations again.
Thank you very much.
Now, to introduce our next fellow, please welcome Academy Secretary for Education and Public Awareness, Professor Hans Bachor.
Thank you. Our next presentation is from Professor Robyn Owens. Robyn has an exceptional record of innovation in computational vision science, including groundbreaking fundamental theory in feature detection and object recognition, and applying these theories across diverse disciplines, from biomedical research to face recognition. Robyn also has outstanding achievements in research training and research policy development. Please welcome Robyn.
Hello, my name is Robyn Owens, and it's my pleasure to spend the next 10 minutes talking to you about the ideas of abstraction, representation, and how we understand what's represented in an image. And nobody said this better than the French painter René Magritte in his work Ceci n'est pas une pipe, which of course is not a pipe. It is a flat, planar surface covered with a distribution of paints to represent different colours and levels of luminance. And yet our brain has no trouble understanding the three-dimensional structure that's represented there. In computer vision, it's our aim to give the same capabilities to cameras, computers, and robots, so that, for example, in a crowded scene like this, a computer could understand what is a car, what is a truck, what is a person, what is a building, and how to navigate itself through such a structure. My journey began at the University of Western Australia, illustrated at the top left-hand corner by an image chosen deliberately to show how the laws of perspective allow us to interpret a three-dimensional shape. I then went on to Oxford University to do my master's and doctorate, and I've illustrated that at the bottom left with a line drawing of the buildings in Oxford, and you can see that just the edges of structures also give you a lot of three-dimensional information. Following Oxford, I spent 3 years in Paris doing more mathematics, and I've illustrated that with an image that shows how digitized and pixelized our images are, both on the retina and in a computer. And still, just with that amount of information, we can understand what is being represented. My first paper was actually in French on a rather abstract topic called almost periodic functions. And yet this work eventually tied into much of the work that I did in computer vision. So let me start with one of the first problems that we looked at, which was trying to understand the optical illusion of Mach bands. Mach bands appear at the edges of shadows. And on the left-hand side is an illustration of luminance values going from dark on the left, ramping up to a medium grey, flat in the middle, and then ramping up to a brighter shade of grey on the right-hand side. And if you look carefully, what you can see along with these shades of grey are some specific lines. For example, at the beginning of the ramp on the left-hand side, there is a dark band. At the beginning of the flat plateau on the right-hand side, there is a white, bright band. These artifacts are not, in fact, part of the image. It is part of what we see through an optical illusion known as MAC bands, and they appear also in X-ray images, and specialists need to understand that they appear and how to interpret them. Our job was to understand why these occurred. And that work led to one of my first papers that appeared in Nature, written with Concetta Moroney, John Ross, and David Burr. The idea here was to look at a different abstraction for how edges may be represented in images. Normally they're represented through looking for places where change occurs, and so often derivatives are the best way of measuring where that would be. Our approach was to look at how edges could be made up by a sequence of periodic waveforms. And on the left, we can see how by adding an infinite number of sine waves of ever diminishing amplitude and ever increasing frequency, we can build a step function. Likewise, on the right, we can build a ridge function using cosine values. And this led us to have an insight as to why these MAC bands would occur, because if you only add a finite number of these curves, you get an overshoot at the edges. It also led us on to a brand new way of looking for features in images, such as edges, ridges, corners, and many other features. This paper was published in Pattern Recognition Letters in 1987. Now I'd like to give you some idea of some of the work that followed on from this initial foundational theoretical work and some of the applications that I've worked in. This particular application was work done over a number of years with Unjung Holden, represented at the bottom left-hand corner. She is now a professor in geological image understanding at the University of Western Australia. But this particular work was around understanding of the Australian Sign Language, Auslan. And over a number of years, we built a system that could look at an image of a person signing, track the movement of the hand and the face and the lips, and decode what was being signed and translate it into English. Additionally, we built an animation that was capable of translating English into signed Auslan. And this work was awarded a prize at the Asia Pacific IT Awards in 1999. Another area of work that I was engaged in over many years was with Professor Peter Hartmann in our Department of Biochemistry, a world expert in understanding how lactation works. In this work, what we used is a projector projecting stripes of light onto the human body, stripes of light patterned in different binary patterns, and these flat stripes change according to the shape that they fall on. These changes in shape can then help the computer build a three-dimensional model of that shape. And through this work, we were able to understand how milk is produced, how quickly it is produced, what the content of the milk is, and many other aspects of lactation. This work was awarded the Rank Prize in 2010, and Professor Peter Hartmann is there illustrated second from the right. Another application was work in face recognition, and this work was done with Professor Ajmal Mian, illustrated there at the bottom on the left. In this work, we're trying to understand and recognize individual faces. You'll all be familiar with this because we have face recognition embedded in our passports now. The work here was to move from simple features such as the edges of our eyes and lips and nose onto more abstract features such as the curvature on the surface of the face. And this particular work used both two-dimensional and three-dimensional attributes of the object in order to build a model that was matched against a database of faces. This work was published in the IEEE Transactions of Pattern Analysis and Machine Intelligence, and in many other papers as well. Of course, for the last 10 years of my career, I worked as the Deputy Vice-Chancellor for Research at the University of Western Australia. And during that time, I was involved in much scientific work across the whole nation, particularly with regards to NCRIS infrastructure. And I had the privilege of sitting on boards of many of our infrastructure capabilities and working with many reviews. I also had the privilege of chairing the Deputy Vice-Chancellor Research Committee for Universities Australia and was very pleased to be involved in the development of research strategy. And just at the bottom is represented one of the major reviews that was written by the collection of academies on Australia's research training and the many recommendations that we provided to enhance the quality of research training for the many careers that research students may have. Thank you very much for this opportunity to speak to you today.
And Professor Robyn Owens joins us live now. Congratulations, Robyn, and thank you for your presentation.
Thank you.
We've got some questions for you. To start off with, I wanted to ask about some examples of how some of your projects or collaborations that you've been involved with have come about. Can you tell us about how they occurred?
Well, I'll speak briefly just about one, and that's the work on lactation, which might seem rather strange for a computer scientist to be working on, but it resulted from an article for the general public that was published just talking about the work on feature detection. And it really had some very simple images, looking at the edges of a coffee cup. And Peter Hartman read this, and at that stage he was trying to understand how he could measure the change in shape of the human breast. And he'd been looking at models where engineers were trying to measure the size of iron ore deposits and not making very much progress. And so he contacted me to see if we could use structured light to in that application. And it worked very well.
And it's just a great example of the way that some of these connections can be made. You never quite know how through the publicity of your research that new opportunities might occur. So that's wonderful. How has rapid technological change provided greater possibilities in the type of research that you've been able to undertake, or the manner in which you do it?
Well, I think the big movements have been developments in cameras, particularly the amount of data that can be captured, but also you saw in one of my applications the use of three-dimensional technology. So whether you're using stereo cameras or laser technology, and then moving outside of the human visual system with hyperspectral cameras moving into infrared or ultraviolet or beyond light. So there are many opportunities to enhance human vision with enhanced technology.
What are some of the applications then for your work that you haven't had a chance to explore yet that may be of interest in the future?
Well, I think there's an awful lot of work that can be done in things like autonomous vehicles. Obviously, there's a lot of work there. And I am working with a PhD student at the moment in the area of adversarial images. So if you've got a computer, a camera, looking at the world as it's changing and having to read road signs and take advantage of the information it has in its environment, you also have the problem that a malicious attack could occur. Somebody could embed in an image something that's designed specifically to confuse a robot, something that a human may not even see. Believe it or not, this is a huge area of work at the moment. So dealing with adversarial images and safety is one of the issues that has to be dealt with.
Yeah, interesting. And actually, there's a question that's just come through that might take that further from Francis Saparovich. She says, congratulations, Robyn. There's been some discussion about the need for greater diversity in those working in computer science, especially after facial recognition errors. Could you comment further about how this is changing?
Absolutely. It's a huge area. So facial recognition has actually been touted as being quite successful with very high recognition rates. But let me just say those high recognition rates are if you're male and if you're white. And the lowest recognition rates are if you are coloured and female. And that is a lot dependent on the sort of training that is given to the initial database. So there's a huge amount of capacity for innate cognitive biases that all humans have to be built into the training of many of these AI techniques. And I think it's certainly a real reason why we need diversity in all of these areas. And I think a clear focus on the impact of ethics in training robots in general. If we're ever to get to a stage where we can trust a robot, we probably have to figure out how we can trust each other, first of all.
Yeah, really worthwhile discussion and interesting to see where the work is yet still to be done. And just one final one, Robyn, along the same lines around the quality of research training. Are there other aspects that you would consider to be important or would recommend are looked into in future?
Look, I think Australia has a fantastic research training environment and PhD students get a terrific opportunity. But the thing that's happened over the last few decades has been the massification of this. So when we were doing our review, at any single year, there were 80,000 students enrolled in PhDs across the country, 10,000 graduating every day, and only 10% of those graduates would ever end up with a tenured academic job. I think a lot of our research training is designed to form new academics or new researchers. And we need to think, really, is that where they're all going to end up? And if not, what sort of jobs they're going to get into and whether we need to rethink that research training for the sort of jobs that they'll get.
Well, wonderful talking to you, Robyn. Congratulations once again.
Thank you very much.
Please now welcome back Professor Malcolm Sambridge to introduce our next presentation.
Thank you. Our next presentation is from Professor Ping Koi Lam. Ping Koi has an international reputation in secure quantum communications and quantum state generation. He was the first to demonstrate successful end-to-end quantum key distribution in Australia and has established a globally recognized web server for quantum random number generation. We will now hear from Ping Koi.
Good day. My name is Ping Koi Lam. It is a real honor to be elected Fellow of the Australian Academy of Science. I'm a quantum physicist, and in the next few minutes, I'm going to talk about my research at the Australian National University, from nothingness to quantum communication technology. I would like to start with a quote from the renowned philosopher René Descartes, who said, "It is contrary to reason to say that there is a vacuum or space in which there is absolutely nothing." Although Descartes argued from a purely metaphysical perspective, during the time when Isaac Newton was still an infant, in 1644, he was absolutely right. This profound statement is only experimentally proven centuries later. Vacuum is a region of space that contains no real physical matter. My research at the ANU focuses on studying the photonic vacuum, a region of space where there is no light, it's perfectly dark, and where there is no detectable photon. According to quantum theory, however, a vacuum is not completely devoid of energy or noise. In fact, famous physicist Paul Dirac said that the vacuum is like a sea of virtual particles, where all sorts of fundamental particles would spontaneously appear and disappear all the time. Even though we cannot catch them, their presence can be felt. In quantum field theory, the vacuum state has a field that contains a small zero-point energy. This zero-point energy manifests itself in many different ways. It is the vacuum noise that contaminates measurements and sets limit to how accurate we can measure nature. This is also known as the vacuum fluctuations. It can also exist as a minute but noticeable force that causes conductors to attract each other. This is referred to as the Casimir poles. At the ANU, my lab has sensitive detectors made for measuring vacuum fluctuations. We are able to use a laser to amplify the vacuum fluctuations, measure it, record it, and even map its complete statistics using tomography. In the Fourier domain, we see that the vacuum fluctuations permeate the entire electromagnetic spectrum. We find that every frequency will have its own independent quantum mix that is unique and random, even in parts of the electromagnetic spectrum where there are absolutely no light. In fact, we can argue that vacuum fluctuations are the purest source of randomness nature has given us. If we were to digitize the measurements in the lab, we can transform vacuum fluctuations into strings of random numbers in binary, hexadecimal, typographical symbols, or even in colors. The randomness generated here is quintessentially quantum in nature. It does not require mechanical parts. It does not repeat itself. It is unbiased, and it is completely indeterminate. Personally, I find these random numbers produced by nothingness fascinating and beautiful. Truly reliable, unbiased random numbers can have many uses. It can help researchers, engineers, and computer scientists with numerical modeling, from modeling the chaotic systems of climate to financial stock market. It can be used by cryptographer to generate encryption keys, help artists and creative content users to make impartial decisions. 10 years ago, we decided to set up a web page. We wired our vacuum measurements to the internet and provide free access of these random numbers globally. The service is provided in real time, 24 hours a day, 7 days a week. To date, the ANU random number generator has been accessed regularly by more than 70 countries. And we have received over 300 million reads for strings of randomness. With an adequate understanding of the origin of quantum noise, we can use optical crystals to manipulate vacuum fluctuations using nonlinear processes. An efficient nonlinear optical process can transform the ubiquitous quantum noise into a range of interesting quantum light states. Some of the examples are squeezed light, a state of light that can be quieter than vacuum. This state is now used for detecting astronomical gravitational wave. We can also generate a smaller version of the Schrödinger cat, the dead or alive state, or the famous Einstein-Podolsky-Rosen spooky action at a distance entanglement. These states are fundamental resources for many quantum computing and quantum communication applications. The pictures shown on this slides are some examples of the tomographic reconstructions of the quasi-probability distributions of these very interesting quantum states. By distributing quantum states of light that are encoded with information between a sender, Alice, and a receiver, Bob, we are able to use quantum noise as an encryption strategy to protect information. When light travels between a sender and a receiver, information may be lost in transit. Theoretically, this loss can be exploited by an eavesdropper Eve, as shown in the diagram. An objective of quantum communication is to protect the communication link between Alice and Bob from potential eavesdropping. In conventional encryptions, communication security is reliant on mathematical complexity. This means that communication may be compromised if an eavesdropper has immense computational resources. In quantum encryption, however, communication security is guaranteed by the laws of physics. Because the laws of physics cannot be broken, quantum encryption is essentially an unbreakable code. At the ANU, my research group has invented a method of using quantum states of bright laser beams to establish ultra-secure communication between Alice and Bob. We encode an encryption key on the radio frequency sideband of a laser. This key can then be transmitted using optical fiber or in free space. With this system, we demonstrated that if all of the losses in transmission were fully intercepted and exploited by Eve, the eavesdropper, she would still be unable to extract even one bit of information from the entire communication. This form of quantum communication technology, also known as quantum key distribution, is now a commercial reality, and Australia is amongst a handful of nations worldwide with our own quantum encryption capability. Over the past few years, we witnessed NASA, ESA, the German Aerospace Center, and many other space agencies setting up their own quantum programs to launch a range of quantum technology demonstrations in space. One of the objectives of these quantum programs is to realize satellite laser communications that are quantum compatible. This will help build a communication system would one day promote global quantum encryption and kickstart a new quantum internet. Australia has direct access to the southern sky, and for this, our country can play an important role in the future space communication network. To me, this is an exciting time to involve in quantum space research and development. I thank you for your attention.
And now we're delighted to have with us Professor Ping Koi Lam. Congratulations, Ping Koi, and thank you very much for your presentation.
Thank you. It's a real honor to be here. And I would like to take this opportunity to thank all my collaborators and students, particularly Michelle Simmons, Hans Bakor, and Tim Ralph, who has been real help in my career.
Absolutely. No, thank you for that. Now, we have some questions for you from the audience. The first one here asks, what sets quantum communication in space apart from other forms of communication?
Quantum communications in space relies on transmission of quantum states. We are not actually looking at sending information per se. We're looking at either generating an encryption code so that this code can be used to protect classical information, the normal information that is sent between ground and space. There's another version of quantum communication in space, and that has to do with distributing quantum information between quantum computers. So people are talking about building quantum computers, and just like the modern internet, eventually we would like to link all these quantum computers together. And the way to transmit quantum information between these computers is to use quantum communications.
And I think you partly answered my next question, which is how widely applicable is quantum encryption, and how can it be used here on Earth?
Currently, there is still some technological barriers that we have to overcome. Quantum encryption has a limited range. The longest range now is between two cities that are separated by hundreds or even 1,000 kilometers. And so this is why I'm now working in the Center of Excellence that looked into extending the quantum communication range from hundreds and thousands of kilometers to intercontinental. And the way to do that is for us to build a quantum repeater. So this is just like the quantum analog of the optical fiber repeater stations that we find everywhere around the Earth, that you can actually relay information from one point to another.
You just mentioned some of the barriers that remain in terms of making the technology used widely. Do you have any idea of when to expect quantum encryption to become widely used?
I would say that there are already companies that use quantum encryption in its various form. To make it global, I think this is really dependent on the interests, I guess, of industry and government. And that's why in my presentation, I mentioned that big organizations such as ESA and NASA are looking into building this really big infrastructure that would enable global transmission of this type of information and encryption.
Are there any particular discoveries that you've made throughout your career that are most satisfying?
I think they are all really satisfying. I find quantum physics really amazing. To see things that you would find in science fiction actually happening in front of you, to build a beam of light that is producing noise that is quieter than darkness, to me, is still quite amazing. And Einstein-Podolsky-Rosen entanglement, Schrödinger cat, all these textbook example of the weird and wonderful microscopic world actually can be seen in the lab. It's really fascinating.
And you were saying also in your presentation, just the beauty in the colors and the randomness, and the fact that that comes actually from nothingness as well is just a beautiful thing to admire. Are there other ways that it can be represented and seen?
Once you've converted it into digital information, you can do it as you please. And that's why our website has quite a lot of hobbies and artists, as well as the famous researchers and engineers who are accessing our random numbers.
Well, Pinkoy, it is remarkable work. Thank you very much for your presentation and time this afternoon, and congratulations again.
Thank you.
Now, to introduce our next fellow, please welcome Academy Diversity and Inclusion spokesperson, Professor Sue O'Reilly.
Thank you. Next, we will hear from Professor Ian Reid. Ian's works in computer vision have made landmark contributions in visual geometry, tracking, novel view synthesis, visual surveillance, and simultaneous localization and mapping. His emphasis on real-time processing and systems work means that he is one of few researchers worldwide to be active in both computer vision and robotics. His work has been instrumental in bringing computer vision into the mainstream of the robotics literature. And now to Ian's presentation.
Good afternoon. My name is Ian Reid, and I'm a Professor of Computer Science at the University of Adelaide. I'm deeply humbled to have been elected to join so many eminent scientists in the Academy. I'm very grateful for this short opportunity to present a little bit about my research over the last 30 years in the area of computer vision. Before I go any further, I want to acknowledge the contribution of an amazing bunch of engineers and computer scientists that I've had the privilege of working with, including three of my fellow presenters this afternoon, especially Robyn Owens, who was my honours thesis supervisor and set me on this research path, and Peter Corke, with whom I've spent the last 7 years working in the ARC Centre of Excellence for Robotic Vision. The idea that computers can be made to see by connecting them up to cameras is a very appealing one that has its roots right at the beginning of artificial intelligence research. In my own work, I've often used the motivation of the camera as a sensor for a robot. Robots are blind. To operate in a dynamic and certain environment, autonomous systems must be able to sense and understand, to perceive their surroundings. Visual sensing is appealing for a number of reasons. Cameras are cheap and ubiquitous, and they're passive. They're typically low power consumption. Video conveys a vast amount of data about the world in real time. And above all, cameras hold the promise of flexibility. There's a number of challenges of using cameras as sensors, however, and this is why it's an interesting and hot topic for research. We have to process a vast amount of data. Much of it is redundant. Information is lost in projecting from a 3D world to 2D images. Although the human visual system is an existence proof that such sensing is possible, we have no clear idea of how the human visual system actually works. And in particular, how we use our experience and our knowledge of the world to make sense of the light that falls on our retina. Computer vision is a study of mathematics and algorithms for processing images and video to infer scene knowledge and understanding, and ultimately to enable robotic interaction. It combines ideas from a range of fields such as computer graphics, algorithms and representations, mathematical ideas such as probability and optimization. Two of these areas stand out for me as having led to step changes in the capability of computer and robotic vision systems, and therefore they've been the focus of much of my research. The first, geometry, came to the fore in computer vision in the mid-1990s to 2000s and involved gaining a clear understanding of perspective geometry. Through this understanding, we've been able to address questions such as, where is the camera? And to create a map of the world as the camera moves through it. The second overlapping and hugely influential field is that of machine learning, beginning in the mid-2000s, but accelerating exponentially over the last decade with the rise of deep learning. The success of deep learning at solving tasks like visual object recognition and image segmentation means that the moving camera can now not only deliver geometry, but also an understanding of the scene. Let me begin with some examples of extracting geometry from images. The challenge is that in projecting from 3D to 2D, information is lost. The Renaissance painters understood the projection process, so let me take you to the Church of Santa Maria Novella in Florence, where Masaccio in 1425 is credited with the first example of a true perspective painting. With my first ever PhD student and a colleague in Oxford, we developed methods for taking accurate 3D measurements using projective invariants. These measurements were enough to create a 3D representation of the scene that Masaccio envisaged. If instead of a single image, we consider an image sequence from a moving camera, we now have the so-called structure from motion problem. In the mid-2000s, my colleague Andrew Davison and I showed that successive 2D measurements of a scene point as it's tracked through the image sequence, such as you can see in red and blue in this image on the left, can be fused using the extended Kalman filter to create a real-time structure from motion system. You see on the right, the structure of the scene being calculated in real time with ellipsoids showing the uncertainty in those measurements. This simultaneous localization and mapping solution, or VSLAM for short, turns the moving camera into a powerful sensor of camera location and scene structure. And as well as for robotic applications, such as robot vacuum cleaners and self-driving cars, knowledge of the camera location can be used for augmenting reality, such as is shown in this early video on the bottom right. You see here the camera is being tracked using the features marked in red and blue. And because we know where the camera location is, a graphics object can be placed in the scene as if it were properly interacting with the flat surface in the scene. Of course, the power and attraction of using a camera as a robotic sensor is that it can deliver more than just geometry. Images and video inform us about what objects and what regions are in the scene and where they are, potentially allowing a full understanding of the scene to be developed. It's here that the power of deep learning becomes apparent. In these images, you see application of an algorithm we developed for classifying every pixel in an image or video into one of many classes. Such a system is trained by showing it many images of, say, street scenes, along with painstakingly hand-labeled ground truth until the network can predict the pixel labels as accurately as possible. With a suitably designed network and a sufficiently rich training set, this will then generalize to new unseen images. And you can see two examples of that network being trained here, once on the parts of a person and another example of it being trained on the constituent parts of a street scene. So it's segmenting the scene into its the street, the cars, the traffic lights, street signs, and so on. As well as classification tasks, we were able to show how a deep neural network can be used for regression tasks. In the work on the left, a deep neural network is trained on many images and their corresponding depth maps. Remarkably, such a network can do a very good job of predicting the depth of every pixel in an previously unseen 2D image. Once again, the training process is painstaking. So for the work on the right, we recognized that by combining geometry with deep learning, we could create a self-supervised training regime. The key insight is that if we can use the scene geometry to make a prediction of how a future frame will look by warping the current image, we can compare this prediction to the frame that actually occurs. And if there's a discrepancy, we say that it's because we predicted the structure of the scene incorrectly, go back, adjust the network weights so that it predicts better the next time. In this way, our depth from single image can continue to improve indefinitely as the camera moves through the environment. And notice here that this trained on street scenes in a self-supervised manner is even able to pick out the pedestrians as they cross over. Lighter here means closer to the camera, darker means further away. A significant topic of recent research has been what's the best combination of learning and geometry? Are there any advantages to learning tasks that are already well understood geometrically? In some recent work, we've looked at how we can use self-supervised learning to get really good matches between a pair of images and use that to estimate the motion between those pair of images. If we combine that over a long sequence, we can estimate the total motion of the camera. So in this work, we use not only a deep network to compute the relative motion, the optic flow between the images, as you see here and here, but also the depth of the image, which we use to account for speed scale ambiguity that is inherent in structure from motion problems with a single camera. What you see here is the set of matches that have been established between the views using that combination of deep flow and the depth network. And we then pass that into a standard geometric process that estimates the relative motion between each pair of frames. And that is accumulated here to give the trajectory that you see over here on the left. Using this technique, we're able to get amazingly accurate measurements of our own motion, solving the so-called visual odometry problem. You can see here less than 1% drift over nearly a kilometre of motion. In summary then, although when I started working in this field, there were few algorithms that worked on anything but one or two images, computer vision is now sufficiently mature that it's the basis for numerous commercial applications. One of the things that has got us to this point is the use of machine learning for visual scene understanding. But that comes with a number of roadblocks that are topics of current research, and I'll touch on just two of them. The first is this idea of unsupervised or self-supervised learning because of the cost of creating these massively labeled data sets. And the second is that we urgently need methods that are not inscrutable, that provide explanations for their results so that we can develop a level of trust through understanding of the results that our networks produce. Thank you very much.
And please welcome live now with us, Professor Ian Reid. Congratulations, Ian, and thank you very much for your presentation.
Thank you very much. Good to join you. And thank you very much to the Academy for this recognition.
Now, can you tell me, to begin with, just how did you get interested in seeing robots and computers in the first place?
Well, so I alluded to this right at the start of my talk. One of the earlier speakers in this session, Robyn Owens, was my honours thesis supervisor. I was an undergraduate at University of Western Australia. Robyn was recently arrived back from Oxford and Paris, and posed a problem about visual motion as an honours thesis topic. I was interested in that. Sounded like an interesting thing to work on, and I thought I was going to work on it for a year. I did work on it for a year, got excited about doing further research, actually worked with Robyn on Peter Hartman's project for a short period before going to Oxford, and then carried on working on it for the last 30 years.
And as you mentioned, along with Robyn, you've also collaborated with Peter Corke, both who've given presentations this afternoon. Is this an indication of the rise of this area of research and the strength of the expertise in Australia?
Well, absolutely it is, yes. And there's more to come from this field in this session as well. But it really is a recognition that both of the field, which has come great strides. Peter has already alluded to the fact that when we both started working in this field, it didn't really work. There weren't commercial applications. And what we've seen gradually is a step change in the number of commercial applications. What's really changed over the last decade has been the interest from big tech and huge investment, not just from governments in research, but huge investments from Google, Facebook, Amazon, and so forth, the big tech companies that recognise that this technology can add value to their products and services.
Could robot vision be better than human vision one day, or are there ways it's better already?
Well, so there are certainly some ways in which it's better already. The great limitation that both Peter and I have alluded to is that we really don't understand how we can leverage all the prior knowledge that humans have that make us so good at understanding the world around us using the visual sense. But there are certainly things that robot vision can do much better, in the same way that a pocket calculator is much better at calculating numbers. Humans are not able to do visual slam, which is one of the things that I talked about. We can't estimate our motion accurately down to the centimetre. We do it in a much more qualitative sense. And part of our challenge is to work out how we can combine the best of the things that robot vision can do better with the stuff that we don't know how to do that the human visual system does so well.
What kind of industrial or real world settings has your research been used in?
Well, so the various applications, so some going right back to sporting events and special effects, where if you can analyse the motion of a person, then you can use that to create a computer graphics character, or to analyse their motion for sporting purposes. Then through to things like understanding where a vehicle is, so for robot vacuum cleaners or robot lawnmowers, that kind of thing, or for drones, as has already been alluded to, the ability to understand where the entity is using cameras, and also where the entity is relative to other things. So you can have a drone that can track a person. I also alluded briefly in my presentation to the idea of augmented reality. So one of my postdocs who was responsible for the video that you saw went on to become an integral part of the development of Microsoft HoloLens, which is an augmented reality product that uses visual SLAM to locate the user's head and the glasses.
I've had a question sent in here from Malcolm Sambridge, who says, "Congratulations, Ian, a very nice talk. Have a general question prompted by your final comments about the roadblock in machine learning. It is, what value is an algorithm if we don't actually know how it works?"
Well, so that's a very good philosophical point. I mean, I think it's fair to say that if an algorithm works empirically well, then even if we don't know how it works, it does have some value. The trouble is, it doesn't have, I think, enough value for us to be able to deploy it in any safety critical situation and so forth. Perhaps we could employ an algorithm that could predict the stock market without understanding how it worked and make a shed load of cash. Of course, that's what I would have done had I known how to do that. But if we understand how it works, then we can engender a level of trust. And so that's why I think that's a really important topic for future research, that we can build things that are empirically good, but we also need to understand how they work so that we can trust them.
Well, Ian, it's a fascinating field. Thank you very much for your presentation, and congratulations once again.
Thank you very much.
Please now welcome back Professor Hans Bachor to introduce our next fellow presentation.
Thank you. Next, we will hear from Professor Svetha Venkatesh. Svetha is a leading computer scientist who has made fundamental and influential contributions to the field of activity and event recognition in multimedia video. Venkatesh pioneered many theoretical and algorithmic foundations for detecting normal and anomalous activities and deriving meaning from digital video. This underpinning theory has significantly impacted applications, including in automated security and video search, as well as advanced risk prognosis algorithms for medical data. Now we'll hear from Svetha.
Hi, my name is Svetha Venkatesh, and I am a director of the Applied AI Institute at Deakin University in Melbourne. My research interest is in machine learning and artificial intelligence. The Applied AI Institute is based both in Melbourne and Geelong, and we are one of the largest AI and ML teams, and there are many researchers and software engineers who work in this institute. So the way my research has progressed is really to start from failures with data or analysis and then go from there. So one of the kind of classes of problems we've tackled is the detection of outlier events. So these are abnormalities in data. So we try and learn from normal and then predict what could be abnormal in some data. And video surveillance would be one of those examples. Another class of problems we look at is the detection of rare events, especially when it's very, very hard to do. And this is an example of this would be in suicide risk prediction, and we have done quite a lot of work in this area as well. We've also worked in spaces where modeling needs to capture some very intricate issues in the underlying processes by which the data is produced. As examples of that would be the work we've done with electronic medical records and the detection of cerebral palsy by watching the movement of babies. And the last part of what I want to talk about is looking at when there is very little data or lean data, and what is the efficient machine learning we can construct in that space. And the focus of the next few minutes is to talk about this last point, which has also been the focus of the last 7 or 8 years of my life. So I call this efficient navigation of complexity, or disrupting how we innovate, and I'm going to try and explain this a little bit further. So this is Abu al-Hatam, and in 965 he wrote this beautiful book on optics, and he also said that a hypothesis must be proved by experiments based on confirmable procedures. And this is the basis of the scientific method and has been a time-honored process for a very long time. So this kind of experimentation has driven scientific inquiry. So what do we normally do? We perform an experiment, we measure the result, and we change the control variables. And we do this in a loop till we get the target that we need. Now, why is this hard? This is hard if the number of control variables increases, because then it becomes difficult to know what you're going to change in the next experiment, and it's particularly hard to explore all possible combinations that can happen with all the variables you're dealing with. And to give you a sense of this curse of complexity, I want to take an example of just testing a car with 10 variables. It's a toy example. And let's say that each experiment takes about 100 hours and you have about 1,000 experiments to do. The whole process would take more than 12 years to finish. Clearly something that is impossible to do. So your question would be, then how do we test a car? And actually what we do is we simplify, right? But we don't know how far from optimal we are. And this often involves us having to over-engineer our solutions. So to understand this a little bit more as to why this is such a problem, consider this simple graphic where you have the number of experiments required on the y-axis and the number of variables on the x-axis. If you have only 4 or 5 variables, you can use simple charts like Excel. Right? That'll do. If you have 6 or 7, then you can use design of experiment methods which were invented in the Second World War. But if you want to do something better than that, then we suggest that we should use machine learning and come up with these Bayesian methods for this adaptive experimental design. Of course, the number of experiments increase as the number of variables increases, but the rate at which it grows is much, much slower, as you can see here. Now, this is of huge advantage in innovation because it means lower developmental cost and faster market entry. And that is why this is an interesting space for us to do research in machine learning. So the underpinning theory, just to give you a glimpse of this, is Bayesian optimization. So what we have here is a measured property, let's say on the y-axis, and say one control variable on the x-axis. And let's say there's some function that maps these input variable to the measured property. You don't know the shape of the function, and that is why it's called a black box function. It's also expensive to do the experiment in terms of either your resource or time, and therefore the task is to find out the maximum of this function by doing the minimum number of experiments. So how do we do this? We do this by sticking with the best so far. So we kind of approximately know what people have done before, and we stay kind of close to that. And this is called exploitation, and it's a sane thing to do. But the problem with this is that if you keep going where people have gone before, you're not going to discover anything new. So it is important that with a bit of madness, you explore where you don't know what's going to happen. But normally, it's impossible to do this in very large search spaces because you'd get completely lost in this desert of search space. But what Bayesian optimization does is gives you a way to do this by saying, okay, with a small probability, I will explore a place where there may be large uncertainty, but there's a small probability that I get a much better result. And this is called exploration. It's a little bit of madness. And really, Bayesian optimization is a mathematical formalism that combines this exploitation and exploration nicely into mathematical form so that we can use it and prove results of convergence. So how do we actually use it? So in order to disrupt the basis of innovation, it's essentially machine learning to assist experimentalists to efficiently design new products and processes and reduce the time to go from idea to product. So how does it work? So you perform the experiment as normal, you measure the product quality, and then instead of the human being suggesting the next experiment, the machine suggests the next experiment. And you keep going in this loop till the optimal is reached. And the design of such a system, this adaptive experimental design, and the innovations and is really the basis of the innovations that we have done in our group over the last few years. And not only have we advanced machine learning so that we can transfer knowledge from experiments, we can work in very, very high dimensional spaces, we can do a lot of things with experiments that you can't do, for example, with design of methods. But we've also been able to work with collaborators to do the design of novel nanofibers, novel wet spin fibers. We've designed alloys with metallurgists, biomaterials with our scientists, and also come up with very, very interesting and novel processing which are efficient. So that kind of brings me to the end of this very short talk, and thank you very much for listening.
Well, please now welcome live with us Professor Svetha Venkatesh. Congratulations and welcome, Svetha.
Thank you very much, and it's an honor to be here today. Thank you.
Now, you describe the desert of uncertainty and the pitfalls of trying to measure with a large degree of uncertainty. Are there any benefits for pushing further past that bit of madness? And is AI getting better at being able to do so?
Well, yes, I think so. And the reason is, as I kind of alluded to in my talk, it's because you can formally store the regions that you have explored with the high uncertainty and record whether it's good or bad. Whereas if we were randomly trying experiments, it would be very difficult to put this in a cohesive framework. So as an example, like some of the experiments we've done for the design of novel nanofibers, the experimenter who's an expert in the field would takes about something like 70 or 80 experiments. But with AI, we can do this in about 10 experiments. So that's a huge difference in what you can do.
That really is just so phenomenal to use AI and machine learning to help to direct where future experiments should be. How new is this idea of using machine learning in this way?
So the interesting thing is the ideal Bayesian optimization was invented maybe 20 years ago, but in typical computer science style, we just kind of wrote it up and we never kind of used it anywhere. And to be honest, even I didn't know where exactly you could use it because for a long time, I've been working with big data like everybody. And when I met with these materials people, I would just keep asking them for data and they'd say they don't have any data. till finally it twigged that we can actually, accelerate the scientific method, and that there's a whole stuff, and so much to do in that space. I mean, almost everything is an experiment, right? Like, for example, if you look at an alloy, and this is one of my favorite examples, is that alloys are in such a small band. If you look at any alloy, and they never move past it, and that's why I call it like, Bronze Age. It takes one age to make a alloy, but we've invented this thing called just chuck it in, where you can chuck in any recycled material, and the strength can stay in a band. So that's the broadest alloy that's been invented so far.
Can this technique be applied outside of materials and processing? How transdisciplinary is it?
It's any experiment. You can do any experiment. You can grow a plant and you can say, what is the best combination of fertilizer and light and whatever that I can grow the plant? It can be used for anything. Anything where you're following the scientific method or the experimental method, you can accelerate.
It really is incredibly exciting. I mean, do you expect to see the rate of innovation move ahead in leaps and bounds in various disciplines that use this technique? And how have you seen that already?
So we've already seen it in material science. We're working, for example, with Ford, and they want to incorporate more cellulose into their products. So, this can be used to accelerate that. Working with some science in material design, it's really phenomenally useful because they can invent all kinds of new materials which can then scaffold other applications. So, as example, bioresponsive materials is something we're working on. So, I think we're working in several fields, but anything which is a mixture, right? So, anything from a polymer to a shampoo to an alloy is a mixture. If you want to figure out what's the best set of ingredients, you can use the method. Same thing with the process. If there's anything to optimize, you can use this process.
What kind of social implications are there for your work? You did touch on suicide prevention and the difficulties around recognising suicide prevention, but are there practical applications that you can see?
For the lean data, I see the practical applications almost in anybody who makes a product or process. And the challenge, of course, is that the machine will suggest the next experiment. So you have to guarantee that the next experiment is safe. So it's important for the human to specify the safe bounds because it can specify things that can be unsafe. So that's another aspect one has to be careful about. But, I see this is only the first step in what I see as a human-machine partnership. And I think the future will be us working closely with machines as our own partners for innovation.
Yeah, that's just a fascinating idea to even think about and to see where that might lead. So can you perhaps give us an insight into some of the other directions that machine learning itself could be heading and how that partnership with machines and humans could evolve? And what most excites you about the possibilities of the next 5 years or so?
Yeah, I think the next thing in machine learning is to do two things. I think one is to make this, it's not the data, right now it's a data over the fence paradigm. We throw data, machine does something, it gives you the result back, but that has to go into this kind of human machine collaborative framework where we can augment humans to do things and increase their creativity. So in the experiments we do with humans, there's a very strange cycle that I observed that initially when they're inventing something new, they won't know anything about it. But after the machine gives them like 3 or 4 experiments or maybe 10 experiments, the humans will then pick a point which is much better than the machine. So obviously the combination is what we need. And of course, there's no formal framework to do it, but we need to invent methods for this. And the other thing is these machines, we start from absolute zero in this. It's kind of astonishing it even works. There's a whole handbook of materials, knowledge out there. So we have to know how to put all that knowledge in. And lastly, causal reasoning. A lot of what we do is to understand why things happen and learn from that, learn the cause. These things haven't learned the cause. So and the field is moving more into learning causal reasoning as well.
Well, Svetha, it is an absolutely fascinating area. Thank you so much for your presentation today and congratulations on your election to the Academy.
Thank you so much.
Now we're going to take a short break and we'll be back at 2:15 for session 4. welcome back. Now, don't forget, you can ask our fellows questions throughout the event by using the panel on the right of your screen. Now, back to the presentations. To introduce our next fellow, please welcome Academy Council member, Professor Louise Ryan.
Thank you. Next, we will hear from Professor Gary Froyland. Gary has made major contributions to the fields of dynamical systems, ergodic theory, and discrete optimization. He has reshaped scientific views on how to characterize complex fluid flow and has also successfully applied his research to physical oceanography, atmospheric science, and open pit mine optimization. Now we'll hear from Gary.
Hello, my name's Gary Froyland, and it's a great pleasure to present some snapshots of my research. First up, what is a dynamical system? It's a rule that transforms a domain X, like this square, to itself. In this example, we're expanding by a factor 2 in the horizontal direction and compressing by a factor 1/2 in the vertical direction, cutting through the middle and stacking the black on top of the white, returning to a square. Now, in dynamical systems, you are repeatedly applying a rule over and over again. And so applying this rule to now this square, we arrive here. And applying the rule to this square, we arrive here. And you see that the black and the white parts are quickly being mixed together. Now, in applications, this transformation is usually highly nonlinear. There is stretching, as we've just seen in the toy example, but also folding in the domain. And this makes analysis very difficult. There is a convenient way, however, to perform a global linearization of this nonlinear T. We take the indicator function, 1a, so this is an indicator on a subset A of X. It takes the value 1 on A and it takes the value 0 outside A. And we define a linear transfer operator L as composition of these functions with the inverse transformation. That is, L acting on an indicator A is indicator A composed with T inverse, which is simply indicator of TA. So in summary, one could apply the nonlinear T to a set A, or through this set function correspondence, one could apply the linear L to the indicator A. So what you can do with this transfer operator, or one of the things you can do, is long-term simulation of floating plastic on the ocean surface. In the movie at the top right, you see lots of small white dots moving around. Each one of these dots is a drifter on the ocean surface. And it has a GPS that is telling its location. And we can collect up all this data and make an approximation of the transfer operator representing evolution on the ocean surface for 1 year. And now moving to the middle panel, what we did is we initialized a pulse of synthetic plastic rubbish at the coastlines of the world. And then we applied the transfer operator many, many times to propagate this plastic out into the ocean. So the movie here runs for 25 years, but you can use this technique to rapidly propagate for hundreds or thousands of years. You see an accumulation of plastic in these red regions, and these are known convergence regions for plastic. There's one in each of the major oceans. And the North Pacific prediction from our simulation was validated 2 years later using plankton net tows. So nets were towed across the North Pacific, and the plastic concentration was verified directly by observation from these nets. Now you can ask, where does the plastic in each garbage patch come from? And the eigenvectors of the transfer operator L will show us the basins of attraction of each of these 5 garbage patches. On the left, you see the 5 leading eigenvectors. The red is the support of the vector. So if you would drop a piece of plastic anywhere in the red, it would get sucked into the South Pacific patch. If you drop any piece of plastic on the red here, it gets sucked into the South Atlantic patch, and so on. And if you sum up all these red pieces, then you form almost a complete partition of the surface ocean. And so what we've effectively done here is redraw the boundaries of the 5 major oceans based on dynamical considerations of the surface currents. And this is more meaningful than, for example, geopolitical boundaries that would separate the Indian from the South Pacific by a line of longitude from Tasmania to Antarctica. Most real-world dynamics have time-varying governing rules. And to model this evolution, we compose transfer operators that change in time. So not a single transfer operator, but now we compose changing transfer operators. It no longer makes sense to compute an eigenvector of a single operator. So we use a theorem of Osiledet's that generalizes eigenvectors of a fixed matrix to time-dependent compositions of matrices, such as those we have here. And we augment this theory for transfer operators so that we can spectrally characterize slow mixing structures in the time-dependent dynamics and track their motion. And the first application we made of this was to the ozone hole. So in blue, you see a lack of ozone. And the reason for the lack of ozone is the polar vortex. The polar vortex is a known slow mixing object over the South Pole. And so using these transfer operator methods, we searched for the slowest mixing structure in the stratosphere over the pole. And that is shown in red in this animation here. And the other colors are air parcels outside the vortex. So there's very little communication, very little mixing between the red and the other colors, as you'll see in the movie. And this provided a much more accurate characterization of the polar vortex than existing methods. Now let me switch gears quite a bit to optimization, which is another research direction of mine. What is an integer program? Let's consider 2 binary variables, x and y. So x is 1 if something happens, and x is 0 if something doesn't happen, and y is 1 if something else happens, and 0 otherwise. You can link these 2 variables by linear inequalities. So y less than equal to x logically means that y can't happen without x also happening. And x plus y is less than equal to 1 means that both x and y can't happen. So with these linear inequalities, you can exclude different combinations of the values of x and y. And in optimization, you typically want to minimize some linear objective subject to a list of linear constraints that encapsulate physical limitations. So one thing I worked on for quite some time was long-term open pit mine planning. And the inputs are multiple stochastic ore body instances. You don't know exactly what is under the ground. And we created a multi-decadal integer program to maximize project value while respecting various constraints. So you're digging a large hole in the ground. The walls of that hole cannot be too steep. You have mining and processing capacity limits. When you do dig up material, you have to decide, am I going to process it? Am I going to throw it away? Am I going to put it on a stockpile? And you have to blend material from different pits to meet narrow chemical quality ranges. So this was packaged up, this mathematics was packaged up in software and sent out to international base metal mines and diamond mines to assist with their long-term strategic plans. Reducing flight delays. Bad weather and mechanical faults are examples of primary flight delays. So primary delays can propagate through the flight network. For example, if there is a crew changeover, the aircraft that is awaiting crew on the first aircraft, if that first aircraft is late, that will delay the second aircraft. So we want to reduce this, and given a schedule of routes to be flown and historical likelihoods of delays for each flight at each airport, we developed an integer program to simultaneously optimize the allocation of aircraft to routes, the allocation of crew to aircraft, and we made small schedule retimings where that was beneficial. And we were able to reduce an average delay of 1,600 minutes, that's for 50 aircraft over a day, down to 1,200 minutes. So this is doing all the same actions, but just putting the pieces together in a better way so that everything is more robust. And finally, optimizing lung cancer radiotherapy. So cancer of the lung and heart is challenging to treat with radiotherapy because you can't immobilize the tumor. The tumor here in red is rising and falling with the diaphragm of the patient. And sophisticated machines can provide doses 360 degrees around the tumor, as you see in this animation. And the treatment time per patient is limited, and you need an even spread of angles of irradiation to the tumor. Now, the machine motion and the radiation timing can be optimized. That's under our control. So we made an integer program to do this optimization, and we could improve significantly on the angular spread of the radiation. So this is a standard uniform motion of the machine around the patient. With the optimization, you could spread this out very nicely into a nice even dosage of radiation, which is much more beneficial to the patient. So thank you for the opportunity.
And Professor Gary Froyland joins us live. Congratulations, Gary, and thanks very much for your presentation.
Thank you. It's a great honour.
And such incredible applications of your mathematics in such a wide variety of fields. To begin with, I just wanted to ask you about the floating plastic example that showed a global scale simulation. Can the same technique be used for micro scale simulations?
In principle, yes. There's nothing about you have to balance the kind of time scales with the spatial scales, but in principle, there's nothing. It's a generic technique that could be used for microscales, yeah.
Yeah, right. And the flight delay example takes into account historical patterns to identify opportunities for optimization. Can the same integer program be used in a real-time or near to real-time responsive way?
Well, that was the original, maybe not the end goal, but that was the next step. And my PhD student who was working on this, he's done further work on this. So this would be, it's called the recovery problem. So you try to strategically plan as well as you can. That was what I illustrated. And then when something happens, you need to recover and get back on track as quickly as you can. And so he has done more work on that.
Yeah, I should have picked your brain before our organising our program today to adjust times when things go a little off track, but very fascinating work. Now, I did also have another question that's been sent in about what is the maximum predictive temporal that can be used that will still deliver a useful simulation?
Sorry, the maximum predictable temporal?
Yeah, it's the maximum predictive temporal that can be used that will still deliver a useful simulation. I'm afraid I don't fully understand that question, but perhaps you do.
Not exactly. Let me try and guess. So maybe it's for the first part, talking about the distribution of plastic.
I think so. Yeah.
So if in that situation, you had a kind of the garbage patches act as attractors, and so that's an extremely stable system over very long periods of time. You know that plastic is going to get sucked into these regions of convergence, and the main questions are what is the concentration and what is the source. But there are many other types of dynamical systems that are much more chaotic, much more complex, and there the predictability is much lower. And what you tend to do is not try and predict individual paths, but you try to predict ensembles of paths, and you look at the statistics of the ensembles of paths, and that is a much more stable thing to look at for systems that are less predictable along individual paths.
All right, terrific. Now, Professor Frances Saparovich has been watching on throughout the day, and she's sent in another question. She says, lovely work. I also like the flight delay example, and it's made me think of triaging patients in hospitals, especially in pandemics. Is this a possible application?
Yeah, I suspect that some kind of optimization routine is already being used there. I know some also Australian researchers that have worked on optimizing hospital triage routines. So I think in the background there is some mathematical optimization at work there.
Now, can you tell us perhaps what your most ambitious future simulation might be and why it's ambitious?
Well, I think I think that what some of the earlier talks today talking about data, I think this really opens up with machine learning techniques, how you can connect these with dynamical systems. I think this opens up the possibility to handle really high dimensional dynamical systems that are maybe particularly not well observed. And in some recent work, we've been able to understand better the ENSO, the El Nino Southern Oscillation cycle that Wenju talked about earlier. And to do that just from observations by satellites of the sea surface temperature of the ocean, for example. So this is a really high dimensional space you're talking about. And the observations were not so large, maybe 600 observations, but we could get a really good description of the cycle from that small number of observations. So I think the future is going to be more what can you really squeeze out of small numbers of observations, whereas classical dynamical systems would say you really need to pay a high price in terms of observations to get a good prediction.
And just finally, I'd love for you to reflect for us on, I guess, the collaboration that you've had with such a wide variety of disciplines. You've touched on cancer treatment to mining, to tracking plastic through the ocean, and then flight delays. How important is that collaboration and I guess openness to share the sort of work that you're doing with other disciplines?
I think it's incredibly important. I mean, I get a kick out of not only doing the mathematics, which is a lot of fun, but also seeing it at work. It's very interesting for me to learn about all these other disciplines as well. It's great to talk to these experts from other areas and learn about what they're doing. It's very impressive. And if I can contribute in some small way to help out with one of their problems, then that's great.
Well, it's very impressive work. Gary, thank you very much and congratulations again.
Thank you very much.
Now, please welcome back Professor Louise Ryan to introduce our next fellow.
Thank you. Next, we will hear from Professor Aurore Delaigle. Aurore is internationally known for her innovative contributions to mathematical statistics, where she has made transformative contributions in several statistical subfields, including functional data analysis and measurement error. Now to Aurore's presentation.
It is a pleasure to introduce you to one of my research topics, which is non-parametric curve estimation from data measured with errors. In many situations, we are interested in a variable x, but we can only collect data from a noisy version W of that variable x. For example, this happens if we are interested in the long-term systolic blood pressure or in the long-term intake of a nutrient, because often we can only observe the blood pressure or the intake at a particular day. And a measurement taken at a particular day is only a rough approximation of the long-term quantity in which we are interested. After a transformation that we're going to ignore here, it is often reasonable to assume that the noisy W can be expressed as x plus epsilon, where epsilon is called measurement error and is independent of x. Now, in statistics, we are interested in estimating quantities from data, and one of them is the density fx of a random variable x, because it is an important tool for describing various features of a population. But the problem when we have measurement errors is that the density of the noisy W is not equal to the density of X. And you can see this on this picture where in black, I show you an example of density of a random variable X. And in red, I show you the density of a noisy version W of that variable X. And you can see that the red density is a smoothed version of the black density, and that is because W is more variable than X, so its density is more widespread than the density of X. A similar phenomenon happens in the regression setting, where if we have a regression curve M that relates a variable Y to a variable X, that is Y is equal to M of X plus a random fluctuation delta, then estimating the regression curve M is reasonably easy if we manage to observe data xi, yi coming from that regression model. So for example, on the picture, I'm showing you the regression curve M in black, and I'm showing you stars, which corresponds to pairs of xi, yi, and you can see that those black stars fluctuate around the regression curve M that we would like to estimate. However, if instead of the xi, yi's, I can only observe noisy versions, that is, I can only observe wi, yi's, where WI is a contaminated version, a noisy version of the XI, then the data that I'm going to observe are the red stars that you see there, and these no longer fluctuate around the black curve, but they fluctuate around another curve, which is the red curve shown on this picture. So again, this is due to the fact that W is more variable than X, so the regression curve that connects W to Y is more flat than the regression curve that connects X to Y, in which I'm interested. So these two examples illustrate the impact of measurement errors. And in particular, we have seen that the density and the regression involving noisy data are only rough approximations to the density and the regression that we would like to estimate, which involve the non-noisy variable X. So therefore, if we want to estimate a density and a regression from noisy data, then we're going to need to make corrections for measurement errors, because otherwise the estimators that we are going to obtain are going to be biased estimators. So there are many techniques for doing this, and many rely on parametric assumptions. So for example, if we want to estimate the density of a random variable X using a parametric method, what we're going to do is that we're going to assume that this density belongs to a certain family of densities, of which only a few parameters are unknown and need to be estimated from the data. For example, we could assume that Fx is a Gaussian density, and then only the mean and the variance of that density are unknown and need to be estimated from the data. This works very well in many cases, but sometimes we're going to make parametric assumptions which are too unrealistic, too far from the truth, and then the parametric estimator is not going to be very good. For example, in this picture, I show you a bimodal density in black. If I assume instead that that density is normal, then the best approximation I can get to the black density is the red curve that you see there, which is very far from the black curve. This is because I made parametric assumption that the density was Gaussian, which was too far from the reality. Therefore, my parametric estimator there is going to be very bad under this wrong parametric assumption. So this shows the limitation of parametric methods, and this explains why there is a lot of interest in more flexible methods, which I call non-parametric estimators and non-parametric methods. These are the methods on which I have focused a lot in my research, not only in the measurement error area, but in other topics of statistics where I've done some research. So those methods do not make parametric assumptions on the curves we are trying to estimate, and instead they try to exploit the data as much as possible to construct the estimators. For example, in the measurement error setting, a non-parametric method to estimate the density and the regression curve from noisy data is often based on Fourier inversion. So for example, if we want to estimate the density of X from data on W, where W is X plus epsilon, and X and epsilon are independent, then the technique consists in computing the Fourier transform of the densities of those three variables and expressing them in a relationship. So we have phi W is equal to phi X times phi epsilon, where phi is the Fourier transform of the density. Now, since we observe data on W, we can estimate phi W from the data we have, and the literature often assumes that phi epsilon is known, so that we can deduce an estimator of phi X in that way. And then from the estimator of phi X, we can deduce an estimator of the density of X by doing regularized Fourier inversion. And a similar technique, but more sophisticated, is applied to estimate a regression curve from noisy data. My contributions to this area have been to develop new non-parametric methods that make less assumptions and are more widely applicable than the existing ones. So for example, I just mentioned that the literature often assumes that phi epsilon is known. Well, one of the contributions I've made with my co-authors is to develop a non-parametric method that does not assume that phi epsilon is known and instead estimates phi epsilon from the data. We have also developed methods that do not assume that the errors are identically distributed for various individuals and instead can have a different distribution. And we have developed various other methods which are more flexible or more general than the existing ones. One of the focus of my research in this area and in general is that I I am very interested in developing data-driven methods. So I want the methods to be fully applicable to data. And that's why I have developed quite a few data-driven versions of methods that existed already in the literature, but were not fully applicable to data. And I have found ways to make them completely data-driven. Finally, to conclude this talk, I would like to give an example of recent work that I've done in the area, and which originates from nutrition. So in nutrition, we are often interested in long-term intake of various types of food, nutrients, and drinks. And some of them, such as alcohol, fruits, or vegetables, are not consumed every day by every single individual. So depending on whether we collect the data on a day where an individual has consumed the food or not, the observations are going to be of very different nature. So if the data are collected on a consumption day, then the measured intake of the individual is going to be of the form that we discussed earlier. That is, W is going to be equal to the usual intake plus a random fluctuation epsilon around that usual intake. However, if we collect the data on a day where an individual did not consume the food, then we're going to observe zero because the individual did not consume the food. So for this type of foods, which are only consumed episodically, we're going to have quite a few observations which are going to be equal to zero because they are going to be collected on a day where individuals did not consume the food. And this is why this is referred to as excess zeros. Now, we cannot just work with the non-zero data because the long-term intake needs to involve both types of data. Both the data from a consumption day and from a non-consumption day are informative about the long-term intake of the food. But unfortunately, the classical measurement error methods cannot deal with the zeros in those data, so we need new methods. There are already parametric methods in the literature for dealing with the zeros, and my contribution is to develop non-parametric techniques that can deal with those zeros. So this concludes my introduction to this measurement error problem, and I thank you very much for your attention, and let me give you a few references of my work in the area.
And please now welcome with us live, Professor Aurore Delaigle. Congratulations and thank you for your presentation. How did I go with my pronunciation?
Excellent, excellent. Much better than your French is much better than my English.
Thank you. Now, I might actually start with a question that I've just received popped up on my iPad here from Professor Nalini Joshi. Thank you for tuning in. She says, interesting that taking Fourier transformation enables improved estimations. Are there limitations to this method? And when does a Fourier transformation not work?
That's an excellent question. Yes, so Fourier transform make the calculations easier because they turn convolution products, that's very complicated integral forms, into a simple product of functions, the Fourier transforms. So that's why they are very widely used in this area. We know better how to deal with products. However, that's an excellent question because at some point we have to divide by a Fourier transform, and we can only do that if that Fourier transform is non-zero everywhere. And that is the limitation. If the Fourier transform is non-zero everywhere, you're not going to be able to do that. Fortunately, in most commonly encountered data, it is non-zero, but that's an excellent question. So we are working on developing alternative methods that do not require to do that.
And just towards the end of your presentation, you mentioned the use of your work in tracking nutrition, for example. Perhaps you could tell us a little bit more about that or other applications.
Yeah, in the measurement area, the main application really is in nutrition. That's where you have, because you have a lot of measurement errors. Of course, there are measurement errors in many different other fields. Most data are collected with a lot of noise. But in nutrition, you approximate a long-term intake by a daily intake, and that's really measured with a lot of errors. So you really have to do something about the measurement errors. So this is a huge research area to try to remove the measurement error in nutrition.
I've got another question that's come in from one of our fellows. David Balding says, congratulations, Uho. Can you give us some intuition about how you infer X and epsilon separately? This seems impossible in general without prior knowledge.
That's a very good question. In general, it is impossible. Indeed, it's impossible to disentangle the two distributions. However, we have done some work recently where we actually managed to do that without any replicated data. So what you do is that you indeed make some assumptions. You assume that the areas that are symmetric, that's often the case in read processes, and you assume that the distribution that you're trying to estimate is heavily non-symmetric. And the nature of the two distributions is so different that in that case, you are able to actually distinguish to distinguish the two distributions. And it works very well in practice, to my biggest surprise, because I've never expected that it would be so easy in practice, but it does work in practice.
Isn't that some of the most joyful part of research, is those unexpected results and that lead you in a new direction?
Absolutely, absolutely, absolutely. You start with a project, it looks messy, it looks complicated, and you manage to make it work, and all of a sudden, not only it works, but it works much better than you had expected. Absolutely. I've had a few projects like that. It was extremely joyful.
Tell me how you found this to be your area of passion for your research.
Originally, I started working on that a long time ago when I was still a master's student. And at that time, I was really interested in the combination of mathematics, programming, and new methods that you could develop. But over the years, I've come more and more interested into the data themselves, the richness of problems that you can find in nutrition. There's a huge variety of very, very captivating problems that can be solved. And I find that absolutely exciting. And I love making methods more practical, less restrictive, and that's really an area for that.
Yeah, wonderful. And just finally, we'd love to attract more women to STEM and to remain in STEM as a career. What advice would you give a young woman with an interest in STEM?
First, I would say don't be afraid. If you are attracted to mathematics, to science in general, go for it. It's not, it might appear scary, but once you do it, it's not as scary as it appears. There's more and more women there. You will find some peers. You will love it. And you have there is a space for women in STEM. We have something to bring that is really, really valuable. I absolutely encourage all women to go to STEM if they are attracted to that.
Thank you. You're a huge inspiration, Uho. Congratulations once again.
Thank you.
To introduce the next presentation, please welcome back Professor Louise Ryan.
Thank you. Our next presentation is from Professor Yihong Du. Yihong is internationally renowned for his contributions to the theory of nonlinear elliptical and parabolic partial differential equations and their application in chemical reaction theory and population dynamics. He is a leader in the use of nonlinear free boundary problems to model spreading phenomena and has resolved long-standing problems on sharp thresholds, boundary blow-up, bifurcation, and multiplicity. Please welcome Yihong.
It's a great honor to have this opportunity to give a short presentation for my research on propagation and reaction diffusion equations. Reaction diffusion equations are models that are used commonly in chemical reaction theory, but they also are used in non-chemical processes such as ecological invasion, tumor growth, and wound healing, and so on. The first application of such models in propagation problems were by works of Fisher and KPP. Both appeared in 1937 independently, and both are motivated by the understanding of spreading of a special gene in a population. And the Fisher model looks like that, where U represent the density of the population that carry the special gene. And there are two parameters, D, diffusion rate, and A, growth rate. Both are positive constant. The KPP model is almost identical to the one used by Fisher. They use a more general reaction function, FU, but it behaves similarly to Fisher's nonlinearity. There are two important discoveries in these works. One is about a special kind of solution called the traveling wave solutions, solutions of that form. So where C is called the wave speed of this traveling wave. So it behaves like that. So for fixed t, it's a curve like that. And when t changes, it moves like a wave. They proved that for c greater than or equal to c0, given by the two parameters in this way, you have such a solution, and no such a solution for c less than c0. The other important finding is that, so they believe that c0 is the spreading speed of the gene in the environment. So Fisher just speculated that, and KPP actually have a theorem showing that if you choose the initial function that way, then the solution of the equation would converge to the traveling wave solution with a minimal speed c0. The first evidence supporting this theory appeared in a work by Scanlon in 1951. Scanlon used this map published in 1930 on the spreading of muscat in Europe to show that the range radius of muscat increased with the time linearly, like that. So this map, H closed curve, represents the range boundary of the animal at a particular year. So Scanlon just measured the area, range area, and found this linear growth. And subsequently, many other evidence supporting the theory appeared by using the same method. Another important work appeared in 1978 by Alison and Weinberg, two University of Minnesota mathematicians. They consider an equation including this particular case by Fisher, but they consider the case in a general space, n-dimensional space. They proved that if the initial population is concentrated in a bounded region of the space, then as time t goes to infinity, the population density converts to 1 in a ball of a radius roughly c0 times t. And out of such a ball, the population density is close to 0. And c0 is the 2 square root AD discovered by Fisher. To say this represents the fact that spreading has a speed c0, and one can use this set to represent the population range. So where the population density is greater than b, that region changes with t. And then you can show by this result the range radius grows to infinity at exactly this rate c0. But there's a shortcoming in this model. That is, the natural population range is not represented by this one, not the population greater than b positive, but a population density greater than 0. This region changing with t. However, the theory tells us that once t is positive, this population range is the entire space all the time. Although at time equals 0, it is a bounded set. So we are in a dilemma. To get the spreading speed, we need to use a b positive. But to get to the natural population range, we want b equals 0. So if we use b positive, then the range boundary would depend on this parameter. But the range boundary is an important notion because very often that is the spreading front, which is very important. Therefore, it's desirable to determine it more accurately. About 10 years ago, in joint work with Professor Zhigui Lin from China, we proposed a modified model of the Fisher one. So we consider the equation, the semi-equation, only over the population range, which is a bounded interval changing with time. And the population outside of the range is 0, of course, and that boundary is 0. But we need to introduce 2 more equations to describe the evolution of the range boundary. So the new model is more difficult to treat in mathematics. But fortunately, we are able to completely determine its dynamical behavior. As time goes to infinity, we prove that either the population vanishes or the population spreads successfully. Moreover, we can also determine a spreading speed by using the associated wave equation. And the relationship between the two, the new model and the older one, is the following. So as the parameter mu goes to infinity, the solution of the new model converts to the solution of the old model, provided they are initially the same. The solution, initial function are the same. The parameter mu appears here. So the meaning of the parameter mu is that 1 over mu is proportional to the sacrifice of the population in order to expand its range. And the theory has been further extended and generalized. Thank you.
Now we're delighted to have with us Professor Yihong Du. Congratulations, Yihong, and thank you for your presentation.
Thank you.
We've got some questions here for you. You referred to the work of Fisher and other mathematicians whose work helped you to reach your solutions. How important was that earlier work?
I think, so these are pioneering works. Before their works, people didn't think about using these kind of equations to model the propagation phenomena. And it took some time for the community to accept this is a good model. That's why I mentioned this work by Scanlon in 1951. So their model predicted a spreading speed in 1937. And this evidence was shown to us so more than 10 years later. So after this 1951 work, people start to recognize that's a useful model. And of course, nowadays it's widely used.
Yeah, it's a beautiful example of how the work of earlier researchers and mathematicians can be built upon. You referenced some of the applications of your work. I wonder if you could describe how it's used. How can it explain ecological invasions, for example?
Of course, ecological invasion is a very complicated matter. And we, to model it in mathematics, we have to simplify it a lot. And in order to obtain a general enough kind of a conclusion. And we have tried to use these with some people in Canada, modeling the spreading for mosquitoes, and mosquito, carry disease and so on. But still, that's on the theoretical side. And I believe to make it useful in a particular spreading phenomenon, so you have to extend the model to include more factors into it, and then use a lot of numerical simulation. So we mainly work on the theoretical side. Yeah.
Okay, yeah, it's fascinating. Now, Professor Nalini Joshi has chimed in again this time with a question for you. She says, congratulations, Yihong. Is there blow up in the travelling wave solutions?
Not so far, but of course, travelling wave is a phenomenon occurring in lots of models. And in the majority of the models, you use nonlinearities in a way that blow up does not occur. But definitely it's possible in certain for certain phenomena, then you have to use different kind of nonlinearities that would make it possible for blow up to occur. It's possible, but I've not worked on that so far.
Okay. Now, I'd like to return to something else that you referenced, your work use in explaining tumour growth or wound healing. Could you tell us a bit more about that, how it's used?
Yes, this is mainly for this moving boundary kind of thing. We in mathematics call it a free boundary. And in these kind of problems, so the area of concern is changing all the time. So therefore, you have to introduce a moving boundary into the model. And I myself has not really worked on this wound healing problem myself, but I know colleagues in other universities like a QUT group, they worked on problems with the moving boundary, like a tumor model, wound healing, and so on.
Yeah, fascinating. Now, would the use of your model in the spread of epidemics also applied for something as global as the COVID-19 pandemic?
Not in my kind of model, and we have to rely on a certain, the spreading individuals move in space more or less according to a rule you can describe in mathematics. But for COVID, there are lots of controls, and there are other models that work better for these kind of, lots of intervention, human intervention into the process. But the models I worked so far are more kind of a natural kind of a process. And if there are lots of interventions, then the model becomes less powerful. It still can be used, have some use, but less powerful.
Yes, yeah, I can understand the complexity grows when you have all sorts of other changes that can occur. Where would you like to take your work next?
Sorry?
Where would you like to take your work next?
I really want to understand, as I mentioned, so the individual behavior moving around in space change. There are random walk kind of dispersal, and there are also kind of long distance dispersal. That's something I'm working on now. I really want to better understand this kind of non-local dispersal effect on the spreading. For example, back in the Fisher KPP model already, they showed there's a spreading speed. But for this process involving long distance dispersal, and there's a kind of so-called accelerated spreading. So you spread faster and faster. That is something I'm very interested in, and I've been working on recently. I want to continue in that direction.
Well, Yihong, congratulations once again, and thank you very much for your time today.
Thank you.
Now, please welcome back Professor Louise Ryan to introduce our next fellow.
Thank you. Our next new fellow presentation is from Professor Rob Hyndman. Rob is one of the world's most recognised applied statisticians and is internationally acknowledged for his research in time series forecasting. Although time series data are widely used in many fields of science and business, forecasting is notoriously challenging. Rob's textbooks and software have transformed the capability of many organisations to make accurate predictions. We'll now hear from Rob.
Hi, I'm Rob Hyndman. I'm a professor of statistics in the Monash Business School. Most of my research concerns time series analysis, especially forecasting and analysing large collections of time series. I'm particularly interested in uncertainty and understanding, describing, and evaluating the uncertainty about the future. When I tell people I do forecasting, they almost always assume I do one of two things, either that I forecast the weather or that I forecast the stock market, but I do neither of those things. The best way to understand what I do is to provide some examples of how my work is used. An early project I did was forecasting the total cost of the pharmaceutical benefit scheme for budget purposes for the federal government. When Australians go to the pharmacy to buy pharmaceutical products, they sometimes get subsidised by the government, and so the government needs to forecast the number of sales of all those different types of products in order to plan for their future expenditure. And I helped develop the tools that they now use to do that. I've also worked for each of the state governments to help with forecasting peak electricity demand, the maximum demand that's going to be needed in several years' time. And they use that to inform their planning around generation capacity. Pretty well every retail company in the world now uses forecasting, and many of them use my algorithms to help forecast retail sales, and that helps them determine stock ordering policies. An interesting project I've been involved with in the last couple of years is forecasting tourist numbers in Australia, particularly the tourism recovery post-COVID. It's obviously a challenging problem because the data from the past is not going to be very similar to the data in the future. I've also been working on forecasting COVID-19 cases to help each of the state governments and the federal government form appropriate policy responses. And since the start of March 2020, we have provided weekly forecasts that are used to determine what sort of lockdowns or freedoms need to be introduced in order to help control the spread of COVID. The beauty of mathematics is that it's very general, and that the same methods that I developed for the PBS are now used by Coles in their retail forecasting. The same methods I developed for forecasting peak electricity demand are now being used for forecasting groundwater levels in New South Wales. When I began my career, forecasting was considered something of a backwater, and I was strongly advised not to work in this space. It was hard to get published, and many researchers considered it a somewhat trivial application of limited interest. But while I was doing my PhD, I was also working as a statistical consultant, and I could see there were a lot of unsolved problems, and people in business and industry were using substandard methods that could easily be improved. So not being one to follow the crowd, I decided it would actually make a good area to focus my research attention. And I think I've been proved correct over time. As more and more data has been collected, the need to have automated forecasting methods has grown. And so I've done quite a bit of work on developing automated approaches to forecasting, which are now very widely used in business and industry. A spin-off from my research papers has been the software I write in the form of R packages. Each time I develop a new forecasting method or a tool, I provide an open source implementation that can easily be used. That's made a tremendous difference to both the number of citations of my work and the impact of my work. I've produced about 50 R packages, and they're now downloaded by several million users every year. Some of those packages are shown there. Forecasting is largely a problem of trying to quantify your uncertainty. Of course, no one knows what the future holds, but we can estimate what are the most likely futures and what are very unlikely events. It's a matter of building probability distributions that describe the uncertainty of future events. Let me give you an example. Suppose we were interested in forecasting the number of short-term visitors to Australia. Here we have some monthly data since the start of 2000 to the end of 2017, and we're trying to forecast the next few years. You can see there's some quite significant seasonality because people come to Australia more at some times of the year than others. There's a little bit of trend, particularly in the last decade, and there's also a fair bit of sort of irregularity and uncertainty in the data. If we fit a model to that series, we can use, for example, the ETS framework, which is one of the frameworks that I've helped develop. And then fitting a model to the training data, we then simulate the future from that model. So we get a possible sample path of what the future might look like in the coloured series. But we can do that again with the same model. We can run forward another time with a different set of random inputs, and we get a different possible future. And as we do this, we can build up an understanding of what the plausible futures might look like and what the unlikely events are. So we slowly build up an idea of what of what all of the different possible futures might be. And then we can summarize those using probabilities. So for example, we can find the mean future, which is the blue line. We can find the 50% most likely futures, which is the darker shaded region. If we zoom in, you'll see it more clearly. So the darker shaded region contains 50% of the possible futures, the light shaded region, 90% of the possible futures, and then some futures are outside the 90% region. So what we've done there is to try to look at the probability distributions of where the future might lie. Often the users of forecasts are less interested in the mean, the blue line, as they are in the extreme tails. For example, the various health departments around Australia are most interested in the upper tail of our COVID-19 forecast. What is the 99th percentile of possible outcomes, the 1 in 100 event? And so most of my work is about trying to understand and quantify and evaluate this future uncertainty. Of course, these particular forecasts were not very good after the first couple of years. So you can see that the black line, which is the true values that occurred during 2018 onwards, they tracked our forecasts extremely well up until around March 2020. And this is an example where the assumptions that we were making were not held throughout the forecast period, which leads me to say something about, well, what makes something easier to forecast? And I've come up with 4 factors, four components that you need to produce good forecasts. First, you need to have a good understanding of the factors that contribute to the thing you're trying to forecast and that you can measure those factors. Secondly, you need a lot of data available. Third, you want the future to be somehow similar to the past in a way that you can build into the model. And fourthly, you don't want your forecasts affecting the thing you're trying to forecast. So in these short-term visitor forecasts, 3 of those conditions are satisfied, but the future is not the same as the past, particularly since March 2020. So that was one component that was not satisfied in our forecast, so therefore not very good. If I was forecasting the stock price, the 4th one is a problem. The forecast can affect the thing you're trying to forecast. A good forecast of the stock price, if it's done by a well-known person and publicised, then the prices can change in response to that. And that's one of the things that makes the stock market a difficult thing to forecast. Forecasting has a very long history. Babylonian forecasters used the distribution of maggots in a rotting sheep's liver to predict the future, a little bit like reading tea leaves in a cup. And they trained budding forecasters at the Babylonian tertiary institutions, except they probably weren't tertiary, the Babylonian universities, by giving them clay models to show how to do that. So this is a clay model of a sheep's liver from the British Museum. And these squares, the sheep's liver is divided into these squares, and where the maggots were, which square the maggots were, was supposed to mean something about how the future would unfold. The fact that we even find these clay models shows how widespread forecasting training was. So this was about 600 BC, and around the same time, people wanting forecasts would also journey to Delphi in Greece to consult the oracle at the Temple of Apollo. And she would provide her predictions while intoxicated by ethylene vapours. So fortunately, we no longer have to get stoned on hallucination and get hallucinations through these vapours when we do our forecasting. Forecasters have had a bad name over time. The Emperor Constantius issued a decree in AD 357 forbidding anyone to consult a soothsayer, a mathematician, or a forecaster, saying, "May curiosity to foretell the future be silenced forever." A similar ban on forecasting occurred in England in 1736 when it became an offence to defraud by charging money for predictions. And the punishment, 3 months imprisonment with hard labour. Fortunately, forecasting is now legal and hopefully based on a more solid statistical foundation. A measure of the way forecasting has changed even recently is the impact factor of the main journal in the field, the International Journal of Forecasting. I was its editor-in-chief for 14 years up until the end of 2018. And during that time, the impact factor increased from 0.4 to 3.4. So forecasting has come a long way, both over the last 2,500 years, but also over the last 20 years. And I'm delighted to see forecasting now receiving greater recognition as an important scientific and research problem, and that I've been able to play a part in making that happen. Let me finish by just giving some ways people can contact me, and there's a URL if you would like to get a copy of these slides. Thank you.
And please now welcome with us live Professor Rob Hyndman. Congratulations, Rob, and thank you for your presentation.
Thank you. It's a pleasure to be here.
Now, I'm going to start with a question that's been sent in during your presentation from Professor Frances Aparovich. She says, nice work, Rob. Most of us will be familiar with outcomes of elections and the difficulty in predicting based on using appropriate sampling. Could you comment on how better data could be collected?
That's a good question, and it's not an area that I have worked directly in, election forecasting, but it has become a little more difficult over time, because collecting appropriate data and having truthful responses from the people you talk to seems to have become more difficult. There's some really good work on that problem in the US, where there's much greater collection of data through polling than there is in Australia, at least. And models have been used to adjust the polling biases. And so the forecasts obtained for relatively recent US elections tend to have been pretty good, much better than they are in Australia. If we were to do better in Australia, we would need to have a lot more data collection, and so that sufficient data so that we could actually estimate the biases in the polling that is done. At the moment, we just don't have enough data to do that.
And I mean, I had a similar question for you myself in terms of the things that can make forecasting difficult. How do you take into account social change in forecasts like the short-term travel? They're difficult things to track, aren't they? And can be unpredictable.
Yeah, so we're always making an assumption that our model captures the way things are moving, both in the past and into the future, and that we've captured, the dynamics appropriately. But if something happens in the future period that's not in your model, then the forecasts are clearly not going to work very well. There's something like, travel. As long as you've got a sort of a stable travel environment, you can do a pretty good job at forecasting travel. But pandemic has shown that your forecast can very quickly go badly wrong.
Yeah, but and then on the other hand, that graph that you showed, it was quite stable for many, many years and actually quite predictable over time.
Yeah, I've been working with the Tourism Research Australia for a couple of decades, and we have produced good forecasts for almost all of that time. It's only in the last year or so that they haven't worked so well.
Yeah, fair enough. Now, Malcolm Sandbridge has written in a question. Great talk, Rob. What would you say to Niels Bohr, who said prediction is very difficult, especially about the future?
Yes, that's one of the great all-time classic quotes in forecasting that often gets used in undergraduate classes on the difficulties of forecasting. Prediction, even about things that are historical but not observed, is also a difficult and interesting problem. We tend to call that prediction rather than forecasting, but it's an interesting problem. But once you talk about predicting the future, then there's a whole other level of uncertainty because of the fact that the environment can change in a way that you're not expecting.
You spoke about the journey that forecasting has been on and getting better recognised, and you rightly predicted it was a good future for you to pursue. How do you see the future of the discipline going now, and where's it going next?
Forecasting is booming in so many ways, and it's partly because of the increased collection of data in just about every field. And so there's lots more data, so there's lots more capacity to do forecasting. And it's partly around the increasing complexity of methods to enable us to use that data. So companies like Amazon, for example, which have a few 100 million products and sales history on all of them, are building really complicated deep learning models to forecast those products. And so the availability of fast computing and much larger data sets is fueling a real regeneration of forecasting methods at the moment.
Well, it's really exciting to learn a little bit about your work, Rob. Thank you so much and congratulations again.
Thank you.
Now we're going to take a short break before wrapping up the day with session 5, starting at 3:30. See you soon. Welcome back to our final session for today, and thank you for all your questions. Keep them coming in. You can use the Q&A panel on the side of your screen. Now to introduce our next fellow, please welcome back Professor Sue O'Reilly.
Thank you. Our next presentation is from Professor Andrew Pittman. Andy is an international authority on the role of land surface processes in the climate system, including their influence on regional rainfall and temperature extremes. As a direct consequence of his work, there is now an accepted role of land cover change in regional climate projections and a new field devoted to utilising land cover to mitigate the worst impacts of regional climate extremes. Please welcome Andy.
Good afternoon. I'm Andy Pittman, and I want to introduce you to some of the work I've been doing on terrestrial processes in the context of a changing climate. Back in the late 1960s, early 1970s, the established wisdom was that the land surface was a passive boundary condition for weather and climate prediction. Work, for instance, by Suki Manabe described the land surface in very simple terms. And unfortunately, that work turns out to have been inconsistent with our emerging understanding of the role of the land in how it constrains the exchange of energy, water, and carbon with the atmosphere, and inconsistent with the spatial heterogeneity the land imposes on the atmosphere through albedo, roughness length, and things like patterns of moisture holding capacity, and also inconsistent with the role that land use and land cover change, primarily through human activity, imposes on regional climate, and inconsistent with the role the land plays in intensifying droughts and heat waves. So as a consequence of all of that, there has been a revolution in how we represent terrestrial processes in our weather and climate models. And there's been a transformation to account explicitly for processes that influence energy, water, biogeochemical cycling, and the particular details of how we represent human modification to those systems in our particularly climate models. And work I've been doing over a number of years now has started with how we represent the critical processes at the levels of complexity that are possible in our global scale models, to include processes that are important like groundwater, snow, soil freezing, vegetation properties, soil properties, how we describe those things mathematically in our models in order to examine important questions like the impact of land surface processes and land cover change on our global climate, regional climates, impacts of land processes relative to increasing CO2, and most recently, the impact of land processes on extremes and extreme events. So here's an example of some early work I published with a PhD student of mine, Maggie Zhao. This looked at the impact of land use and land cover change on the 20-year return values for maximum temperatures, so quite extreme temperatures. And we contrasted that with the impact of increasing CO2. So the bottom panel here basically shows the increasing CO2, the impact of increasing CO2 on 20-year extreme temperatures, and the top panel shows the impact from land use and land cover change. And the critical thing here is, whilst the increasing CO2 tends to warm everywhere, the impact of land use and land cover change is highly regionalised. So we can see an amplification of CO2-induced warming due to land use change over North America, but a suppression of warming over Europe. And an important question was why? Why does land use and land cover change sometimes amplify and sometimes suppress? Which led us on over a long period of time to finally understand that, to first order, the regional impact of land use and land cover change is determined by the nature of the background climate. In other words, it matters a great deal how much snow's there, how much moisture is available, the nature of the background climate in terms of its expression in the form of extremes. And this picture that comes from our 2011 paper basically shows that you can get an amplification due to land use and land cover change in the cold climates in spring, but a suppression of the CO2-induced warming patterns in summer as a consequence of land use and land cover change. But the critical thing was the role of the land was predetermined, if you like, by the nature of the background climate. And that work evolved over time to begin to address to what degree and where can the land be used to moderate global warming at regional and global scales, all the way down to more local scales. So questions like, how does the future risk of heat waves and droughts influence the viability of reforestation? Or can you use brighter crops to manage heat waves? And the answers to those questions, it turns out, depends on the background climate, the frequency of change in extremes, and a whole variety of other things. There is not a simple answer to that question. It is highly regionalised. Here's an example of some work we published in Nature Geoscience more recently. Here we have perturbed the albedo to reflect more sunlight back out to space as a surrogate for brighter crops that might be grown. And on the left-hand column, you can see that if you increase the reflectivity of crops over Europe or over Asia, you do get a cooling, quite a significant cooling, maybe 3 degrees in extreme temperatures. In some regions where you perturb crop albedo, like in Europe, you don't see any real negative effects. But if you do it over Asia, you see a significant reduction in rainfall over the regions where you've increased the crop reflectivity. In other words, again, if you're going to try to moderate the impacts of global warming using land processes, you have to understand the detail of how that regional land surface influences the atmosphere. Inevitably, when we do this sort of work, we end up in a continuous cycle of model improvement. So, normally, when we undertake experiments and we delve deeply into the processes that help us understand our results, it feeds us back into the need to improve the representation of terrestrial physics, biology, and biogeochemical cycling in our models. But work that we have done more recently has added a major complication to that. Back in 2004, a paper was published in Science, which I, along with many others, were involved in, which introduced coupling strength, or omega. And this is to do with the covariance of, in this case, soil moisture with atmospheric processes, although it's been defined in many ways since. And work that was done back in 2004 identified hotspots of land-atmosphere coupling. And what we did is took that work and applied it in new and innovative ways to demonstrate 2 basic things. One was this coupling strength was strongly influenced by the land cover type, and that, therefore, land use and land cover change strongly influenced the coupling strength. And we did that using an innovative experimental design which let us identified for the first time that the coupling strength was strongly controlled by the formulation of the planetary boundary layer. And the picture on the right here shows the difference we identified in the impact of land use and land cover change on this coupling strength, and how that was largely determined by the way we represented the planetary boundary layer. This was work done with another PhD student of mine, Annette Hirsch. We then took that work to resolve one of the big conundrums in our field. What is the impact of Amazonian deforestation on climate? And we're able to show that while most models predicted warming as a consequence of Amazonian deforestation, but a really inconsistent result on rainfall, we could demonstrate that that was basically related to the strength of the land-atmosphere coupling in the models that we used. This land-atmosphere coupling is an emergent property. It's not something that's put into the models in the form of a parameterization. And we're able to show for the first time that small amounts of deforestation in strongly coupled regions would have a larger impact than large-scale deforestation in weakly coupled regions. This led us to the recognition that this coupling between the boundary layer and the land surface was perhaps the single way that we could begin to understand how the land and the atmosphere interacted in ways that helped us improve the predictability of our models. And all this really translates into that if you want to understand the patterns of emerging extremes and how the land can help us mitigate global warming, we first of all have to co-locate the role of the land and the role of the boundary layer in a coupled system. And then we have to understand how that coupling strength will respond to changes in the land and a warming atmosphere. Two enormous challenges to our science moving forward. And with that, and with thanks to my former students and postdocs and many collaborators who have contributed in my work over the years, I'm happy to take questions.
And Professor Andy Pitman joins us live. Welcome and congratulations, Andy, and thank you for your presentation.
Thank you, and happy to be here.
I'd love to start by asking you a quite current question, actually. I'm sure you've seen the news that world leaders have committed to ending deforestation by 2030, 100 or so of them in recent days. What sort of difference does that commitment make?
So deforestation releases a very great deal of carbon dioxide into the atmosphere, and it also has quite profound implications for the environments where that deforestation is undertaken. So it's a good news story. It's one of several good news stories today. What I haven't seen is the countries that haven't signed up to it. So I haven't actually got the list of the countries and whether some of the primary deforesting countries are all signatories, but it's clearly a good news story.
Yeah, fascinating. No, thank you, and very relevant in some of the descriptions you've been giving and the work that you've been doing. Now, can you tell us a little bit about how your work might help with adaptation strategies for climate change?
So the really biggest question in adaptation is where you've got a spare $1 billion, you've got to decide where to spend the spare $1 billion. And being able to take climate risk information and nuancing the spatial detail of that information so it informs risk is extraordinarily difficult. But what we have to be able to do is determine where that investment is the most effective and avoid investing that money where it would have been a waste of money. The sorts of things that we do help that granularity of how climate change will be imposed on landscapes. We certainly don't get down to the property level, but we do help understand how regional patterns of land use and land cover change or reforestation or the regional patterns that cities impose on the landscape, how that changes the surface of vulnerability to climate change. And hopefully that's very useful. But I don't think we've got there yet. I think there's decades of of some awesome climate modeling and other sciences ahead of us before we can really quite confidently use these sorts of sciences to advise policymakers on whether they should protect that catchment or this catchment.
Professor Helene Marsh has written in and says, "Hi Andy, fascinating talk. I'm very interested in environmental restoration. At what scale can your models provide advice?"
I'm a climate modeler, particularly interested in climate modeling. At the moment, the granularity of our global climate models is about 100 by 100 kilometers until somebody gives us an exascale computer. The ability to take that 100 by 100 kilometer pixel and downscale it using similar mathematical techniques that we use in our global models means that we can get to some regions for some purposes at very much higher resolution, down to maybe 1 kilometer. But even a 1 kilometer pixel is quite coarse when it comes to actual on-the-ground decision-making for adaptation and risk mitigation. Therefore, you need a very great deal of expert judgment at that interface between the people at risk and the people who provide information on future risk to make sure you don't make inappropriate decisions.
And just finally, what's been the response to your findings, especially given that different regions have different results?
Mostly not all that favorable because it complicates the story. It's much easier if people can give simple answers that policymakers can take on board. But as we all know, our science is highly nuanced. And when we get into the detail of our science, we realize that the solutions that we need to bring to the table are much more complex than the early work suggested. So it's a very challenging communication problem, and it confronts most people working in my field.
Well, Andy, congratulations on your work. Thank you very much for it and for spending some time with us today.
Thank you very much.
Now to introduce the next presentation, please welcome Academy Vice President, Professor Helene Marsh.
Our next new fellow presentation is from Professor Justin Marshall. Justin's research has made fundamental contributions to sensory neuroscience and ecology, including the discovery of the world's most complex colour vision system, the first demonstration of sensitivity to circularly polarised light in any animal, and discoveries defining modern visual ecology in both vertebrates and invertebrates. Now to Justin's presentation.
Good afternoon. My name's Justin and I have the pleasure this afternoon of talking to you about my research. I was elected as an interdisciplinary fellow of the Australian Academy of Sciences in 2020, and I'd like to thank my fellow fellows and the Australian Academy for giving me the great opportunity to convey really the wonder of nature. I'm very lucky in that I work on an array of superb animals with amazing colours and visual systems, and this is what I am. I'm a visual neuroscientist trying to understand how other animals see. So therefore I look at the eyes of animals, and here's an array of eyes. I could be called a comparative visual ecologist. I also am very interested in the colours of animals, and here you see the colours of a number of reef fish in close-up. So I try and interpret the colours of other animals, how they use these colours for communication and camouflage, but do so from the animal's perspective rather than the way that we see these colours. Probably what I'm most famous for, if you like, is the discovery of the mantis shrimp visual system, because they have the most complex vision of any animal known. Here you see a mantis shrimp narrowly missing a fish and showing its other famous attribute. They have these amazingly nasty spines and clubs with which they hunt. The eyes is what I'm most interested in. Here you see a mantis shrimp's compound eye, and this very specialized middle band region is what I spent much of my PhD working out. What does this part of the eye do? I was very interested in these animals because they're very colorful and wondered whether or not they had color vision. Let me remind you that color vision requires at least 2 photoreceptor types in the retina. We have three. We have red, green, and blue photoreceptors or cones in our eyes. Cats only have two. They can still see color. But we use our trichromatic color vision to spot things like ripening fruit in green trees. Mantis shrimps, it turns out, have 12 color channels. Why? We still don't quite know, but we're beginning to realize that they do color vision in ways which are completely different to other animals. As well as color vision, mantis shrimps and indeed other crustaceans also have polarized light vision. They can see polarized light without using sunglasses. Mantis shrimps, of course, are very special. And as well as having linear polarization vision, they have circular polarization vision. And without going into details, what they do is convert incoming circular polarization to linear, which a specific part of their retina can detect. Why? They use it for mate choice because males reflect circular polarization, females don't. And these animals, the females, are probably judging the male on the quality of this circular polarization reflection. Fortunately, if you like, we can use the polarized light reception of mantis shrimps in a number of bio-inspired sensors, and we can use these in several application areas. This includes increasing the storage on DVDs and CDs, early cancer detection in terms of skin cancer, for example, redesigning satellites to look back at the Earth better than they currently do, and GPS-denied navigation and geolocation on the surface of the Earth. These are nice knock-on effects from the research that we do on animals and animal vision. Another animal that we work on or another set of animals are the cephalopods, the squid, octopus, and cuttlefish. And these guys also have polarized light vision, but they don't have color vision. Therefore, how do they do this? How do they do what this octopus is about to show you? Where's the octopus? There's the octopus. It can camouflage itself or communicate. It's unhappy at the moment. It went white to communicate to the cameraman that it's been seen. How does it do that without color vision? We are delving into the brains of the cephalopods using MRI, magnetic resonance imaging, and looking at the connections from the optic lobes into the central brain and trying to work out how these invertebrates with the largest brains of all the invertebrates, how are they doing vision? So again, getting into the head of other animals and working out how they see the world. We're very interested in how close neighbors, such as the fish that use color, close neighbors to stomatopods that live on reefs, how are these animals seeing the world also? And it turns out that they also have surprises. So many of the reef fish see the world in ultraviolet. They have ultraviolet patterns that we can't see. Here's how we see this fish. Here's how it might appear to the same fish, which does have ultraviolet vision. So again, we're interpreting the visual world of animals from the perspective of that animal and using special cameras. And it was lucky that Sir David Attenborough also got interested in how animals are seeing color and recently produced Life in Color, a 3-part documentary to examine this. And we helped Sir David and his production team by providing the cameras to look at ultraviolet patterns in the real world and polarization patterns in the real world that other animals see and we don't. I also worked with Sir David and his team on a documentary a few years ago that looked at the Great Barrier Reef and went down in submersibles with Sir David, guided him around the reef and talked about problems that the reef is facing. These problems became apparent to me 20 or so years ago when I began to see the colors of the reef changing and came up with this small color chart which enables anyone to measure the color of coral, and that tells us the health status of the coral. And this is a citizen science project. It allows anyone who can see color to go to the reef, put this against a coral, and send us back data which tells us about the reef that they're close to and how it's faring. So Coral Watch is a citizen science group now active in 137 countries with over 9,000 members. And really the question that many people ask because they all know as soon as they begin to look at the reef that it's disappearing is, well, will my grandchildren get to see a reef? What's happening? Now, what can I do? And as a citizen science group, we try and answer that by giving them the facts, giving them fact sheets about what's going on reefs worldwide. What are the main threats? What is coral bleaching? We suggest ways in which they can help, even at home, by all of these things that we know, examining the way that you get to work, look at your food waste, energy use, all these things which are increasing carbon emissions and causing global climate change. We're actually in the middle of publishing a book called What Can I Do?, encouraging people to help save the reef from home by looking at 7 activities that they might do and realizing that these activities will benefit them in many ways. Of course, the Australian Academy of Science is also heavily engaged in educating people about the risks of climate change and leading the way with several publications. I'd also like to suggest that there are other ways that the Australian Academy of Science could approach the public. There are many fellows that focus on aspects of climate change and are great communicators of science in their own institutions. I'd like to suggest that we need to make this more personalized. And here is a suggestion for how we could do that.
Hello, my name's Justin Marshall. I'm a neuroscientist and marine biologist at the University of Queensland. I'm also the founder and director of Coral Watch, a citizen science group that helps people understand what's happening on reefs worldwide. Reefs are diminishing, and we all need to take action to help save the reef from home. Here's me taking action by riding my bicycle to work. Reefs are disappearing because of climate change, a problem that can seem insurmountable. However, there is hope, and hope lies with you, individuals that are concerned about not only the reef, but the rest of nature worldwide. We can make a difference, and we can reverse climate change. Don't feel helpless, because as an individual, you can affect change. It's always been large groups of individuals that have made things change in the past, so we can do it. And remember, as well as actions such as cycling to work, which help combat climate change and provide a better future for our children, there are many other things that you can do, as suggested by other fellows of the Australian Academy of Science.
If that is, you'd like to join me in providing personal messages, either on your own websites, or perhaps the Australian Academy of Science would consider a collection of these, in which individual fellows suggest ways in which the public could help combat climate change and show their own choices in their own personal lives, as well as providing the science to guide people through to a better future. Many thanks for your attention, and I'm very happy to take any questions.
And now we're delighted to have with us Professor Justin Marshall. Congratulations, Justin, and thank you for your presentation.
Yeah, well, thank you. Thank you for being there. Thank you for those of you that have stuck it out through the day and are still listening, and hopefully we can move forward.
Absolutely. And Justin, might I also say to your suggestion at the end in terms of educating and reaching out to the public, I would love to talk to you further about those ideas. I'm very passionate in that space as well. So we'll be in touch. I'd love to start by asking you, what led you to study how other animals see the world?
I've always been fascinated by animals underwater. My parents were actually marine scientists, and they took me around the world and showed me life underwater. I just started asking questions about, what does a fish see? And then as I did a degree in zoology at St Andrews University in Scotland, I began to be introduced to other, invertebrates, lobsters, and my PhD was on those amazing stomatopods or mantis shrimps that have this extraordinary visual ability, which is at first sight seems to be so much better than ours, but it's clearly just very different. And it's always been one of these things where humans assume that they're the best, we're the pinnacle of evolution. In fact, we're very colorblind compared to many animals. And that fascinates me.
Yeah, it really is fascinating. I'd love to dig into some of your other parts of your presentation. I have just received a question sent in from Professor Frances Saparovich. She comments, lovely work, Justin. would these polarized light detectors in cephalopods or crustaceans see or react differently to a solution of L or D molecules?
Almost certainly, given that they will emit or transmit different degrees and angles of polarization, depending on how you hold them. But yes, they would. And that's part of what we're looking at, is how, or what the animals actually respond to. And they respond to these amazing reflections from themselves, which are set up through a number of polarization production mechanisms, including molecular.
Yeah, wonderful. Now, I want to ask you, how do bio-inspired innovations come about, such as the satellites? I love the reference to prawns in space and cancer screening technology examples. Does this come from interdisciplinary scientists reading your papers, or do you approach potential collaborators?
A little bit of both. The Prawns in Space project was actually an Australian Research Council linkage project. And there we tried to link with satellite engineers and speak directly to them. And we got some of the way with that. It's quite interesting when you mix biology with engineering. Anyone who's tried to do that will recognize the hurdles that you have to overcome. And then, of course, when we publish papers, there are people that that jump on the suggested technology. So I'm very much in the space of suggesting the technology rather than exploiting it. So, for example, after we published the paper on the quarter wavelength retarder in the mantis shrimp's eye, which is physics, a group in Taiwan had produced that within 2 weeks of the paper appearing. So a bit of both.
Yeah, fascinating. I can see how it would work both ways. I'd love to ask you as well about the mantis shrimps. Do you have any theories on why they've evolved to have such complex visual systems?
I have. It would take a bit more than a minute or 30 seconds that's left to explain that, but basically they're doing their visual input very differently in ways which involve sparse coding. So one of the problems that we face now is we cram too much information into our satellites and our input and our camera systems, which basically causes a log jam. Many invertebrates, many insects, crustaceans, sort this out at the front end of their sensory systems, if you like. And mantis shrimps are doing this with a scanning system, which is much more like a satellite. And they get rid of the extraneous information which is not needed right up front to enable their relatively small brains to make rapid and smart decisions based on whatever they need to decide upon.
And just to finish up on where you ended in your presentation, talking about human impact on climate change, often people might feel disillusioned that their own actions might not make a difference. What inspired you to make that message? And is there anything further that you'd like to say to someone who might feel they can't make a difference?
Absolutely. I mean, I think that, the thing that inspired me was actually the people themselves. So I spent the last 20, 25 years working with the public and people that are interested in reefs, whether they can get to a reef or not, school kids. And these people have inspired me because they just won't give up. They really want the reef to be there in the future, as we all do. And they see that as a personal challenge, a challenge for local government, a challenge for larger society, and of course, a challenge for the international community. So it's this multi-scale approach that we need to take now. So I would encourage anyone who's feeling disillusioned, all right, spend some time wallowing in the nasty numbers and the facts that you see from the Academy and other papers, but then go, I am going to solve it because we can. And the only way this has been done in the past is by groups of individuals getting together and doing what's necessary and acting now.
Thank you. Very well said, Justin. Thank you once again for your time today and congratulations.
My pleasure. Great to be with you.
Now, please welcome back Professor Malcolm Sambridge to introduce our next fellow.
Thank you. Next, we will hear from Professor Hala Sveikat. Hala has made significant contributions to biomaterials, regenerative medicine, and translational orthopedic research. She introduced advanced cell molecular biology and nanoscience approaches to the biomaterials field. Her achievements are leading to the development of novel orthopedic implants, including the world's first synthetic material for healing large bone defects under load, enabling personalized approaches to the repair and regeneration of musculoskeletal tissues. And now to Hala's presentation.
Good afternoon, everyone. I am honored to be inducted as a Fellow of the Australian Academy of Sciences, to be recognized among my peers, esteemed cohort of Australians who are making extraordinary contributions to the scientific community. As I make this presentation, I would like to declare a conflict of interest related to my work with Allegra Orthopedics on one of the materials developed that I will be talking about in this presentation. Our work involved looking into strategies to regenerate musculoskeletal tissues, and this will encompass the development, design, and manufacturing of synthetic materials and scaffolds that mimic the tissue structure, and then also the biological enhancements of synthetic material. And maybe then we can end up with a new material that encompasses both the synthetic material and the material with the biochemical or cellular cues. The synthetic material I will be describing here are made of ceramics, like the cup of china, but we introduced into them trace elements like zinc, magnesium, and strontium that transform the base ceramic material and make it into a material that we can use to regrow bone. We chose ceramic as an alternative to other materials such as metals, which is currently used in the clinic. We then use these ceramics into two ways. Either we use them to plasma spray or spray them into clinically used orthopedic implants like metals, or we make them into 3D printed scaffolds or personalized implants. The tissue we're looking at regenerating is bone. As we know, bone is an amazing tissue. It has the same strength as cast iron, but achieves this while maintaining as light as wood. It has a unique structure depending upon the demands or the mechanical demands on it. So we have the dense solid bone where stiffness and strength is required, and we have the spongy open structure of bone where shock absorbance is required. So within the past decades, various inorganic materials have been used, and these listed here are all used in the clinic right now. The problem remains to be that none of them have the optimal bioactivity needed to regenerate bone. Most importantly, they are not mechanically strength for regenerating large bone defects under load. So the need for development of new bone substitute really stems from the fact that diseases or defects like that, where you have large bone defects in the jaw or in the femur or in the spine, are very hard to regenerate at the moment. We don't have the right technology to regenerate these defects. So scientific laboratories often feels a long way away from the people we're trying to help in our research. However, every now and then the stars aligns and something very special happens to remind us that the hard work and the long hours we spend in the lab definitely are worthwhile. I had one of those experiences exactly 3 years ago now, and I want to share it with you. Before I delve into the research, I would like to share with you a short video that illustrates the medical need for effective bone regeneration. I was a Radcliffe Fellow at Harvard University back in 2018, and while their undergraduate students from Harvard applied to work on a project with me at the time, it was on 3D printing of synthetic bone substitute. 3D printing considered a sexy subject, so I had a number of applicants applying to work with me. However, the CV of one applicant stood out, and I asked her to come into my office for an interview. Little I knew that this young lady was coming into my office with crutches. And what I would like to share with you now is the video that illustrates the medical need for effective bone regeneration. So
As Hala already mentioned, I had osteosarcoma when I was 10, which is a form of bone cancer. There was an extra growth in the bone, and the doctor had to do an operation to remove that section of bone, and that leaves the question of what could be put into that gap. And what they did back then in 2006 was to get bone from a bone bank and substitute it into the gap where my original bone was taken out, and putting a metal plate to hold everything together. There were many complications with that original bone graft because it was not my own bone, so there were problems with healing and various complications with the metal parts coming apart. So I've had to go through a lot of surgeries, 10 surgeries in total over the past 10 years since I was first diagnosed with osteosarcoma. So I felt that Hala's project is really meaningful in the sense that it negates the need for metal for patients that have bone defects.
So Lynn isn't alone. Of course, huge number of people are affected by skeletal issues. As you can see from this slide here, the size of the global market for orthopedic interventions spans between spine, knee, trauma, and hip, with a total market of around $48 billion worldwide. And despite the enormous financial burden of these cases, we still do not have available treatment that works properly for people like Lynn. So this has been a real challenge in the field. And if we look at developing synthetic material for bone regeneration, we know that this material has to have the matching stiffness, strength, and toughness of that of the replaced tissue, which is bone. Otherwise, it will break under physiological load. And of course, then no bone will grow. So the approach we have adapted in our lab is to develop the solutions by developing a synthetic material that have the appropriate microstructural design that imparts strength and toughness, but also the chemical composition to impart bioactivity to induce bone formation. And over the past decade, we have developed and patented a number of ceramics. Today, I will talk about the material number 3 here, which is a calcium silicate material that has strontium and zinc in it. And this material is now with a company called Allegra, an Australian orthopedic company. And this material has progressed to large preclinical animal model studies. Using this material, we 3D printed it, and we were able to develop a scaffold that's highly porous, highly interconnected, that for the first time now, this ceramic has the mechanical strength of that of cortical bone. We then took it into the preclinical animal model, and what we see here is that we implanted in a large defect, as is no cells, no growth factor, using a recommended orthopedic animal model. At 3 months, we can appreciate that the scaffold, the white struts here, maintained their mechanical properties. And if we look at bone, bone is this color here, infiltrating from the edges of the defect at 3 months, abutting the struts of the ceramic. And by 1 year, we can see the defects completely filled with brand new bone forming. So it is important for us to take it to the clinic to treat people like that of Lynn. And here we the IP for this material, or the intellectual property, was assigned to Allegra Orthopedic last year in return to equity to the university. And the company will then seek regulatory body approvals from different countries, conduct clinical testing and manufacturing before it's ready to go to market. We now have developed the next generation of the spine fusion devices ready for clinical testing, supported by NHMRC Development Grant and Biotech Horizon Grant. The problem remains is, can we regenerate complex defects of bone? And this remains to be a challenge, but we have developed the 3D printing technology that allows us now for the first time to print materials of complex structure to impart a stiffness, strength, and toughness required for the particular defect, no matter what size or shape that is. Of course, this will allow us now to produce different shapes and sizes of these materials. We now have collaboration with ETH Zurich and Zurich University, combining both technologies to provide the next generation of materials for cartilage and bone regeneration, osteochondral regeneration, and other synthetic material that we have developed is synthetic tendon and ligament for the regeneration of tendon and ligament. And we just received an Australian Research Council linkage grant to take this to the next level. At the ARC Center, we have a number of research going on, and I would be delighted to work with my fellow academies to progress our research to the next level. With that, I would like to thank all the funding bodies and the past, present, and future members of the lab, and of course, the Australian Academy of Science. Thank you.
And please welcome, joining me live now, is Professor Hala Zrakat. Welcome, Hala. Congratulations. And what incredible work that is literally changing lives.
Thank you very much. Thank you for that.
Now, we have some questions for you. Given your work spans research, development, design, and manufacturing, how do each of these different stages inform each other in a cycle?
I think it's all interconnected. Really, it starts from day one when you start an idea and you start a project to develop it. And I think having had the success we have had in securing funding from the government, we owe it to the taxpayers to repay what we have done. And not always we can do that, but our research is informed into the translation. So there are research that takes a lot of time, fundamental basic research, which is mostly important. And we know the outcome of that in the vaccination that we have had now. But this research, if we do it right, we can get to the translation a bit quicker than basic biology. And I think once you start developing things, the idea comes to mind. First of all, I am a biologist, I'm not an engineer. And quite often people think that I'm a ceramic or anything like that, but I'm far from it. But it's the idea that comes to mind, and then you surround yourself by the appropriate people to help inform the execution of that idea. And that will include clinicians, that will include industry, to take it to the next level, lawyers to file IP. The reason we file IP is because we want to take it to the clinic. And without companies, we cannot take it to the clinic, and they need IP. So
it's really inspiring work, and I think a great example of how all these different fields and disciplines can work together to bring about such an effective outcome. Considering the rapid development of 3D printing technology and its increasing accessibility by consumers, what do you see as the future of patient-designed orthopedic products?
Yeah, I think it's very, very close. It's there really. And what I think the problem here, and we're writing a paper about that, is not to develop a personalized implant, is to get the approval of the regulatory bodies to really change their long regulations that they've had over the past 50 years to now start looking into accepting single devices for single patients to go into the approval process, because they have certain designs based on the fact we couldn't do personalized implants, but there was an umbrella, if you like, of approval for certain designs, but that is not what personalized implants is. So I think the FDAs and the regulatory bodies are trying now to adapt to the new technologies that will allow us to come to a personalized implant.
And Hala, I'd love to spend the afternoon asking you more questions, but I will just ask you one final one. What does this new approach mean for patients? Is the recovery time shorter? Is the result more robust and long-lasting?
We're hoping so. I mean, we have not gone to the clinic. We were delayed with the COVID, with the preclinical studies, but definitely, you don't, for a start, if you cannot use metals and use a ceramic that becomes part of the body and dissolve and degrades, that's a big plus. Less operations, less funding spent, and less recovery time.
Well, I'm sure we're all excited to see where your research goes next. Hala, once again, congratulations and thank you for your time this afternoon.
Thank you.
Now, please welcome back Professor Helene Marsh to introduce our next fellow.
Professor Gordon Smythe is our next newly elected fellow. Gordon is a statistician and bioinformatician with long-standing interest in nonlinear estimation, dispersion estimation, and algorithm development. He's made influential contributions to the analysis of genomic data, especially to linear modelling and empirical Bayes methods for the analysis of gene expression experiments. Together with collaborators, he's used genomic data to make important discoveries relevant to breast cancer, malaria, and other diseases. We'll now hear from Gordon.
Thanks very much to the Australian Academy of Science for the opportunity to talk today about my work in statistics and bioinformatics. For the first half of my career, I was a mathematical statistician working on statistical modelling and statistical computing, and particularly on generalised linear models, which gives one the ability to model both normal and non-normal data, including counts, in a very consistent way. So I eventually wrote a textbook on this topic with Peter Dunn, who was a PhD student of mine at one time. So one of my particular interests was the modelling of dispersion or variability in generalised linear models, and especially modelling dispersion as a function of extra parameters. So this approach works for both non-normal and non-normal data. One of the key concerns is that dispersion estimates are naturally biased due to the fact that the systematic part of the model, the location model, is estimated from the same data. So a key concern is to adjust the estimation methods to account for the fact that the location model has been estimated from the same model. So another ongoing concern of mine has been numerical issues. And so I've spent a lot of time developing efficient computational algorithms to fit these statistical models. So all of this was mathematics, but it proved to be really useful in 2001 when the human genome was published, and I left my first career as a mathematician to join the Walter and Eliza Hall Institute of Medical Research in Melbourne as a medical researcher. So I then became part of the modern research effort in genomic research. So 20 years ago, the new technology for measuring gene expression were microarrays. And this is a picture of a microarray slide, much larger than reality. Each dot represents a gene. The microarrays give the ability to measure the expression level of every single gene in the genome simultaneously for the same biological samples. So genomic data, because you're measuring tens of thousands of genes, genomic data is massively parallel. And meanwhile, we're working with tiny sample sizes. So we might, for example, be comparing 3 mutant mice to 3 wild-type mice, or perhaps even just 2 mice in each group. So we're doing tens of thousands of statistical tests with really, really small sample sizes. So traditional statistical tests don't work at all in this context. They give very poor results. We need new statistical strategies that are able to borrow information between genes, to take advantage of the parallel nature of the genomic data and to learn from the whole ensemble of genes. So this is just a simulation to show how much difference that makes. So on the left, I've got differential expression results using ordinary t-tests. The x-axis is log two-fold change and the vertical axis is log p-value. And the blue line shows the 5% false discovery rate significance cutoff. And for the ordinary t-test, most of the truly differential genes do not reach the significance cutoff. But if we use empirical Bayes t-test instead of the ordinary t-test, almost all of the truly differential genes are now well above the 5% false discovery rate significance cutoff. It's really an enormous increase in statistical power and reliability. This can be applied to all our technologies. So a little over 10 years ago, next generation sequencing technologies arrived, and which revolutionized the whole field again. So now we have RNA-seq, which is able to measure gene expression in all of its forms, and DNA sequencing combined with different genome technologies allow us to learn about all the different mechanisms of gene regulation. For example, microRNAs or ChIP-seq and cut and tag allow us to learn about epigenetic marks. Bisulfite sequencing tells us about DNA methylation. ATAC-seq identifies open and closed DNA regions. And HiSeq allows us to identify different parts of the genome which are touching one another, allowing us to learn about DNA folds in the 3D structure of DNA. So over the years, I developed with my group a number of software packages to analyze this sort of data, starting with the Lima and EDGAR packages. And together, these packages are downloaded over 800,000 times a year, and the journal articles associated with them are cited over 10,000 times a year. So one of our core activities is to identify differentially expressed genes which are significantly up or significantly downregulated for any given comparison. But we're also interested in higher level processes, high level biological processes. So one of our favorite techniques is to use specially designed experiments to identify gene expression signatures for particular biological processes. Then we can look for those signatures in new data sets, for example, in cancer samples. So as I say, Hi-C is able to measure DNA-DNA interactions. So we can identify, and in this way, we can identify genomic regions which are quite far away from a given gene, which are associated with the promoter of that gene and which help that gene become expressed. And then we can look for changes in those enhancer patterns between cell types and experimental conditions. See, in recent years, we've given more and more attention to single cell processing. So here I'm just showing an example where we look at the development of the mammary epithelial from embryo to pre-puberty to puberty. And can we see that in the embryo, the mammary cells are virtually all of primordial basal type, but over from pre-puberty and then to puberty, the different cell lineages in the mammary gland develop, and by puberty, we have a fully developed set of cell lineages. So one of our interests is to identify when these cell lineages emerge. In recent years, so over time, I've had a big interest with my collaborators, Jane Visvader and Jeff Lindemann, in identifying the cell of origin for different cancer subtypes. And over the last 7 years, I've been very pleased to be able to to be head, or more recently joint head, for the bioinformatics division at the WEHI. This is what the division looked like 7 years ago when I became head. We've been able to expand the division quite a bit since then. So thank you very much.
Now, we're delighted to have live with us Professor Gordon Smythe. Congratulations, Gordon, and thanks for joining us.
Okay, thanks very much.
Can I start by asking you what attracted you to statistical modelling and computing in your early career?
That's an interesting question. Well, I was interested in mathematics, but I was also interested in science. So when I first went to university, I hadn't really heard of statistics, which I think was a good thing. But somehow statistics and statistical modelling has allowed that synergy of science and mathematics together.
Well, I wonder how much you drew on that with your shift to WEHI later in your career. It's impressive to see the application of mathematics in other science settings, in your case, biology. Did you have a strong interest in biology prior to your move to WEHI?
I had a little interest in biology, but I hadn't, prior to moving to the WEHI, I'd had an ongoing interest in scientific applications, but it was all over the place. A little bit of microbiology, a little bit of plant pathology, a little bit of criminology, a little bit of engineering, but I really wanted to commit full time to one particular area, to medical research. So I actually, when I moved to the WEHI, I didn't have any track record in medicine at all. They took a chance on me.
Sometimes the best results can come from those chances taken, can't they? So
I was very confident about that. I knew how to interact with scientists, but it was a new field for me.
Yeah, it must have been a huge thrill. Tell me a bit about the early discoveries or things that you had to grapple with in making the jump from statistical modelling and computing to dealing with genomic data.
Well, modern genomic medicine is really exciting because they're just generating, because new technologies come along with just very regularly, and each new technology brings a new data type with it, which then becomes the task of the statistician and the bioinformatician to come to grips with. So I actually feel, so I still supervise students and staff from mathematical backgrounds, and I actually feel that I do more mathematics in a medical research institute than I used to do in a maths department, because there's just so much more data. In a maths department, I would sort of work on a maths problem and then wonder who will ever use this. Whereas in the medical research scheme, they're saying, well, have you figured out how to do this yet? Have you figured out how to do this yet? So it's where we have the proverbial avalanche of data.
Another of your fellows, Professor David Balding, has sent in a comment and a question. Congratulations, Gordon. The t-test versus empirical Bayes example seems almost too good to be true. What information are you sharing over genes to obtain this huge advantage?
Well, what's specifically been shared is information about the variability, because the t-test assumes that we know nothing about the precision of the expression estimates, of the gene activity estimates, but in fact, not every gene has the same variability, but there's a range of possible variability. So it's that. So we learn something about the underlying precision of the data from the other genes, and that's what's used in the inference about each individual gene. It makes a big difference. So genomic data. So I was interested in this idea of indirect inference, of using information from related data sets before I moved into medical research. But medical research genomic research is the perfect application for that sort of idea. It was sort of the application that those mathematical methods were waiting for decades, as it was.
And you shared as well the team you've had at the WeHi 7 years ago, which has now grown. What about the field itself? Do you have many international collaborations as well?
Well, we have international collaborations. It's bioinformatics is certainly a growing field because genomic data becomes a larger and larger part of medical research. My group is particularly statistical bioinformatics, which is also a growing niche. I mean, it's been it's been interesting over the last 20 years. We were sort of, 20 years ago, we were like, in a way, the only bioinformatics centre or the only statistical bioinformatics centre. Now those centres have grown at lots of other medical research institutes around Australia, and I think we've played a role in seeding some of those groups and showing what can be done.
Well, that's very exciting, and congratulations on your work and also becoming a Fellow of the Academy of Science, Gordon.
Thanks very much.
Well, that now brings us to the end of our first day of presentations by fellows elected to the Australian Academy of Science in 2020 and 2021. Thank you all for joining us and for all the questions throughout the day. We hope you've enjoyed hearing from our fellows. Tomorrow, we'll continue to hear from more of our newly elected fellows, and we hope that you'll join us then. And don't forget that the celebration of science continues tonight with the Prime Minister's Prize for Science. Until then, I'm Paul Richards. Good afternoon.